Welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Welcome everyone to our Advancing Generic Drug Development Workshop, Translating Science to Approval. This is our 6th year of having the privilege to collaborate with CDER's Office of Generic Drugs and its Office of Research and Standards to produce this workshop. Good day, good afternoon, or good night, depending on where you may be. It is extremely exciting to see so many of you online with us here today. And we certainly appreciate your commitment in attending this event and meeting and overcoming any scheduling challenges you may have experienced. We invite you to chat amongst yourselves in the chat pod when it is available. And I see, from earlier this morning, we already have wide global representation. What an encouragement to see such lively participation. My name is Brenda Stoddard, and I'm with the Small Business and Industry Assistance SBIA program, which operates within the Division of Drug Information, which in turn resides in FDA's Center for Drug Evaluation and Research's Office of Communications. It is my great pleasure, along with my CEDA colleagues, to be with you here today. And this workshop will feature an unsurpassed cast of subject matter experts from across the Office of Generic Drugs, the Office of Pharmaceutical Quality, and the Office of Translational Sciences. We entreat you to make the most of this experience. Attend all the sessions that interest you, learn all that you can learn, submit your questions during the available Q&A sessions, and network in the chat pod. In addition to the Adobe Connect platform, we are offering live YouTube streaming, so please go ahead and invite your colleagues to attend as well. However, please note that only those attendees who register and attend via Adobe Connect and who log into Room 1 will be able to submit questions to our speakers. At this point, I'm very pleased to say that we have for this two-day workshop in its entirety, we will offer a total of 13.25 contact hours to physicians, pharmacists, and nurses. The CEs are available to all real-time attendees who self-identified as healthcare professionals during the Adobe Connect registration process and who attend via Adobe. Please note the emphasis on real-time and on Adobe Connect. No CEs can be given for those who attend via YouTube or for post-event viewing. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single supporting company. CE faculty speakers are required to disclose to the attendees when products or procedures being discussed are off-label, unlabeled, not FDA approved, and any limitations in the information that is presented. We have a spectacular lineup of speakers, and all of the relevant financial relationships listed for these individuals have been mitigated. Dr. Robert Califf has various financial relationships, and his financial disclosure report and ethics agreement are publicly available. Dr. Susan Bach discloses that she holds individual stocks with the Honest Company, Inc., Johnson & Johnson, and Target Corporation. The planning committee, speakers, and CE consultation and accreditation team have nothing to disclose. Participants must attest to their attendance and complete the final activity evaluation via the CE portal. That's ceportal.fda.gov. For multi-day activities such as this one, participants must attest to their attendance and complete the faculty evaluation for each day. Final activity evaluations must be completed within 2 weeks after the activity. So, in this case, the deadline for this two-day workshop is September 28th. And we apologize in advance, but we are unable to make exceptions to that deadline. So when you get that email today and tomorrow, please act upon it immediately and submit it. Pharmacists should note that failure to provide your correct NABP and date of birth information in the required format may result in the loss of credit for this activity. Your NABP profile number should be the 6 to 7-digit profile number assigned by the CPE monitor, and your birth date should be in the month, date format, example 0913. Do not provide your pharmacy license number. Please click the My Account tab and then navigate to Edit Contact Information to verify that your information is correct. Physicians and nurses may then view and print their statement of credit. Pharmacists should log into the CPE monitor 8 weeks after the last session of the activity to obtain their CE. It could be ready before 8 weeks, but definitely not after 1 week. So please allow sufficient time for that to occur. Now, as we set up for this poll, which I try to do every conference, I know that some of you may have been with SBIA for quite some time, perhaps even going back as far as 2010 when we first started, although I dare say that many of you are also first-time attendees. A special welcome to you as we work towards expanding our audience base and increasing our portfolio of SBIA events. And I see some of you, many of you are ahead of the game here. So to break the ice and get us thinking, we have the following question. Please participate if you can. I see the numbers are increasing. The question is, how many SBIA conferences have you attended? And the choices are, this is my first one, 2 to 5, 6 to 10, more than 10, and most since 2010. Let's see if we settle down here a little bit. Couple more seconds. Okay, it seems that 23% of you in the audience, this is your first experience with SBIA. So you chose a great event to attend as your introduction to SBIA. That's, not saying that all of our events are great. And next time you'll be able to select B. And for those of you who indicated that you've been with us before, and 3% indicated that they have been with us almost since 2010. Thank you so very much for your continued support to all those who have been with us before. Excellent, thank you. Okay, now these numbers have risen since this slide was made, and as of this morning, we have over 3,000 registrants representing 86 countries. Over 42% of these registrants have self-identified as small businesses, and of those, over 24% report having 1 to 9 employees. These stats continue to be extremely important to us here at the SBIA program, as they confirm that we are indeed reaching the small business sector of our audience, and they also validate our mission and the very existence of the program itself. This, what you see in here, is a snapshot of a geo map for today's event. And this map is always quite invigorating to us here at FDA, for it provides a very powerful visual as to the impact and the extent of our outreach. And you can have fun with this. You can actually click on the image, which will take you to your browser, where you can zoom in and have a closer look at our global participation. And I invite you to try it. Zoom in on your geographic area and see if anyone else is attending from your neighborhood. Moving on, this slide here provides the QR codes for our social media avenues. You can scan them with your phones and be taken to the appropriate platform. They will also be advertised across the screen banner and during the breaks if you miss it now. So let's turn our attention to some logistics. Again, this event is being offered 100% virtual. And over the next 2 days, we will have a total of 55 speakers and panelists, not to mention our own organizers and technical staff, all working from our homes. As such, there may be some internet issues, but please bear with us. And if in fact we do encounter those, we will try to get them resolved as quickly as possible. And speaking of tech issues, here is the link for a one-stop shop for answers to your technical questions, for downloading the files, accessing the survey link to obtain the certificate of attendance, and so much more. It's the same page that you used this morning for your online access to the event, so please bookmark that page as you'll need it over the next 2 days. If you have any technical questions about this conference, please check there first, and you will most likely find your answer. Again, we invite you to submit your questions to us so that we have a vibrant and productive Q&A segment. And that is so crucial to these events, because your questions and the answers provided will benefit the entire audience. This is a learning opportunity, and we encourage you to take full advantage of the expertise we offer here. We will answer as many questions as time allows. And when you do submit your question, please identify that individual presenter by prefacing your question with the speaker's name as shown here. Also, during each presentation, there is a link on the right side of the pod where you'll see the speaker's headshot or signature block, and it says download the slides. Please note that this link is only active during the real-time presentation. There is also a link to speaker evaluation, and we encourage you to take the time to complete that, as your feedback is much appreciated by the individual speakers. In addition, we also have our final event evaluation, and upon completion of this survey, an attendee can then download a certificate of attendance, which may be used in support of RAPS, SOCRA, ACRP, and SQA credits. Now, the trick is that the certificate must be downloaded immediately after completing the survey. Otherwise, that opportunity is lost. And even if you do not want or you do not need a certificate, please take a few minutes to complete the conference evaluation, as your feedback will help guide the design of our future events. Note that both the evaluation and certificate are only available for 2 weeks, which means the deadline date for that also is September 28th. The survey link will be posted on our one-stop shop page the afternoon of each day's event. And as a reminder, all the sessions are being recorded. It will take us a week or two perhaps to process the recordings, but you will soon be able to access them from our SBIA's homepage, which I hope you have already bookmarked, and that's fda.gov/CEDAR SBIA. But in the interim, the YouTube link for each day's recording will be posted on the One Stop Shop webpage at the end of the day. Now, I know I've said a lot, and to simplify things, each night, SBIA will send one email to the attendees of that individual day's event. That email will have information on both the Healthcare Professional CE claim code and the conference evaluation link. And please remember, there are two different processes. Okay, let's take a couple minutes to highlight our upcoming events. September 18th and 19th, we'll host our use of biomarkers for diagnosing and assessing treatment response in non-cirrhotic NASH trials. And this workshop will assist the FDA in identifying current knowledge gaps for using non-invasive tests as diagnostic biomarkers and reasonably likely surrogates, as well as provide a framework for additional data that are needed to fill these knowledge gaps. On September 28th, we invite you to our annual electronic drug registration and listing workshop using CEDAR Direct. Here, we will provide basic instruction in the registration and listing policy and process for those who are new to this regulatory program, as well as offer regulatory professionals more in-depth information on issues and current events affecting registration and listing. On October the 11th, we'll host our 2023 Nano Day Symposium, Continuous Manufacturing of Nanomaterials. And during this event, FDA will provide an overview of CEDAR experience with approving several solid oral drug products, which benefited from the continuous manufacturing technologies. On October 16th, we'll have a webinar on the GDUFA III Regulatory Science Pilot Program. And note this is a two-part meeting, with the webinar, the virtual part, being held on October 16th, and the in-person part being held on October 26th. Detailed information is posted on our website. Then on October 31st and November 1st, we will be hosting our biannual Pharmaceutical Quality Symposium, where we will explore topics related to pharmaceutical quality regulation, supply chains, and advanced manufacturing technologies. More information is available on our homepage, seen here. And we also invite you to visit our CEDAR Learn webpage, where you can link to SBIA tutorials along with other educational resources, many of which offer healthcare professionals CEs. This slide calls your attention to a short video which will give you an idea of the SBIA resources available to you, and it will be available for you to play during the lunch break. So don't miss out on that. And at this point, I would like to acknowledge those individuals instrumental in making this event possible. Many are listed here, but so many more deserve credit. Special thanks to Maria Monroy Osorio, who led the coordination of all the details for this event, to our CE consultation and accreditation team, but most of all, our heartfelt gratitude to you who took time out to attend this event, again, regardless of the inconvenience of the various time zones. And I want you to know that we do not take this commitment lightly, for without you, this conference would not be possible, and definitely would not be the success that it is. And, you know, it contributes to the existence and longevity of the SBIA program. A very special recognition and introduction to my SBIA colleague, Captain Ray Ford, who will be your SBIA host over the next 2 days. And now, as I get ready to close this introduction, I would like you, the very early birds, those who are with us at this point in time, to be the first to know that we are planning on holding this conference in a hybrid format for 2024. That means we will have an in-person option, and we will be looking forward to meeting many of you face-to-face again. So please stay tuned to that. But now, I'm sure you're ready for us to launch fully into today's agenda. And as a reminder, our speaker bios are all available on the One Stop Shop website. But for now, I am honored to introduce our keynote speaker. Dr. Robert M. Califf is the Commissioner of Food and Drugs, and as the top official of the FDA, he is committed to strengthening programs and policies that enable the agency to carry out its mission to protect and promote the public health. Previously, Dr. Califf served as Commissioner of Food and Drugs from February 2016 to January 2017. Dr. Califf also served as the FDA's Deputy Commissioner for Medical Products and Tobacco from February 2015 until his first appointment as Commissioner in February 2016. Please, let us welcome Dr. Califf.
Thank you, Brenda. I'm pleased to once again be a part of this important workshop organized by the Office of Generic Drugs and the Office of Pharmaceutical Quality in collaboration with the Office of Small Business and Industry Assistance. There are a few things as important to our work in healthcare as stimulating and supporting innovation in generic drug development. To this end, I also want to thank the many developers and other stakeholders who joined today's discussion, as well as the support provided by industry to the generic drug development program to help make the program possible. The word innovation is an important one, and while there's sometimes a tendency to overuse it, when it comes to generic products, I think that's an extremely appropriate description and aspect of our work in supporting advances in this industry. Certainly, the impact and benefits of generic products and the competition they produce to help lower costs and increase accessibility for patients cannot be overstated. According to the Association for Accessible Medicines, in 2021, the healthcare system saved $366 billion thanks to generic prescription medicines. Americans clearly appreciate and take advantage of these savings. Generics today represent 91% of prescriptions filled, accounting for 18% of all prescription drug spending. The value that patients place on generics is for good reason. They provide access to affordable, safe, effective, and high-quality medicines. This enables patients to obtain effective treatment at a low cost, clearly a public health priority for the FDA. We're taking numerous actions to support the generics program. For example, OGD has continued its implementation of the FDA's Drug Competition Action Plan, or DCAP. Last October, we published draft guidances and product-specific guidances that provided recommendations for physicochemical and structural characterization tests, in vitro release test studies, and in vitro permeation test studies for topical generic drug products. These guidances will be discussed in today's workshop. One of the key aspects of our work to promote innovation in the generic drug space is our engagement with developers. Early communications can help reduce a generic drug product's time in the pipeline from concept to development to market input by strengthening development programs, improving FDA assessment efficiency, and enabling better ANDA submissions. In FY 2023, the FDA held numerous additional scientific meetings with industry as part of the first year of the second reauthorization of the Generic Drug User Fee Amendments, or GDUFA III. Many of these meetings were related to complex products, which are harder to develop, and so those communications can reduce time and increase efficiency in the development process. Those meetings occurred before ANDA submission, during ANDA review, as well as after a complete response action. GDUFA III also included new goal dates added for publishing product-specific guidances, or PSGs, for newly approved complex products. Industry can also request to meet with FDA if in vivo bioequivalence studies have been started that are different from FDA's recently published new or revised PSGs. Since I mentioned GDUFA, let me focus on one of the most important components of GDUFA since it began more than 10 years ago. That is its emphasis on science and research, which is in line with the FDA's focus on and application of the best available science to help ensure the best technology, most definitive studies with clear results, and the best decisions. While the innovator deal with American society with patent protection for a limited period of time has undoubtedly stimulated drug discovery and new treatments, the generic deal is also critical, and science doesn't stop at the point of transition to generic status. The GDUFA Science and Research Program is designed to help enable the FDA to address particularly challenging scientific issues for product-specific guidance development and to communicate with applicants through pre-ANDA meetings to help clarify regulatory expectations for prospective applicants early in the generic drug development cycle. Let me briefly highlight one recently approved generic product that I think exemplifies the meaning of FDA-supported scientific innovation and what can come out of years of GDUFA-supported research. In July 2023, the FDA approved the first generic to Vivitrol. Vivitrol is a one-month extended release injectable naltrexone that is used to help people maintain abstinence while recovering from opioid or alcohol dependence. These and other naltrexone drug products are important tools to treat substance abuse. This is also the first generic that uses biodegradable PLGA, polymer microsphere technology. The PLGA microsphere formulation gives rise to the month-long extended release of naltrexone. Although a more complex product to develop than a once-daily tablet, the once-a-month extended release injection reduces the dosage regimen frequency and therefore may be advantageous in helping ensure patient adherence to the therapeutic regimen, which is critical to reducing the risk of relapse. The complexity of developing and manufacturing products that use control-release PLGA technology is exemplified by the limited number of products that have been approved, even though PLGA is a well-known and highly tunable material to create extended-release formulations. Since the first FDA-approved PLGA product more than 30 years ago, there have been approximately 12 other approved products, and this is the first generic product that has PLGA. As with all companies developing a PLGA product, this generic had to address several complex challenges in developing, manufacturing, and demonstrating their naltrexone microspheres product is therapeutically equivalent to Vivitrol. OGD initiated its first research activities on PLGA-based long-acting products in 2013. The two main foci of these research activities were to develop analytical methods that can facilitate reverse engineering, characterization, and selection of PLGA, which is a critical first step for generic development. In vitro drug release testing methods was the second step to improve our understanding of how the drug is released from the formulation and how different polymer and manufacturing-induced characteristics affect drug release behavior. These methods have been included in FDA's product-specific guidance and have been adopted by a generic industry for supporting their generic development. In addition, the research on polymer and formulation characterization also enabled FDA to develop assessment criteria to make the generic pathway feasible for PLGA-based products. The approval of the first generic Vivitrol provides a good model for future generic development of PLGA-based products. GDUFA research will continue to explore how modern analytical methods and modeling tools can be helpful for guiding product development and bioequivalence study design. The improved understanding of the design space for this group of products will provide a good scientific foundation to explore novel approaches for product development and bioequivalence testing. In today's workshop, You'll have the opportunity to take a deep dive into some of the many ways that we work to modernize generic drug product development and enhance our science, policy, and business processes in support of this goal. In addition to strengthening our process to increase interactions with industry, to make submissions more complete and reviews more efficient, these modernization efforts include developing efficient bioequivalence approaches for products that eliminate unnecessary human studies and provide scientific approaches for generics for complex products, and helping industry maximize the use of the 505J pathway and gain clarity around how to develop generic versions of complex products, such as generic drug device combination products. Before I close, I want to mention one other important facet of generic drug development, which is its global significance and impact. As we all know, the development of drugs today, including generic drugs, is part of a global enterprise and the product of a global supply chain produced with the intent to market in multiple countries around the world. I'm pleased to see that one of today's sessions will focus on the topic of global collaboration to support efficient generic product development and regulatory assessment. Though it's not the primary focus of the discussion, I think it's important to mention one aspect of this dynamic, which is the challenge of drug shortages. Shortages are very frustrating, consume an enormous share of talented pharmacist and clinician time to find alternatives and explain these issues to patients, undermine the underlying goal of increased accessibility of these often life-saving products, and indeed, simply put, shortages put patients at risk. As you know, the FDA doesn't make drugs, and we can't tell manufacturers which or how much of a drug to produce, but we do try to anticipate shortages and take action to prevent them, including working with drug makers to submit applications for new manufacturing capacity to increase supply of existing drugs or to facilitate market entry for new drugs. In addition, we've encouraged the adoption of advanced manufacturing technologies and mature quality management practices to help avoid or mitigate drug shortages. We also partner across government, academia, health systems, and industry to strengthen and diversify the supply chain and identify and address supply chain vulnerabilities and accelerate domestic manufacturing. There are many reasons for drug shortages, but one I want to mention involves manufacturers of older generic drugs, and particularly injectables, who sometimes face intense price competition, uncertain revenue streams, and investment requirements to maintain quality conditions. U.S. patients benefit from access to these less expensive generic drugs, but if the basic economics and contracting practices of the generic drug market are not fixed, more patients will be impacted by these shortages. We need to develop transparent measures of the quality of manufacturing coupled with contracting practices for high-quality manufacturers that ensure the supply of a high-quality product. This is just one of many topics you'll be addressing in your discussions today. The diversity of generic products is enormous, as the meeting agenda makes abundantly clear. You'll have discussions related to complex APIs, complex injectable products, topical drug products, ophthalmic emulsion products, orally inhaled drug products, noteworthy guidance development and updates, new processes, research and assessment tools, common issues related to the ANDA process, and more. Quite a meeting. In short, you've got a full plate, and we're here to support both the discussion and the developments that we hope flow from them. By working in mutually supportive ways, we can translate science to approval and continue to deliver timely, high-quality, safe, affordable, and effective generic products to the public. Thank you again for your participation, and I hope you have an engaging and productive day.
Thank you for that great keynote address. This is Ray Ford working with SBIA to serve as your moderator and host for today's remaining sessions. Welcome to session 1, Noteworthy Guidances and Generic Drug Approvals for Topical and Transdermal Products. Our speakers for this session include Dr. Priyanka Ghosh, Lead Pharmacologist, Division of Therapeutic Performance 1, Office of Research and Standards in OGD, and Dr. Hiren Patel, Senior Staff Fellow, Division of Bioequivalence 2, Office of Bioequivalence in OGD. Next, we'll hear from Dr. Megan Kelchin. Dr. Kelchin is a Senior Pharmacologist, Division of Therapeutic Performance 1 in the Office of Research and Standards, OGD. And finally, before our break, Dr. Ahmed Zidan will present. Dr. Zidan is a Senior Staff Fellow in the Division of Product Quality Research, Office of Testing and Research in the Office of Pharmaceutical Quality. For a complete bio on each presenter today and Thursday, please refer to our speaker's biography located in the same area on our website as the conference agenda. Now, please join me to welcome our first presenter, Dr. Ghosh.
Good morning, everyone. Thank you so much for the kind introduction. My name is Priyanka Ghosh, and this morning I'm going to talk about the general guidances that we have published in 2022 related to characterization-based bioequivalence approaches for topical products. In part 1 of this talk, I'm going to talk about the Q3 guidance. In part 2, Dr. Hiren Patel will cover the IVRT and the IVPT guidances. This presentation reflects my views and should not be construed to represent FDA's views or policies. The learning objectives for us this morning are to understand the key concepts in the general guidances related to Q3 sameness versus Q3 similarity, and understand how an evaluation of formulation sameness or the no significant difference standard influences categorization of a product as Q3 same or Q3 similar. The guidance I'm going to talk about today is physicochemical and structural characterization of topical drug products submitted in ANDAs. We often refer to this as Q3 guidance in short. And towards the end of the talk, I'm going to talk about how to interact with the FDA if you are interested in developing a Q3 similar product. Before I go into the definition of Q3, I briefly want to remind you about Q1 is the components in the drug product. These are the inactive ingredients that make up the drug product. Q2 is the composition of the product. These are the concentration of each of the inactive ingredients in the drug product. One key thing to remember are topical products applied to the skin are not required to be Q1, Q2 the same as the reference listed drug by regulation. However, we often recommend Q1, Q2 sameness or formulation sameness in our guidances. When we do recommend formulation sameness in our guidances, we state that the test product should contain no difference in inactive ingredients or in other aspects of the formulation related to the reference standard, or in short, the no significant difference standard. Now, while I will not be able to go into the details of the similarities or differences between a Q1, Q2 assessment and a no significant difference assessment within the scope of this talk today, I encourage you to review this presentation from November of 2022, which outlines the agency's current thinking related to the no significant difference assessment. Now that we have reviewed Q1 and Q2, let's talk about Q3. Q3 is the arrangement of the active and the inactive ingredients in a product. This is an understanding of the microstructure of the drug product, and when we talk about comparative Q3 characterization in our guidances, what we are really referring to is measuring specific Q3 attributes such as the pH, the particle size, the globule size, and comparative assessment of each of those characteristics of the prospective test product and the reference product. Now, this is just an example from our research, and I included this to illustrate that when we typically recommend characterizing 3 batches of the test product and 3 batches as available of the reference product for comparative Q3 characterization. And for a product to be categorized as having the same arrangement of matter, the expectation typically is that the observed data for the test product will fall within the variability of the observed data for the reference product. So again, the expectation is that the data that we obtain for the test product falls within the variability of that obtained for the reference product. And this is important to remember, because the Q3 guidance talks about categorization of products as Q3 same, Q3 similar, and Q3 different. So how do we do that? In the guidance, the first question we ask is the arrangement of matter the same? If the arrangement of matter is the same, then the second question we ask is the underlying matter the same? What this really means is, does your product meet the no significant difference criteria? If the answer is yes, we categorize those products as Q3 same. And we have recommendations for products that meet the Q3 sameness criteria in our product-specific guidances. On the other hand, if the arrangement of matter is not the same, we categorize these products as Q3 similar. At this time, we don't have recommendations for these types of products in our guidances, and therefore we strongly encourage you to come meet with us through the pre-NDA program if you intend to develop a product, a Q3 similar product. Lastly, if the arrangement of matter is not the same, those products are categorized as Q3 different products. And for these products, we often recommend either using the comparative clinical endpoint bioequivalence studies or the vasoconstrictor studies when those are relevant for establishing BE. So now that we have talked about Q3 sameness, Q3 similar, Q3 difference, let's discuss a little bit about what are the specific Q3 characteristics that we typically recommend in the general guidance and in our product-specific guidances. The Q3 guidance summarizes basic Q3 characterization. These are characterization of appearance and texture, the phase states, and the structural organization of matter. And what we really mean by this is, for example, does your product have globules? Does your product have particles? And this information really allows us to assess if your prospective test product is the same dosage form as the reference product, and therefore supports an evaluation of pharmaceutical equivalence. In our guidances, the product-specific guidances, we often recommend comprehensive Q3 characterization. And these include, in addition to the basic Q3 characterization, characterization of polymorphic form, rheological behavior, water activity, drying rate, pH, etc. And the types of Q3 characterization that we identify and recommend in a product-specific guidance for a given reference standard depends on an understanding of the microstructure of that reference product itself. And Dr. Kelchen, in her talk a little later this morning, will demonstrate how we identify some of the relevant Q3 characteristics that we recommend for a specific reference product in our guidances. So with that background in mind about Q3 characteristics and Q3 sameness versus similarity versus difference, let's talk a little bit about how to interact with the FDA if you are interested in developing either a Q3 same or a Q3 similar product. So if you are interested in developing a product that meets the no significant difference criteria, which is essentially a product that may be characterized as a Q3 same product, if a product-specific guidance is available, and we have about 80 of those currently published. If you have a question, you can always submit a control correspondence. If you would like to propose an alternative approach, you can always submit a pre-NDA meeting. If a PSG is not available, we encourage you to review the relevant general guidances, and we encourage you to submit a pre-NDA meeting, including your proposed formulation. You can include up to 3 formulations in a given pre-NDA for assessment, and any empirical data you may have to support your proposed bioequivalence approach. On the other hand, if your product does not meet the no significant difference criteria, or products that may be categorized as Q3 similar products, we encourage you to review the relevant general guidances, but we strongly encourage you to come and talk to us using the pre-NDA program. And for these kinds of products, we want you to summarize your assessment of what the differences in formulation are between the proposed test product and RS, any Q3 characterization data you may have that illustrates why you think your product meets the Q3 similar criteria, and finally includes your proposal for the types of studies you would like to conduct to mitigate the risk associated with potential failure modes for BE that may arise due to the differences in formulation. And when you include studies, we really prefer that you include any preliminary data you may have, because that really helps us to assess the viability of your proposed approach. So with that in mind, let's go to a challenge question. My challenge question for you this morning is, which of the following statements is true? Basic Q3 characterizations can demonstrate that a test topical product and its RS are the same dosage form, supporting a demonstration of bioequivalence. Comprehensive Q3 characterizations matching the detailed profile of the Q3 attributes of the test product to the detailed profile of the Q3 attributes of the RS substantially mitigates the risk of potential failure modes for bioequivalence, supporting a demonstration of bioequivalence, and all of the above. The correct answer is B. Basic Q3 characterizations typically support a demonstration of pharmaceutical equivalence, and therefore this statement is not entirely true. With that, my acknowledgements for our entire team at the US FDA, specifically Dr. Sam Rainey, Dr. Markham Luke, Dr. Paula Simamura, and Dr. Bing Kai, who were heavily involved in the development of the Q3 guidance. And I would also like to thank all of you for attending this talk this morning, and I look forward to your questions during the panel discussion. Thank you.
Good morning, everyone. Thank you for the nice introduction, and thank you, Dr. Ghosh, for a very informative presentation to kick off this session. Now I'm going to continue with two more guidances for characterization-based bioequivalence approaches for topical products. Before going into the details of my presentation, I have the following disclaimer. This presentation reflects my views and should not be construed to represent FDA's views or policies. The learning objective for my today's talks are to discuss general recommendations pertaining to critical aspects related to conduct and analysis of in vitro release stage, that is IVRT, and in vitro permeation test studies, that is IVPT, for submission in ANDAs for topical drug products. Also, to identify important updates provided in the scientific recommendations within the general guidance for IVRT and IVPT studies. Let's begin with in vitro release stage, that is IVRT. As you all may be aware that the agency laid out the recommendation for the conduct and analysis of IVRT studies in the draft PSG of acyclovir before October 2022. The recently published new general guidance on IVRT in October 2022 now reflects the current thinking related to the IVRT studies for topical products submitted in ANDAs. For today's discussion, I'll be focusing on the important updates in terms of evolvement in the scientific recommendations within the IVRT guidance. I would suggest you to go to the detailed recommendation in the IVRT general guidance before you plan on conducting IVRT studies. Let's begin with IVRT method development. The guidance provides in-depth recommendations for the selection of critical IVRT method parameters, including receptor solution and membrane, as a part of method development. As you may know that the USP General Chapter 1724 historically recommends the typical IVRT study duration to be between 4 and 6 hours. In multiple instances, the shorter IVRT study duration was used without adequate justification. Therefore, the guidance emphasized that the study duration should be at least 4 hours to capture the release rate, which can represent the steady state release. Under very limited circumstances with adequate justification, also supported by data, may be considered acceptable, but it is not typically recommended. The linearity of the drug release rate, or the slope, across all time points should ideally have an R-square value of greater than or equal to 0.97, which was previously 0.9 in the draft PSG of acyclovir. Let's move on to the IVRT method validation. The guidance provides recommendation on what type of data can support membrane surface temperature as a part of equipment qualification. The equipment was termed as apparatus previously. Membrane innerties should be evaluated in relation to the membrane binding of the drug in the receptor solution at a concentration relevant to the range of drug concentration in the receptor solution during the test. To add to this, one concentration toward the lower end and one on the higher end of the study sample should support the range of drug concentrations. The guidance further provides details on expectation for receptor solution sampling qualification. For precision and reproducibility, it is expected to have a minimum of 3 independent runs with the nominal strength to support this particular validation parameter. The linearity of drug release across all time points should be calculated and reported for each diffusion cell and compared within and across all IVRT runs. As I mentioned earlier, for the release rate to be considered suitably linear, it should have an R-square value greater than or equal to 0.97 across IVRT study duration. Moving forward, it is recommended to report dose depletion only and not necessarily mass balance and recovery. In this regard, the guidance further indicates that the IVRT method may be considered adequate despite a depletion of greater than 30% when experimental evidence illustrates that the release rate or slope remains suitably linear for each diffusion cell when plotted versus the square root of time. I would like to make a note that only the explanation on dose depletion higher than 30% may not be appropriate to have a shorter study duration. Next validation parameter is sensitivity, specificity, and selectivity. It is typically recommended to compare the release rates from the test formulation with the higher strength, that is 150%, and a lower strength formulation, that is 50% of the nominal strength. There may be challenges under certain situations. For example, if precipitation of the active ingredient is observed when formulating the 150% strength, it may be necessary to use different strategies which may be discussed with the agency before the submission of ANDA. For IVRT specificity, an IVRT method should be proportionally linear for the correlation of the formulation concentration to the average IVRT release rate with a minimum R-square value greater than or equal to 0.95, which was 0.9 previously. To support IVRT selectivity, both alter formulation, that is 50% and 150% of the nominal strength, should fail to show equivalence to the release rate from the nominal strength formulation using statistical approach. The supplemental demonstration of the selectivity of the IVRT method further validates that it can detect the difference in the release rate which are associated with aspect of the formulation other than the strength which was demonstrated under fundamental selectivity. The alter formulation may utilize different strategies which include but not limited to the change in an active ingredient and or its concentrations, the manufacturing process, or combination of this. In addition to this important aspect, the guidance also provides some additional detailed recommendations. Like there is a separate section about sample analytical method validation, handling and retention of sample, control of study procedure with quality management system, as well as how and where to submit the specific study information in ANDAs. In the interest of time, I will not go in details as it is self-explanatory. With that, it's time for the first challenge question. If the dose depletion is higher than 30% for the IVRT study, despite the linearity of IVRT method greater than 0.99 for all cells, the IVRT method is not considered acceptable. Is it A, true, or B, false? If you selected the option B, false, then you are correct. Time to move to the next general guidance for the in vitro permeation test, that is IVPT. As I mentioned earlier, the new general guidance on IVPT reflects the current thinking related to the IVPT studies for topical drug products submitted in ANDAs, and it supersedes the recommendation in the draft PSU of acyclovir cream. Similar to the IVRT guidance, I'll be focusing on the important updates in terms of evolvement in the scientific recommendation within the IVPT guidance. Let's begin with IVPT method development. The guidance provides in-depth recommendation for the selection of critical IVPT method parameters, which include but not limited to equipment, termed as apparatus previously, dose amount, skin source, type and preparation, along with its barrier integrity test, the study duration, and the dose depletion if the wiping off procedure has been utilized. I will not go in details for each parameter in this slide. Once these preliminary parameters are laid out, it is critical to optimize the dose amount in terms of the consistency with which the dose can be applied, the reproducibility of the flux profiles, the influence of dose amount and dose duration on the shape of the flux profile, and the approximate range of drug concentration in receptor solution samples at different time points. Parallelly, the sampling schedule and study durations should also be optimized, that ideally should include a complete flux profile to identify the maximum flux and a decline in the flux thereafter across multiple subsequent time points. In certain situations, the decline in the flux may not be observed, for which the alternate approach may be used. The guidance provides recommendation to utilize wiping off the applied dose after a suitable duration from the skin and continue to monitor the receptor solution for an extended period. The next parameter is the skin barrier integrity testing. Previously, the most common types of testing were listed. For example, transepidermal water loss, that is TWL, tritiated water, electrical base, for example, transepidermal electrical resistance. However, new guidance provides detailed recommendations about the process for each type of testing, along with considerations on acceptance criteria. I would like to make a note that the testing data may not correlate with the permeation of most topically applied active ingredients, and it also recommends the do's and don'ts while conducting respective testing. Now let's move on to the IVPT method validation. I'll begin with the receptor solution qualification. Similar to the previous recommendations, the composition and pH of the receptor solution should be qualified in relation to the stability and solubility of active moiety in the receptor solution, along with the use of antimicrobial agents such as around 0.1% sodium azide or around 0.01% gentamicin sulfate to mitigate potential bacterial decomposition of the dermis and epidermis. For hydrophobic drugs, the solubility of active moiety is always a challenge in the physiological buffer-based aqueous receptor solution. It may be appropriate to use 0.1 to 0.2% polyoxyethylene ole ether in the receptor solution to enhance solubility of hydrophobic drugs. I would like to make a note that the use of organic solvents and alcohol in the receptor solution to enhance solubility of active moiety are not recommended, as it may have potential to alter the skin barrier function and ultimately alter the permeability of the skin. Let's move on to the foundation of IVPT method validation. The discrimination ability of the IVPT method may be described using two concepts, sensitivity and selectivity. The IVPT sensitivity studies are typically performed towards the end of the IVPT method development phase, and the key purpose of this study is to incorporate the final IVPT method parameters like target dose duration, dose amount, and study duration to be used in the IVPT method validation or pilot and pivotal study. In terms of IVPT selectivity or pilot study, it should be performed once the IVPT method parameters are established. The IVPT pilot study typically supports multiple IVPT method validation parameters. I would like to make a note that IVPT pilot and pivotal study parameter should be the same as the IVPT sensitivity study. Let's look at the consideration individually, and we'll begin with IVPT sensitivity. While the draft PSC of acyclovir cream enlisted modulation of dose amount, dose duration, and product strength approaches to demonstrate IVPT sensitivity, the new guidance provides detailed recommendations or reflect current thinking for each approach. For example, in terms of modulation of dose amount, it recommends the selection of lower and higher dose amount that is threefold compared to target dose amount as a starting point. Also, it further provides scientific considerations for the dosage form and formulation along with its limitation for the utility of this approach. For the modulation of dose duration, the guidance recommends selection of lower and higher dose duration compared to target dose duration using the controlled dose amount across all treatments along with preliminary thinking on which type of drug product this approach is more suitable. In terms of modulation of product strength, the guidance indicates that the utility of this approach should be considered only under certain situations because of the overall limitations. Moving on to the IVPT pilot study, which should be conducted with 3 parallel treatment, taste, reference, and a topical product or formulation that is known or designed to be different from the reference topical products using multiple donors, for example 4 to 6, with a minimum 4 replicates per donor per treatment. The pilot study supports a validation of IVPT method parameters like permeation, profile and range, precision and reproducibility, dose depletion, and IVPT selectivity. This data further help in estimation of number of donors that may be needed to adequately power the pivotal IVPT study. Speaking of the pivotal IVPT study, the IVPT endpoints include maximum flux, that is Jmax, and total cumulative amount permeated into the receptor solution across the study duration, that is AMT. Please note that this was termed as AUC in the draft PSC of acyclovir cream. The flux profiles should be plotted as the flux on the y-axis versus time on the x-axis, and the maximum values for each diffusion cell provides Jmax. The extent of drug permeation should be plotted as the total cumulative amount of drug permeated on the y-axis versus the time on the x-axis. Let's talk about the statistical analysis. If SWR value of the reference product is greater than or equal to 0.294, it is recommended to use reference scaled average bioequivalence approach, otherwise use the average bioequivalence approach for B determination. At the completion of the study, if the number of skin replicates are the same for all donors in the taste and reference topical product treatment groups in the IVPT study, use a balanced design statistical analysis. If the skin section or diffusion cells are excluded from the final statistical analysis because of experimental loss or issue and the resulting data set is unbalanced, then use an unbalanced design statistical analysis. I would like to make a note that the study design should always be balanced despite of the data being balanced or unbalanced. In addition to this important aspect, the guidance also provides some additional detailed recommendations. Like there is a separate section about sample analytical method validation, handling and retention of sample, control of study procedure with quality management system, as well as how and where to submit the specific study information in ANDAs. In the interest of time, I will not go in the detail as it is self-explanatory. Time for next challenge question. To support IVPT sensitivity using modulation of dose duration approach, the dose amount should be the same across different dose duration, that is low, target, and higher dose duration. Is it A, true, or B, false? I'm going to give you 10 seconds to think about this one. If you selected the option A, true, then you are correct. I have added some resources in the slide which maybe add further value for your topical drug product development program using characterization-based approaches. With this, I would like to acknowledge my OB colleagues, Drs. Nair, Dr. Vivian, Dr. Chiang, Dr. Cheng, Dr. Lee, Dr. Roy, as well as my ORS colleagues, Dr. Ramazanli, Dr. Kos, Dr. Rane, and bioequivalence assessors. Last but not the least, I would like to thank all of you to patiently listen to my presentation. Please keep sending your questions and I'll be happy to address as many questions as we can during the Q&A session. Thank you.
Hello, my name is Megan Kelchin, and today I'll be providing an overview of the current product-specific guidances for topical products that are applied to the skin. Here is my disclaimer. After today's presentation, you'll be able to identify the various approaches for bioequivalence for topical products that are outlined in product-specific guidances, or PSGs. You'll also be able to explain the rationale for inclusion of different components within the scope of a characterization-based bioequivalence approach for topical products, and describe how to obtain the agency's feedback on a proposed bioequivalence approach when a PSG is not available. So let's first go over how we might demonstrate bioequivalence for a topical product. The first approach is a comparative clinical endpoint bioequivalence study. This is an in vivo study in which the test product is shown to be as efficacious as the RLD, and both products are shown to be superior to a placebo control. The next approach is a characterization-based bioequivalence approach. This is a weight of evidence approach comparing the formulation, microstructure, and performance of the test product and the RLD. And I'll discuss the characterization-based bioequivalence approach in more detail in the coming slides. Next is an vasoconstrictor or VC study, and this is an in vivo study comparing the skin blanching effect of the test product and the RLD. Additionally, we may recommend a waiver of in vivo bioequivalence studies for some products, and to support the request for a waiver, we may compare the formulation or the dosage form of the test product. It's important to note that within a single PSG, there may be multiple bioequivalence approaches. For example, a single PSG may contain two options, a characterization-based approach or a clinical endpoint study. And another example would be a PSG that recommends a waiver of in vivo VE studies, or a VC study if a test product does not meet the requirements of the waiver. Topical products are utilized for a variety of indications, and it's these indications that we recommend as part of our comparative clinical endpoint studies. However, it's important to keep in mind that a single indication may have multiple ways of establishing bioequivalence, and this is dependent on things like the active ingredient, the dosage form, or aspects related to the disease. So let's look at psoriasis as an example of this. So shown in the graph are the different types of drugs that are indicated for the treatment of psoriasis. And if we look at the vitamin D derivatives, we see that there are three types of bioequivalence approaches that we recommend in our PSGs for these products. A waiver of in vivo BE studies may be recommended for solution-based products that meet a formulation sameness standard compared to the RLD product. For semi-solid drug products, we may recommend a characterization-based bioequivalence approach for a test product that meets a formulation sameness standard compared to the RLD. And for products that do not meet the formulation sameness standard of the waiver or the characterization-based approach, we recommend include a recommendation for a comparative clinical endpoint BE study. For corticosteroid products, we recommend similar types of bioequivalence approaches. For example, we recommend a waiver of in vivo studies or a characterization-based bioequivalence approach when the test product meets a formulation sameness standard. However, because corticosteroids have a skin blanching effect, we commonly recommend a VC study for these products. We do not recommend a VC study for products intended to be applied to the scalp, so in those situations, we recommend a comparative clinical endpoint study for products that do not qualify for the waiver approach or the characterization-based approach. For products that contain both a corticosteroid and a vitamin D3 derivative, we can recommend a combination of bioequivalence approaches. For example, we might recommend a VC study in addition to a comparative clinical endpoint study, or VC study in addition to a characterization-based bioequivalence approach. So the take-home message for the slide is that there are often multiple approaches for establishing bioequivalence for topical products, even within a single indication, and these recommended BE approaches within PSGs are product-specific. As Dr. Ghosh and Dr. Patel mentioned in the previous presentation, in October of 2022, the agency published 3 new draft guidances for industry related to Q3 characterization, in vitro release tests, and in vitro permeation tests for topical products. Additionally, the agency published a revised draft guidance for industry related to in vivo bioequivalence for topical corticosteroid products, and this focuses on those VC studies. This guidance will be discussed in the second part of this session today. In addition to the general guidances, we also published approximately 80 new or revised PSGs for topical products. The majority of these PSGs included characterization-based bioequivalence approaches that aligned with the recommendations in the new general guidances. So let's talk about the components of these characterization-based approaches. The first component is the no significant difference standard. The no significant difference standard is a comparison of the test product formulation and the RLD formulation, and it's based upon the principles of assessing Q1, Q2 sameness, but also considers certain differences that have previously been determined by the agency to be acceptable based on available scientific evidence. And it's important to note that a no significant difference standard expands the eligibility for a characterization-based approach for topical products. So as an example of the no significant difference standard, is a test product that contains a minor differences in inactive ingredients. For example, an RS formulation may use carbapol 934P, which is a benzene-containing carbomer that is not preferred by the agency. Under the no significant difference standard, a test product may use a carbomer homopolymer type B, which is non-benzene containing. I refer you to the December 2022 FDA CRCG presentation that's highlighted here if you'd like to learn more about the no significant difference standard. The next component is a physical, chemical, and structural characterization, or comparative Q3 characterization. And Q3 characterization is recommended to mitigate failure modes for bioequivalence associated with differences between the test product and the reference product in the microstructure. And you'll note here that in the snapshot of the PSG that's shown here, there are a variety of different Q3 attributes that are recommended. And the recommended Q3 attributes for a specific product are based on an understanding of the microstructure of that product. The next component is an in vitro release test, or an IVRT study. An IVRT study is a very sensitive and reproducible test that's able to capture differences between the test product and the RLD that may not have been captured during, with formulation sameness or Q3 characterization. For more complex dosage forms, we often recommend an in vitro permeation test or an IVPT study. This study is recommended to mitigate failure modes for bioequivalence associated with complex interactions between the formulation and the skin. For example, an IVPT study may be recommended for drug products that are multiphasic in nature or contain complex and active ingredients. An example of this would be petrolatum-based ointments. Finally, in some cases, an in vivo systemic PK study may be recommended when there's a question of whether the systemic drug concentrations contribute to the efficacy of the drug product. So as you can see on the graph here, we have developed characterization-based approaches for a variety of dosage forms, and I'll talk about this more in the coming slides. You'll also notice that at this time, characterization-based approaches are included in only a subset of published PSGs. I wanted to note that at this time we are actively working on incorporating characterization-based approaches into both new and existing PSGs. However, if you would like to propose a characterization-based approach as an alternative BE approach to what is recommended in the PSG, we encourage you to submit your proposed BE approach through the appropriate communication pathway, such as a pre-ANDA meeting request. And I'll discuss the recommendations for the pre-ANDA meeting request process later in my presentation. But for now, let's look at the components of the characterization-based approach, starting with the topical aqueous gels. First, I'll note that a no significant difference standard is recommended for all characterization-based approaches within PSGs at this time. And that's because at this time, Q3 sameness is recommended within the scope of the PSGs, and that relies on a test product that meets the no significant difference standard. Next, all characterization-based approaches within PSGs include a recommendation for basic Q3 characterization. And this includes an assessment of visual appearance and texture and microscopic images. And this is to understand the structural organization of matter of the test product and the RLD. The rest of the Q3 are recommended based on the microstructure of the drug product. So in the case of topical aqueous gels, we always recommend rheology, pH, and specific gravity. Particle size distribution is recommended when there's suspended drug in the finished drug product. The majority of topical gels are single-phase drug products, and so for these products, we do not recommend an assessment of globule size distribution. And finally, for topical aqueous gels, these products typically contain large concentrations of water, so Q3 attributes such as water activity, drying rate, and oleaginous components are not considered to be relevant for these drug products. I'll note that an IVRT study is recommended as part of a characterization-based approach for all drug products. And again, this is to capture differences between the test product and the RLD that may not have been captured with formulation sameness in Q3 characterization. Finally, I'll note that you'll notice that for diclofenac sodium topical gel, this drug product is multiphasic. So for this product, we recommend an assessment of globule size distribution, in addition to a recommendation for an IVPT study. Additionally, it's suggested that this drug product has a systemic contribution to efficacy, so in this case, we recommend a systemic PK study. So how do these recommendations compare to the recommendations for topical non-aqueous gels? Well, you'll note that for the non-aqueous gels, there's typically a high concentration of volatile and active ingredients. And so for these products, we recommend an assessment of drying rate. Additionally, these non-aqueous gels often contain low concentrations of water, and so an assessment of pH is not recommended for every product like we see for the topical aqueous gels. I'll also note that all of the non-aqueous gels are considered to be single-phase, and so we do not recommend an assessment of globule size distribution, nor do we recommend an IVPT study. So let's move on to a little bit more complex drug product like topical creams. So you'll note that the majority of topical creams are multiphasic, and this is evident by the recommendation for globule size distribution for almost every topical cream product. And for these products that are multiphasic, we include a recommendation for an assessment of IVPT as part of the characterization-based approach. However, you'll note that there are some exceptions to this rule, as there are some PSGs that are multiphasic and contain the recommendation for globule size distribution, but do not contain an IVPT study. And there are specific situations in which we might not recommend an IVPT study as part of a characterization-based approach. So for example, for mupirocin calcium and silver sulfadiazine topical creams, these drug products are applied to the skin with no stratum corneum. And so therefore, we do not require an assessment of the interaction between the formulation and the drug, or the formulation and the skin via an IVPT study. For dicosanol is considered to be considered to have unique physicochemical properties and dosage form, and therefore does not necessitate an IVPT study. Finally, some drug products have a site of action that is in the superficial stratum corneum. These products might include things like ketoconazole and butanedione hydrochloride topical cream, which are indicated for treatment of fungal infections where the infection is located in the superficial stratum corneum, and therefore an IVPT study is not needed. I'll note that when an IVPT is not recommended as part of a characterization-based approach for a drug product that is multiphasic in nature, we do recommend an assessment of drying rate for those products. And that's to evaluate differences in the metamorphosis of the drug product over time, which would typically be captured via an IVPT study. Like the topical creams, topical lotions are mostly considered to be multiphasic drug products, as you can see by the recommendation of globule size distribution for all cream lotion products. Similar to the creams, though, you'll see that an IVPT study is recommended for most topical lotions, but there are some notable exceptions. For example, In the case of ammonium lactate, the site of action is in the superficial stratum corneum. And then for this product, we recommend an assessment of drying rate to capture those differences in metamorphosis. In the case of benzyl alcohol or ivermectin, these drug products are indicated for the treatment of lice, and the site of action is an external organism, in this case, the lice on top of the skin. So for these products, we recommend an ex vivo pediculicide hair tuft assay as the biorelevant study for these drug products in lieu of the IVPT study. So as previously mentioned, an IVPT study is commonly recommended when the drug product is multiphasic or if it contains complex and active ingredients such as a large concentration of oleaginous components. So if we look at the topical ointments, you'll see that when a PSG recommends both an assessment of oleaginous components and assessment of globule size distribution, we also recommend an IVPT study. Even for products where the drug product does not contain globules but does contain a large component of or a large concentration of oleaginous components, we recommend an IVPT study. However, as always, there is a notable exception. For miconazole nitrate, white petrolatum, and zinc oxide, the drug product contains a large component of or a large concentration of oleaginous components, but does not include an IVPT study, and this is due to the indication of the drug product. Finally, I want to highlight that the recommendations within the PSGs are research-driven, and a great example of this is Terbanibulin topical ointment. Dr. Zaiden will be discussing the research that was done to support the development of this specific PSG in the next presentation. So you might be thinking, what happens if I want to develop a generic for a specific product that does not have a PSG? The first thing I recommend is to check the FDA's upcoming PSG website. This contains a list of PSGs that OGD plans to publish within the next 12 months. You can also send in a request for guidance development, and this helps OGD identify which products are of high interest to the agency. Finally, you can submit a pre-ANDA product development meeting request with your proposed bioequivalence approach. In a pre-ANDA meeting request, we recommend you include the following information: a clear, specific, and comprehensive outline of your exact BE approach, specific questions related to your proposed BE approach or your product development program, a formulation table with up to 3 proposed test formulations, and empirical data to support the proposed BE approach. So let's move on to some challenge questions. Challenge question number 1, which of the following is false? A, when a PSG contains a characterization-based approach, a no significant difference standard is always recommended. B, comparative Q3 characterization is always recommended. C, an IVRT study is always recommended. Or D, a systemic PK study is always recommended. The answer is D. A systemic PK study is not always recommended as part of a characterization-based approach for topical products. Challenge question number 2. In general, when is an IVPT study recommended as part of a characterization-based approach for topical products? A, when the drug product is multiphasic. B, when the drug product is a petrolatum-based ointment. C, always. Or D, A and B, when a drug product is multiphasic or when the drug product is petrolatum-based. The answer is D. So in summary, PSGs for topical products contain harmonized recommendations in alignment with the relevant general guidances. The recommendations within a characterization-based bioequivalence approach for a topical product are product-specific and are based on an understanding of both the microstructure of the drug product and the performance of the drug product. Finally, when a PSG is unavailable, applicants are encouraged to submit a pre-ANDA product development meeting request to get the agency's feedback on their proposed bioequivalence approach. With that, I'd like to acknowledge all of my colleagues at the FDA. And I also would like to direct you to a number of resources that may be helpful as you start to develop your characterization-based bioequivalence approaches. With that, I'd like to thank you for your attention, and I look forward to answering your questions during the panel session. Thank you.
Good morning. In today's talk, we are going to discuss how research supports the development of product-specific guidance for topical drug product. I am Ahmad Zidan. I am a senior staff pharmacologist at DBQR of FDA. First, Let me ground my presentation with this statement of pharmaceutical quality. Everyone deserves confidence in his next dose of medicine, and pharmaceutical quality, it assures the availability, safety, and efficacy of every dose of the medicine. Pharmaceutical research is not only the heart to develop and manufacture of topical drug product, but also it is hard to develop guidelines and to ensure that a science-based decision about drug approval is taken. Pharmaceutical research is involved in all stages of life cycle of drug product, starting from formulation development and selection of excipients and running compatibility studies, and in formulation design to ensure an enhanced topical penetration and delivery of the drug. It is involved in preclinical studies to get more information about in vitro performance, for example, the drug release rate and drug permeation across the skin, and to get more information about the rheology and sensory attribute of the formulation. And also it's involved in the in vivo studies to get more information about the pharmacokinetic biodistribution and elimination after topical application. It is involved in the safety and efficacy assessment, for example, to ensure safety of topical product, in development of skin irritation studies, and in clinical trials to get information about the clinical endpoint studies to ensure efficacy and dosing and patient outcome. And also it involved in the robust data collection and statistical analysis, and this is all to comply with the regulatory requirement. And the main outcome is to ensure that the generic product is similar to the reference drug product in safety and efficacy and also of quality. So in my talk today, we are going to cover how research contributed to availability of generic topical products, and how research is used for demonstration of bioequivalence, in particular about in vitro characterization to support the product-specific guidance development. And we are going to highlight two of FDA case studies about the physicochemical research that supported BSG development. We are going to talk about terbinafine topical ointment and clascoterone topical creams. And we will have some challenging questions and key takeaways. So by the end of my presentation, the audience should gain a comprehensive understanding of the framework used to assess bioequivalence of generic topical product, and they should explore the multifaceted aspects of bioequivalence, including in vitro dissolution permeation study, quality equivalence evaluation, safety, efficacy assessment, and label consideration. And they should comprehend the pivotal role of pharmaceutical research in shaping the product-specific guidance for topical drug products. Indeed, a comprehensive research process is fundamental to ensure the availability and quality of generic topical product. This research facilitates overcoming the barriers for developing generic topical products in terms of demonstrating bioequivalence, ensuring formulation similarity, establishing IVIVCs, assessing sensory experience and setting reference standards, and understanding absorption variability and confirming therapeutic effectiveness, and conducting comparative clinical trials and monitoring product post-market. So let's go over some of these points in detail. For example, bioequivalence studies are done to demonstrate that the generic product performs similarly to the reference product in terms of drug release and absorption. And these studies are essential to establish that the generic product is therapeutically equivalent to the innovator product. And the qualitative and quantitative variability in excipients between generic and reference product may impose a challenge to demonstrate formulation sameness and similarity, specifically because it impacts the drug release and absorption, making it more challenging to demonstrate bioequivalence. So establishing also the correlation between the in vitro dissolution testing and the in vivo performance parameter is crucial for development of generic topical product, and demonstrating that the in vitro performance matches the in vivo performance of the reference product may be a barrier for availability of generic topical products. And the patient's sensory perception of generic product can also be influenced by factors like packaging, branding, past experience with generic medications. So negative perceptions may be challenging and may result in deterrence from trying a generic product, and it affects the availability of a generic topical product. And sometimes it's also challenging to determine what is the reference standard for a generic topical product, and lack of appropriate reference standard may hinder demonstration of comparability. Topical drug products such as creams and ointments can have a variable absorption rate due to difference in excipients or formulation design or structure. So achieving consistent and comparable absorption to a reference product may be challenging for development of generic. And the last but not the least is that ensuring the generic topical product have a same therapeutic effect as the reference product is essential for a regulatory approval and market acceptance. And any differences in therapeutic effectiveness may pose a barrier for availability of generic product. And this is where the research comes to alleviate these barriers and make generic topical products available to the patients. Demonstrating equivalence of topical product involve a single or a combination of the in vivo, in silico, and in vitro approaches. Historically, in vivo studies such as in vivo pharmacokinetic studies, in vivo pharmacodynamics such as vasoconstriction studies, and in vivo comparative clinical endpoint bioequivalence studies are used, which are comparative clinical trials conducted on human subjects to directly assess the clinical equivalence by monitoring absorption, therapeutic effectiveness, safety, and adverse effects. In silico modeling approaches are also used for demonstration of bioequivalence. For example, computer-based simulation predict how the drug behaves without the physical experiments. Examples include pharmacokinetic modeling and the in silico skin permeation model, which help forecast the drug absorption and permeation. And most recently, the in vitro laboratory-based experiments to define clear criteria for qualitative, quantitative, and structural similarity, as well as refine in vitro testing methods such as in vitro release testing and in vitro permeation testing, are proposed for demonstration of bioequivalence. The research finding of these in vitro approaches informs the development of product-specific guidelines that streamline the regulatory approval for generic topicals, and it ensures that they are closely match the reference product in terms of sensory attributes, composition, and drug product performance. And of course, this PSGs based on this research can provide patients with more affordable options while maintaining safety and efficacy standards. So indeed, the physicochemical and structural similarity or sameness are critical for bioequivalence performance of topical drug product. And as stated in the Code of Federal Regulation, any ANDA may include different inactive ingredient, provided that the applicant identifies and characterizes the differences and provide information demonstrating that these differences does not affect safety efficacy of the proposed drug product. And the agency have released several product-specific guidance that recommend bioequivalence approaches of the products that show no significant difference in formulation to the reference standards. Most recently, the FDA released a draft guidance on the physicochemical and structural characterization of topical drug product that is stating what may be the criteria that used for Q3 characteristics. For example, the assessment of appearance, texture, investigation of phase status, and structural organization of matter, and assessment of the polymorphic form of the active ingredient, and the rheological behavior characterization, and the characterization of water activity, drying rate, pH, and buffering capacity, and similarity in the oleaginous component and specific gravity, as well as characterization of any metamorphisms related to changes in the formulation as it is applied to the skin surface. So for example, here we can see the product-specific guidance for metronidazole cream that's released in October 2022. and the guidance is stating two options for demonstration of bioequivalence. The first option for bioequivalence study with clinical endpoint, and the second option is in vitro bioequivalence study and other in vitro characterization to demonstration sameness of Q1 and Q2 and Q3. And in particular about Q3, visual appearance and texture, phase status, structure, organization of matter, rheological behavior, pH-specific gravity is recommended, and more specifically, any other potentially relevant Q3 attributes in addition to in vitro release testing. Now research is needed on determination of what is these additional Q3 attributes that may be relevant to this specific drug product, and the reference standard is listed. Therefore, the in vitro physicochemical characterization and innovative research have played a critical role in shaping the FDA guidance for topical drug product. This influence has been seen in inclusion of physicochemical data that informed the development of bioequivalence guidance for ensuring standardized and comprehensive assessment criteria, and also it had been seen in introduction and validation of innovative analytical technologies when backed by solid research. And also it has been seen in addressing the challenges of multiphasic formulation through extensive characterization and research, and also in real-world data from the physicochemical studies when it contributes to the integration of real-world evidence into the bioequivalence evaluation process, and it enhances the regulatory decision. And also it had been seen in continuous guidance improvement based on scientific advancement and stakeholder feedback, with data from physicochemical studies contributing to this continuous improvement. The last but not the least is that the agency always aligns with international guidelines through physicochemical approaches that contributed to harmonizing the regulatory standard globally. So these approaches not only support the FDA's commitment to ensuring safety and efficacy and quality of generic topical drug product, but also it fosters the international harmonization efforts and then benefiting the patients, healthcare providers, and pharmaceutical industry. So now let's go over a couple of FDA physicochemical research studies that supported the BSG development for topical drug products. The first case study of bioequivalence assessment of tirbanopolene topical ointment using in vitro studies. Tirbanopolene ointment 1% contains mono and diglycerides and propylene glycol as inactive ingredient. The optical images showed insoluble phases within the ointment matrix. This insoluble phase may be attributed to the insoluble portion of the mono and diglyceride, and of course further investigation was then performed to explore the chemical identity of this dispersed phase of the formulation and to determine the drug distribution among these phases. So the research included microscopic examination, staining test using different dyes, and then chemical identification using Raman spectroscopy. And the technique that was used, it was morphological directed Raman spectroscopy. To determine the chemical identity of these insoluble structures, a library of Raman spectra for each individual component as well as for the insoluble fat separated from the matrix were done. The Raman spectra were compared, and the results showed that the spectra of the matrix and these insoluble structures are the same. In addition, propylene glycol showed the highest correlation in both the insoluble fats and the matrix. So this study confirmed that these insoluble structures are insoluble fats of mono and diglycerides rather than immiscible droplets of any other inactive ingredient. And the API tirbanopolene is mainly distributed in the continuous phase and to a lesser extent in the dispersed oily structure of the ointment. Therefore, this ointment may be regarded as a single-phase ointment for other bioequivalence assessment studies. And based on these results, a product-specific guidance of tirbanopolene topical ointment was released in October 22nd. The BSG does not recommend the IVPT here for phase status. However, it recommends characterization of visual appearance and texture of the ointment. as well as characterization of phase status and structure organization of matter using microscopic examination with representative high-resolution microscopic images at multiple magnification, among other in vitro characterization methods for demonstration of bioequivalence. The second case study included bioequivalence assessment of clascoterone topical cream using several in vitro studies. Clascoterone topical cream is a multiphasic formulation made of several hydrophilic and hydrophobic excipients and surface active agents. In vitro permeation testing may be then used to assess the dynamic distribution of the active ingredient among different phases of the formulation, and to study how this distribution may affect the continuous absorption and availability of clascoterone. It is worth noting that clascoterone molecules are not stable in the physiological solution and can be hydrolyzed to cortoxylone via cortoxylone 21 propionate at body temperature. Therefore, this study aimed at assessing the feasibility of conducting IVPT for clascoterone cream in assessment of its bioequivalence, and to determine what is the appropriate analyte for comparing the rate and extent of clascoterone bioavailability after topical administration. For this, the research included developing an analytical method using liquid chromatography with tandem mass spectrometry for quantitation of clascoterone, cortoxylone, and cortoxylone 21 propionate in IVPT and mass balance samples. And the tool that had been used is liquid chromatography with tandem mass spectrometry. The chromatographic analysis of the permeation sample was done without any immediate sample processing, and the chromatogram of the 24 and 36-hour samples showed a significant decrease of clascoterone solid blue peaks and a significant increase of cortoxylone 21 propionate white peaks within 12 hours period. However, the internal standard peaks intensity was stable over this observed period. The permeation profile of clascoterone and cortoxylone was then obtained using the inline flow-through system with constant removal and replenishment of the receiver solution. This in vitro permeation profile of clascoterone and its metabolites were obtained over the course of 48 hours. So this test was performed using a dermatome skin samples from 4 donors, and the IVPT cumulative permeation and flux data demonstrated good reproducibility and similar trend of clascoterone permeation among all donors. And the highest flux of clascoterone permeation appeared over about 6 hours in all donors. The outcome of this study supported a revision of the product-specific guidance of clascoterone topical cream, and based on this study, IVPT testing was feasible and may be used to characterize the effect of globule site distribution of clascoterone topical cream and its topical absorption. And clascoterone and cortoxylone may be used as analyte for permeation samples. These analytes were observed as early as 2 hours in the receptor solution, indicating metabolism of clascoterone during IVPT testing. And timely sample processing as the test progressed could prevent post-permeation metabolism and loss of clascoterone in the permeation sample. The guidance revision is scheduled and it will be released this month. So the key takeaways from these slides is that pharmaceutical research supports the development of BSGs for assessing bioequivalence of generic topical product, and it provides evidence for safety, efficacy, and therapeutic interchangeability between generic and reference topicals. And comparative characterization of the physicochemical and structural properties of complex topical product is a part of the comprehensive assessment for pharmaceutical equivalence, bioequivalence, and product approval. And as the product complexity increases, so do the risk and the need for more comprehensive Q3 characteristics. And research-driven evidence ensure approval of safe and effective generic topicals, and indeed it benefits the patient having affordable and reliable medication. Let's go to a challenge question. Which technique may be used for providing morphological information and identifying molecular components through chemical mapping of topical formulations? A. Scanning electron microscopy. B. differential scanning calorimetry, C, Fourier transform infrared spectroscopy, and D, morphologically directed Raman spectroscopy. So the answer is D, morphologically directed Raman spectroscopy, as it combines Raman spectroscopy image and imaging to enable obtaining morphological information and identify the molecular component in multiphasic topical products through chemical mapping. So at the end of my presentation, I would like to acknowledge the team of topical drug product in both OBQ and OGD, all the name listed there so that I'm not forgetting anyone. And thank you for listening to my talk, and we can open more questions during the panel discussion. Thank you.
Thank you for the wonderful presentations. We'll now enter into our break until 10:50 a.m. Eastern Time. Please enjoy your break. Welcome back from the break. We hope you're able to refresh your coffee or your tea. We'll be moving into our remaining sessions before the Q&A panel. Our next presentation will be provided by Dr. Key Wren. Dr. Wren is Deputy Division Director in the Division of Bioequivalence 3, Office of Bioequivalence and OGD. And then finally, Dr. Kevin Fang will provide the final presentation. Dr. Fang is a Senior Chemical Engineer in the Division of Quantitative Methods and Modeling in the Office of Research and Standards in OGD. Now, please join me to welcome our first presenter, Dr. Wren.
Good morning, everyone. During the next two talks, we will discuss the in vivo bioequivalence guidance for topical dermatologic corticosteroids. I will present on the overview of the guidance and summarize the major changes introduced in the October 2022 revised guidance. Following my overview, Dr. Kevin Fang will address ANDA challenges associated with vessel-constructed studies. Here are the learning objectives. Understand the revision to the draft guidance for industry on topical corticosteroids. Identify and summarize the major changes to the pilot study and the pivotal study criteria and designs. Provide updates on the public comments received through FDA docket for the draft guidance. Let's touch on some pivotal moments in the guidance history. In 1992, the topical corticosteroids in vivo bioequivalence and the in vitro release method was first published as interim guidance. This guidance included both dermatopharmacokinetic studies, skin stripping, and in vitro release studies. At that time, the agency did not have sufficient data to endorse the skin stripping method for documenting the bioequivalence of topical corticosteroids. This approach could be adopted for bioequivalence documentation given sufficient method validation. In 1995, the topical dermatological corticosteroid in vivo bioequivalence guidance was introduced. This document introduced a pharmacodynamic approach, specifically the vessel-constructed assay, and include both a pilot dose duration response study and a pivotal bioequivalence study. After 27 years, the guidance received an update and was shared for public input in 2022. The latest revision expanded on the recommended methodology and provide more detailed instruction regarding the study design and the vessel construct method qualification. This guidance outlines an in vivo pharmacodynamic approach to demonstrate bioequivalence of such corticosteroids. They induce the vessel construction in the skin blood vessel, results in the pharmacodynamic effect of skin branching. The extent of the branching changing in skin color is influenced by the corticosteroid's potency, increasing as more of the corticosteroid permeates the skin. Therefore, the pharmacodynamic vessel-constructed response can serve as a surrogate measure for the rate and extent of the topical corticosteroid availability at the site of action. The guidance recommends ANDA applicants to conduct 2 in vivo VCA studies, a pilot dose duration response study and a pivotal bioequivalence study. A pilot study is designed to set the parameter for the pivotal study which supports the bioequivalence determination. All subjects who complete the study should be included into the ED50 calculation. The pilot dose duration response study is similar to performing a standard curve for an assay. The results provide the dose duration response data required to determine ED50 value for use in the pivotal BE study. This study used the reference product only. Typically, 7 to 9 dose duration are used in the pilot study, with a sample size of around 20 subjects. The pivotal BE study is based on the dose ED50 as estimated from pilot dose duration response study. 3 doses will be used in the pivotal study: ED50, D1, half of the ED50 value, D2, double the ED50 values. For the simple E-maxima model, a subject qualifies as a detector if the ratio of their area under effect curve, AUEC, response at D2 to AUEC at D1 is at least 1.25. Only those qualified subjects are considered for statistical analysis. Here is an example of the study design for the pilot dose duration response study. In the left panel, the drug product will be applied to the surface of each subject's ventral forearms for duration range from 0.25 to 6 hours. In addition, 2 untreated control sites will be designated on each forearm. The pharmacodynamic response will be assessed by the degree of skin bleaching observed, which is measured using a chronometer. Please note that corrected baseline adjusted chronometer values are used in the data analysis. You can see the dose response curve. X represents the dose duration. Y represents the response. The ED50 is the dose needed to achieve 50% of the E-maxima value. The ideal dose range should be within the leading portion of the dose response curve. If the dose range is chosen at plateau, even if the 2 formulations differ, their PD response will appear similar. However, when the dose range is on the leading portion of the dose response curve, even a minor difference between 2 formulations can result in a significant difference in the PD endpoint. In the next slide, we will summarize the major changes from the October 2022 draft guidance. In addition, I will briefly clarify and explain the rationale behind the changes. I will begin with the background section. In this guidance, a new term reference standard has been introduced. The reference standard is a specific drug product that the FDA has chosen and which the applicant is required to use when conducting the in vivo BE testing to support the approval. Typically, the reference standard selected by the FDA is the RD. The background section also touches on model selection and the optimization for the pilot dose duration response study. Beyond the VCA studies approach, there is an alternative method, an in vitro characterization-based approach for establishing the bioequivalence of topical corticosteroid product might be acceptable if the proposed test formulation has no difference in its inactive ingredient compared to the reference list drug, which could significantly impact the availability of the API. The 1995 guidance specifies that AUEC should be calculated from 0 hour to 24 hours post-product removal for each subject, each baseline adjusted, untreated control site, and corrected dose duration. There are two major reasons why starting the parameter reading at 0 hour after drug product removal is not reasonable. First, in the VCA study, there isn't a true 0 hour reading from the time of the product removal. The process from the start of the product removal to the completion of the parameter reading is not instantaneous. During this period, the developing the vessel-constructed response could be influenced by the effect of the removal process. Secondly, a stabilization period is necessary for the skin return to its normal state before the initial parameter reading is taken. In the revised guidance, we have specified that 0 is a point within 15 minutes post-product removal. From a practical standpoint, in the revised guidance, we only recommend staggered application with a synchronized removal method. The synchronized application with staggered removal technique has been removed from the guidance. There are different models which are available to express the relationship between drug dose and the pharmacodynamic effect. However, the agency recommends the E-maxima model to characterize the dose duration response of the topical corticosteroids. If applicants found the E-maxima model not suitable, alternative model may be considered, provided they are supported with justification and a proper model selection process. If E-maxima model is selected, applicants are recommended to detail their model optimization procedure and clarify the rationale behind their ED50 selection. For example, the model parameter distribution assumption, be it normal or log normal for both E-maxima and ED50 parameter, need to be specified. Dr. Fang will provide deep into this topic in his presentation. Importantly, the guidance has been updated to exclude the naive pooled data method used for estimating the population ED50. In addition, the guidance no longer references the software names, including PFAM. In the pivotal study, the criteria for selecting the individual subject, termed a detector, is determined by comparing the mean reference AUEC at D2 to that at D1 for the same subject. If proposing the simple E-maxima model, the ratio should be minimum of 1.25, with both mean AUEC value at D1 and D2 being negative. However, based on the choosing dose duration response model, alternative ratio value may be acceptable if adequately justified. To ensure a reliable reading value for D1 and D2, we have increased the minimum requirement from 1 side per arm to 2 sides per arm for both D1 and D2 dose duration. Consider the following example. There was significantly variability in the D1 value. The D1 value reported for the right and left arm different, with the right arm showing a positive value, on the left arm a negative one. However, if we only consider the value from the right arm, this subject would not qualify as a detector. By averaging the D1 and D2 reading from both arms, the subject 1 meets the criteria to be considered as a detector. In the revised guidance, we have introduced a new section titled Vessel Constructor Method Qualification. The chronometer is the apparatus most used to measure the skin branching response. To ensure the procedural consistency both within and between chronometers, the variability of the both intrachronometer and interchronometer measurement should not exceed 15%. Operator validation assess the variation between different operators to ensure the procedural consistency both within and across operators. The coefficient of variation for intra-operator and inter-operator measurement should not exceed 15%. Certain changes have been made to the ECT summary table. Now we have two recommended tables focused on chronometer validation and operator validation. We have removed the skin site validation as well as intra-subject and inter-site validation from ECT summary table. The reason is that the skin tone can differ within an individual and between different individuals. A site validation essentially captures the variability in the measurement within an individual across different measurement locations, and the variability between measurements from different individuals. Therefore, this does not fall under vessel constructed method qualification. To view the modified ECT summary table, please check the link list here. In the appendix section, 3 new appendixes have been added. Appendix 2 provides an example of the skin branching study design for pilot study. Appendix 3 covers the calculation of area under effect curve. And Appendix 4 focuses on the E-maxima model. In addition, the figure showing synchronized application with staggered removal has been removed from the guidance. This slide summarizes the key updates in this guidance and is provided as a ready reference if needed. We received 7 public comments through the docket. As example, those comments touch on concerns such as clarifying the term study subject within the vessel constructed method qualification, asking for clarity on the randomization of the dose duration skin site, and the inclusion of the source code for ED50 assimilation for the pilot study. Here are 2 examples of the feedback we received. Please note that FDA is actively reviewing these comments, and we anticipate publishing either a revised or finalized guidance in the future. Summary and the recommendations. The updated guidance served as an essential guide for NDA applicants use a pharmacodynamic method to evaluate bioequivalence for topical corticosteroids. Applicants can submit questions through control correspondence or to request a product development meeting for relevant complex products if need FDA's input before the study. The FDA has established different communication pathways, including control correspondence, product development meetings, and post-complete response meetings to address industry questions throughout the generic drug development and approval process. I would like to thank PASA and Apple for their input on the guidance revision. Additionally, I want to thank the working group members from the Office of Bioequivalence, the Office of Research and Standard, the Office of Generic Drug Policy, and the Office of Clinical Pharmacology for their input and collaboration. Challenge question time. Question number 1. Only detectors should be included in the pivotal study for bioequivalence evaluation. A, false. B, true. The correct answer is B. Only detectors should be included in the pivotal study for bioequivalence evaluation. Question number 2. Which statement is true? A. Only the reference standard is recommended to be used in the pilot study. B. Both the reference standard and the test product are recommended to be used in the pilot study. The correct answer is A. On behalf of OGD, thank you for your attention. I'm now passing the stage to Dr. Feng, who will discuss ANDA challenges related to vessel construct studies.
Today, I'm going to present a topic of ANDA challenges related to vasoconstrictor studies. My name is Kevin Feng. I'm Senior Staff Fellow working at Quantitative Clinical Pharmacology team at DQMM, ORS, OGT, CEDAR. Here is the disclaimer. This presentation reflects the views of the presenter and should not be construed to represent FDA's views or policies. Here is the outline of my presentation. The first one is background of vasoconstrictor studies. The second one is challenges with ED50 determination and nonlinear Emax modeling. The third one is impact of incorrect ED50 selection on detector sample size. The fourth one is on other challenges and deficiencies with vasoconstrictor studies. The last one is challenge questions and summary. The introduction of vasoconstrictor studies. FDA drug guidance was published in 2022 for corticosteroid in vivo bioequivalence study, recommends conducting a pilot dose duration response study and a pivotal in vivo vasoconstrictor assay bioequivalence study for the corticosteroids. The pilot study established the dose duration response relationship using the Emax model. The pivotal study is conducted at 3 durations based on ED50 determined in the pilot study to access BE between test products and reference standard. The ED50 was determined by Emax modeling. So here is the equation of Emax model with the Emax and the dose duration and ED50. So here is a nonlinear dose-response relationship. The response is the pharmacodynamic skin blanching via assessments of baseline skin and skin blanching. For the y-axis is the AUEC of the response. Is the, for example, AUEC of 0.5 to 24 hours. As dose level like a half an hour, 2 hours, 4 hours, up to 24 hours. And the dose duration, including like the time periods for stagger application with a synchronized removal, for example, removal after 15 minutes, 30 minutes, 45 minutes, and 1 hour up to 8 hours or more. The data is coming from the reference standard VCA studies. So here is the curve to show the dose duration response to show the Emax response curve. The rationale of ED50 determination, there are two aims. The first aim is the responses attained in the study are situated in the sensitive, the stiff region of the dose duration response curve, allowing for effective discrimination between the test product and reference standard. The second aim is the pivotal B study detector identification to be included in B analysis is defined as AUEC of D2 of D1 greater than 1.25 of the simple Emax model. The methods for the Emax model depend on two factors. One is model-dependent factor, the other one is data-dependent factor. So you can see there are two data and model shown in the graph. On the left is the simple Emax model. However, on the right is the sigmoid model. The results is really to optimize the ED50 is two crucial parameters for effectively detecting the formulation difference. So the question here is now, why are pilot studies necessary for the no-RS product? So here is a list of ED50 estimation from ENDA submissions. In the table, we listed 4 corticosteroids. So each corticosteroid with the same RLD, but with different ENDAs included. So given the ED50 for each of the ENDA, it ranges very differently. If looking at corticosteroid 4, is coming from 30 minutes to 146 minutes, is up to 4-fold difference. And looking at the detector rate at pivotal study on the table, you can see the detector rate also very different. I'll give you two examples. The example with a marking at 1 and 2, is corticosteroid 2 for the two ENDAs. So the first ENDA is 6 minutes of ED50 value, but it corresponds to a lower detector rate for 57 minutes, percent of detector rate. However, for the second corticosteroids, for the lower the ED50 corresponding to higher detector rate, 74%, and for the higher ED50 value, 800 minutes, given the very low detector rate. So therefore, conducting a pilot study for accurate ED50 determination becomes essential, and employing appropriate modeling practices can lead to the most optimal solution for ED50 selection. There are a few challenges with ED50 determination and nonlinear Emax modeling. The first one is different software programs use varying parameter estimation using different population modeling and analysis methodologies. The second one is incorrect estimation of ED50 can result in decreased detector rates in the pivotal study, potentially leading to a smaller sample size for determining in bioequivalence. The last one is to overcome software limitations. It is essential to undertake focused model optimization and establish standardized procedures. The impact of incorrect ED50 selection on detector subjects. It is very important for accurate ED50 estimation is to avoid the ED50 being too high or too low to ensure accurate sensitivity in those response relationship. The impact if the ED50 is too high is reduce the cortical constrictor response as strength increased, resulting in a flattened response curve at a higher concentration. It results in the higher ED50 will lead to fewer subjects meeting the dose response criteria, like D1 of D2 is greater than 1.25. For the too low for the impact of ED50 for the low-potency corticosteroids. The challenge in eliciting vasoconstrictor response despite increased dose duration, the lower ED50 lead to higher uncertainty at the very beginning, variability, and require more subject for reliable results. Here is an example of ED50 optimization by looking at exploratory data analysis, and is given the list of the individual subjects using the spaghetti plot. And if looking at each individual, some of the individual going up and down, some of the individual does not have a good dose duration response curve. So the estimation, there are two methods. One is the naive pool method that pull all the individual subjects together. The second one is the nonlinear mixed effect model, the NLME method, that incorporating the individual variability, so it produces less bias to abnormals observations. So the NLME method was the recommended method. By looking at the scientific and regulatory challenges, divergent ED50 outcomes as observed across software platforms. So here list 5 software platforms: PFARM, SAS, Monolix, Norman, and Phoenix. With different estimation algorithms, there are 2 example datasets, dataset 1 and 2, for the ED50 estimation. For the dataset 1, the ED50 value are very similar from 80 to 90 minutes. However, for the dataset 2, the ED50 value produce very different results from 60 minutes up to 390 minutes. Therefore, different software platforms with different datasets produce very different results. And if you're looking at a particular software platform, Norman and SAS, the estimation model and algorithms are also very different. If looking at the model, there's a model parameter, normality assumption, Emax, and for the Emax and ED50, and residual error also different, active error model, normal error model, for example. And estimation algorithm, you have FO, FOC, SAEM, important sampling, and also the adaptive Gaussian projected algorithms. Again, let's looking at deeper insight within a software Norman example. They also have the scientific and regulatory challenge for the ED50 determination. In looking at their list of 7 different models, they are color-coded. For example, the estimation algorithm difference, and the ED50 have produced very different results. Also, the normality difference for ED50 assumption, they also produce very different results. And also the Emax normality assumption, the error model, and also estimation algorithms. You're looking at from 9 minutes to 36 minutes. Therefore, even within one software platform, ED50 can be different. So here we present ED50 optimization example, and as example 1, we have 2 software, Norman and SAS. Each one has 6 models with different normality assumption, error model, estimation algorithms, and AIC values, and then the estimation result for Emax and ED50. The aim is for the optimization is determined by the lowest archive key information criteria value. That's AIC value. For the model 6, we have the lowest AIC value, and the ED50 is 35 minutes instead of 9 minutes. And you were looking in the SAS algorithm. The SAS algorithms perform is subbound and is failed to show changes in the AIC value with different normality assumptions. So if looking at ED50 normality difference, normal and log normal, the AIC keep the same without changing. So different software have different performance. And example 2, I'm looking at the spaghetti plot on the right. The train is clear, and here we also have two software, NAMAN and SAS. Again, we are looking at the optimal result is determined by the lowest AIC value. So here is the lowest AIC value. The ED50 the 60 minutes. And even looking at both NAMAN and SAS ED50 results, they are pretty the same, around 60 minutes. And however, the SAS algorithm is not sensitive to parameter normality assumption. Again, the normality difference, but the AIC value keep the same. At this stage, you have already know that different software, even with the same software, they will produce different ED50 values given the different data set. So here is the recommendation for model selection and model building. So when you're doing the model selection and model building, consider that a software platform that can support the following modeling procedures. So the first one, that is you can clearly define a predetermined model selection process. And the second one is Emacs model selection. The third one is comparison of estimation methods. The fourth one, selection of model parameters. The fifth one, choosing error models. The sixth one, procedure for initial estimates. The last one is the appropriate model diagnostics. So that's the model recommendation for you to consider. We list other challenges and deficiency with VCA studies. So there are two challenges with vasoconstrictor response. The first one is weak vasoconstrictor response for low potency drug hinder establishing a dose-response relationship. The second one, truncated vasoconstrictor responses and incomplete plateau levels affect accurate ED50 estimate. So the deficiency in pilot study, that including 3 examples. So the agency's ED50 estimation, for example, we got 80 minutes, was 5 times greater than the applicant estimate, 60 minutes. The second example is the applicant selected ED50, for example, 16 minutes, falls in an insensitive regions under like 20% of Emacs values. The agency also requests analysis with good sensitivity, for example, D1 and D2 responses in 33% to 60% of Emacs. So last, we looking at the deficiency in pivotal study. There are 2 examples we produce. The first one is a short dose duration of D1, for example, 8 minutes from estimated ED50 shows high variability and low response. The second one is observation suggests unreliable selection of evaluable subjects. So here I provide 2 challenges questions. Question 1, what challenges are associated with ED50 determination in vasoconstrictor study for the development of generic corticosteroids? A, variability in parameter estimation for optimal ED50 due to different software programs and analysis methodologies. B, difficult in selecting detectable subjects for the pivotal study. C, lack of standardization in model optimization for software programs. D, all of the above. So the answer is all of the above. And challenge question 2. What is the impact of selecting an incorrect ED50 value in vasoconstrictor studies? A. It reduces the sensitivity in dose-response relationship for highly potent corticosteroids. B. It leads to fewer subjects meeting those duration response criteria for low-potency corticosteroids. C. It causes incomplete plateau levels in vasoconstrictor response. D. All of the above. Yeah, the question is all of the above. The summary. The first point is background and challenges. Vasoconstrictor study in corticosteroid development face challenges in determining ED50 due to very parameter estimations and bias values. Second is impact of incorrect ED50 selection is high ED50 lead to fewer subjects meeting those duration criteria, while low ED50 requires more subjects for reliable results. Estimation model and vasoconstrictor response challenges. Differences in models estimation and weak responses in low potency drugs affect ED50 accuracy. The last one is deficiencies in studies, significant difference in ED50's estimation between agency and applicants, requiring analysis and addressing unreliable subject selection. Here are some resources. The first one is 1995 guidance. The second one is the 2022 drug guidance. Within the revision of the guidance, there are guidance and reference were produced. The first one is POP-PK guidance. The second one is exposed-response relationship guidance. The third one is concerning the FDA 1995 guidance. The last one is the introduction to PD modeling methods. Finally, we have closing thought. Choose an appropriate software platform, adhere to the population modeling process, and carefully determine the ED50 to ensure optimal sensitivity in the dose-response relationship. For any question, we can discuss in the panel discussion. Thank you so much for your listening.
Thank you all for the great presentations. If you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. In addition to our previous speakers, we welcome the following to the Q&A panel: Dr. Markham Luke, Division Director, Division of Therapeutic Performance 1, Office of Research and Standards in OGD; Dr. Sam Rainey, Associate Director for Science, Office of Research and Standards in OGD; Dr. Prahala Simamora, Division Director, Division of Liquid-Based Products 2, Office of Life Cycle Drug Products, Office of Pharmaceutical Quality; and Dr. Rong Wang, Associate Director, Division of Bioequivalence 1, Office of Bioequivalence in OGD. Looks like we have a few questions coming in right now, and the first two questions are addressed to Dr. Sam Rainey. And here's the questions. How would you define a Q3 similar product, and can Q3 differences be justified based on other data sets, for example, IV-RT and IV-PT?
Thank you, Ray. I appreciate the question. So let me tackle the first one first. So how would you define Q3 similar product? So in its simplest kind of conceptualization, for a Q3 similar product, the Q3 attributes of both products are the same, but there's some difference in the Q1 or the Q2 a difference that's beyond the no significance difference criterion that you heard us talk about a little bit earlier. So it's a Q3 similar product because the Q3 attributes are actually the same. Now, there is a nuance here that there can be a situation where the Q3 attributes are not all exactly the same, that there's some variation in the Q3 attributes that's beyond the range that the applicant has characterized in the reference product, but it's within the range that FDA has already determined to be acceptable variability for that product. For example, let's say a prospective generic might say, we've measured the reference product, different batches to be pH 7, 7, and 7.1, and our batches are 7, 7.1, and 7.2. It's outside the range of what we characterized in the reference product, but FDA may have information to say, well, we actually consider this to be acceptable for the reference product. In that instance, although there's a characterized difference in the Q3 attributes, it would be considered that all the Q3 attributes are essentially within the acceptable range, and therefore this is a Q3 similar product. In terms of whether an IVPT study can be used or IVRT study can be used to justify differences in Q3, the short answer is yes, but let me unpack that a little bit too. So first, to clarify what we mean by Q3 differences. So on the face of it, if there are some differences in the profile of Q3 attributes between two products, then they have different physical, chemical, and structural arrangements of matter, and they would generally be considered Q3 different products, not same or similar, but actually Q3 different products. So, for example, can IVPT help justify differences in Q3 attributes for two topical gels? Well, the evidence that's needed is evidence to systematically mitigate the risks that are associated with the specific differences in Q3, and an IVPT study may actually provide compelling evidence to mitigate the risk that any of these differences in Q3 or Q1 or Q2 between the gels may impact topical, local bioavailability. And so, yeah, an IVPT study could be very helpful in that instance. And for a particular product, so that was just one example, but for you as an ANDA applicant, for example, if you wanted to know specifically about your product, the best way to figure that out is really probably with the pre-ANDA meeting. And if you're proposing an IVPT study as part of your demonstration of BE, I'd really encourage you to include some pilot IVPT results in your pre-ANDA meeting package, both to demonstrate the feasibility of your proposed approach with an IVPT study, and also so that FDA can provide some timely feedback to help you ensure that your IVPT method is developed and validated in a manner that's compatible with what FDA would recommend. But for most differences in Q3, a passing IVRT study would not likely be enough, but IVPT may not always be necessary either. And so there are other characterization tests like even modeling and simulation data that could be sufficient. So I guess I'll close this out by saying that sometimes when people talk about Q3 differences, when referring to minor variations in Q3 that may exist for Q3 similar products, then an IVPT study may not be needed. There are instances in which, for example, the reference product may have undergone a post-approval change that altered its rheological behavior. FDA has already determined that the broader range encompassing both the old and the new ranges of rheological profiles are acceptable for the reference product. So as long as the new test product's rheological behavior falls within that broader range, then it may be adequate without any further evidence. Or there may be instances, for example, there's particle size distribution differences, but it may be possible to justify that using modeling and simulation evidence. So it really depends on the nature of the product, the nature of the Q3 differences, and how those differences impact the physicochemical properties and the mechanism of action. So I hope that answers the question.
Thank you, Dr. Rainey, for responding to that question. Moving on to our next panelist, we did have a new question come in for Dr. Markham Luke. And here is the question. What is the difference between RS and RLD FDA drugs designation? Are both valid as a comparator in BE studies or for demonstrating equivalence of generic drugs?
Thank you, Ray. That's a good question. So the reference listed product is the a reference listed drug is the drug that's listed in the Orange Book. The list refers to the list of drugs that's printed in the Orange Book. If the reference listed drug is taken off the market, then a reference standard is proposed or put in place of the reference listed drug as the comparator product for the wannabe generic applicant. Of note, sometimes the reference listed drug product comes back on the market, so you may have an interposed time where you might have both present. A appropriate comparator for a generic is what's available on the market currently, and that's if the reference listed drug is off the market and only the reference standard is available, then that's the comparator. Of note, it's really important to look at what's available, and if you're going to be doing Q3 evaluation testing, you want to make sure that the product is an available product that you're comparing your product to. So if the reference listed drug is no longer available and you're relying on the ingredient list and the labeling for the reference listed drug, you would have some problems comparing your product if the for Q3 if the reference listed drug is no longer available for comparison. So and it also helps if you want to substitute ingredients or excipients in the context of achieving Q3 sameness, etc., to have a pre and a meeting or control correspondence submitted for further evaluation and for FDA to weigh in on what kind of concerns we might have if they deviate from what is usual practice. Thank you.
Thank you, Dr. Luke, for responding to that question. We do have more questions coming in. The next question is addressed to two of our panelists. First to Dr. Ahmed Zadan, and then passing the baton over to Dr. Priyanka Ghosh. And here is the question. Please advise guidance regarding excipients, and referring to slide number 3, such as approved by FDA, or is there also a quantity limit?
Yeah, so excipients and topical drug products are really a complex issue, and based on most of the PSGs showing no significant difference regarding the excipients of the generic to RLD may be demonstrated using qualitative and quantitative methods. But the point here is some of these excipients are really complex excipients, and it may be formed of different components in one excipient, and it may have different functions also in the performance of the drug product. So showing similarity of the excipients in terms of quantity, several reverse engineering research may be done to justify sameness regarding quantity. And of course, the applicants are encouraged to submit a controlled correspondence of 3 formulation suggestions where FDA can assess the similarity at 5% difference in quantity of some excipients. But in other excipients, identifying the excipients qualitatively may be challenging, and a combination of evidences and the research study may be used. For example, for polymers and cross-linked polymers that's used in topical drug products, a summation of evidences showing the same, for example, molecular weight distribution, like viscosity, some of the qualitative measures to describe the excipients may demonstrate sameness in some cases, and this of course contribute to the Q3 similarity.
Thank you so much, Ahmed. I would just like to add to that when utilizing a characterization-based approach, like Dr. Zidan said, we typically compare the proposed formulation to the reference listed drug. And if you think that your formulation has values higher than the reference listed drug, it is always a good idea to look at the inactive ingredient database that we have. It has information on maximum potency or maximum daily exposure for a given inactive ingredient. And you may take those into consideration while you are designing your drug product. On the other hand, if you are designing a drug product for the when utilizing the comparative clinical endpoint bioequivalence studies, we still strongly encourage you to look at the inactive ingredient database, often alluded to as the IID, and make sure that either your proposed levels are lower than what has been accepted in previously approved FDA products, or if you'd like to propose higher levels, we encourage you to send in control correspondences to assess if those levels would be acceptable by the agency. Thank you.
Thank you, Dr. Ghosh, for following up on that question. And we do have another question for Dr. Ghosh. And here's the question. For an IV RT altered batch, can we change the formulation, for example, from a gel to a cream to prove discrimination?
Thank you, Ray. So I will answer the question in two parts. For IVRT altered batch, if you are referring to the batches of the product that are used for method validation studies, such as sensitivity, selectivity, and specificity, we encourage you not to change the dosage form. Specifically, in those studies, we encourage you to use altered formulations where you have changed the concentration of the active ingredient and the corresponding diluent to 50% and 150% of the nominal strength of the reference product. If you were referring to supplemental selectivity studies, in those studies, we encourage you to use altered formulation that has differences in formulation, but differences in formulation, like you can use a cream if your target formulation is a gel, but in those cases, we do encourage you to keep the concentration of your active pharmaceutical ingredient the same between the gel and the cream. Thank you so much.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Hiren Patel. And here's the first question for Dr. Patel. What are your thoughts about using 2 doses higher than the normal dose for an IV PT study?
Thank you for the question. I'm not quite sure that 2 doses higher is in reference to 1 dose amount that is 2-fold higher than the nominal dose amount, or 2 different dose amounts that are higher than the nominal dose amount. I will touch base on both aspects. In my opinion, the conduct of IVPT sensitivity using 1 lower and 1 higher dose amounts compared to nominal dose amount assures that the IVPT method can detect the differences in permeation profiles on either side of the nominal dose amount. As mentioned in the guidance, 3-fold differences in the dose amount is a good starting point, but if it is higher or lower fold than the recommendation, and if the data shows well discrimination in permeation profiles, it may be considered acceptable. On the separate note, while selecting the 2 higher dose amounts provides the comparison of a total of 3 dose amounts where the nominal dose amount is at the lower end, it may not necessarily add weightage to the validity of the method. In this regard, the guidance also indicates that the difference in the IVPT permeation profiles are not necessarily expected to be specifically proportional to the difference in the dose amount. If there are challenges, I would encourage to utilize appropriate avenue to reach out to the agency related to the additional feedback for your specific product development question. Thank you.
Thank you for responding to that question. I've got a few more questions for Dr. Patel, and here's the next question. For IVPT studies, some donors show Jmax at either first sampling time point or last sampling time point. Would you please share your perspective on how critical this observation is, and its impact on IVPT studies?
Thank you. It is a good and loaded question. I'll try my best to break it down and answer it. I understand that the general guidance indicates that IVPT endpoint, that is Jmax, is somewhat analogous to the pharmacokinetic endpoint of Cmax. However, there are some distinct differences. Specifically, Cmax represents the concentration at that particular sampling time point. However, Jmax represents the average permeation rate between two sampling time point or duration. In this regard, there are some unique observations related to the Jmax as mentioned in the question, and I will break it down into two aspects. The first one is Jmax at first sampling time point. This may be potentially related to the frequency of sampling at early time points. In my opinion, if this is observed during method development, it would be a good idea to optimize the sampling time point, specifically at the early time points. On the other hand, if it is observed in validation or pivotal IVPT studies, after well-developed IVPT method for which there was no Jmax at first sampling time point, and it is not related to the methodology or procedure, for example, issues related to the dosing application or spreading procedure, it may be concluded that this observation is specific to one particular donor where similar observation is noted across different treatments and may be justified. In terms of last sampling time point that provides Jmax, it may be potentially be related to the study duration. Similarly, if it is observed during method development for all donors, as I mentioned earlier, an alternate approach like wiping off the dose after certain durations should be considered and methods should be optimized. If it is observed for validation and pivotal study and it is specific to the donor, it may be justified. The take-home message is that it is critical to optimize the IVPT method as precisely as you can, and if it is observed for later studies and as long as it is adequately explained that it pertains to the donor and not the IVPT method, in my opinion, it does not make the IVPT method inadequate. Thank you.
Thank you for responding to that question. We have one more question for Dr. Patel, and here's the question. Do you have general recommendations for the selection of acceptance criteria for skin barrier integrity testing?
Again, thank you for the question. As I mentioned in the presentation, the new general guidance on IVPT have detailed recommendations for different types of skin barrier integrity testing with specific recommendations for the conduct of the study and what to avoid. In my opinion, the testing condition generally impacts the results. Therefore, one should have an experimental data to characterize the normal distribution of integrity testing values among multiple replicate skin sections from multiple donors using the selected methodology, which illustrate where the cutoff value is positioned relative to the normal distribution of the values and whether it discriminates from the intentionally compromised skin section. In my opinion, it may not be necessary to have this data for different drug products or studies as long as it's utilized the same experimental condition throughout the study or the drug product. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we have a few questions that came in for Dr. Megan Kelchin. Here's the first question for Dr. Kelchin. There are several PSGs that call for a comparative clinical endpoint study rather than a characterization-based BE approach. Will those guidances be changed to alternate bioequivalence approaches in the future?
Thank you for the question. I want to first emphasize that a comparative clinical endpoint study is a viable bioequivalence approach for many drug products, in particular those prospective generic products that don't meet the no significant difference standard that's recommended as part of the characterization-based approach. Additionally, there are a few situations where a characterization-based approach may not be amenable to a specific drug product, and for those drug products, a comparative clinical endpoint study is the viable bioequivalence option at this time. As I mentioned in my presentation, we are actively working on incorporating characterization-based approaches into both new and existing PSGs, including those that currently only include a comparative clinical endpoint study. But in the meantime, if you would like to propose a characterization-based approach as an alternative bioequivalence approach to what's in the PSG, we encourage you to submit a pre-ANDA product development meeting request to get feedback from the agency on your proposed approach. Thank you.
Thank you for responding to that question. We've got the next question for Dr. Kelchen is the following. Is there a schedule for new updated PSG publication, or can FDA release ad hoc PFGs? If so, what are the key driving factors for this?
Thank you, Ray. In general, batches of PSGs are published on a quarterly basis, with batches posting generally in February, May, August, and November. There are some rare situations where a PSG might be published outside of these batches. An example of this would be when a PSG is posted alongside with the agency responding to a citizen petition for a specific product. But again, generally, most PSGs are published as part of a batch, again, on a quarterly basis. I also want to highlight that the agency maintains a list of PSGs that the OGD plans to publish or revise over the next 12 months. And so I encourage you to review that list to see what PSGs are on our list. Thank you.
Thank you for responding to that question. We have one more question for Dr. Kelchen, and here's the question. Would you elaborate on cases where IV-PT and IV-RT may be critical and recommended in support of demonstration of BE?
Thank you. So for a characterization-based bioequivalence approach, an IVRT is generally recommended for all topical products that are applied to the skin. For an IVPT study, those studies are generally recommended for topical products that are applied to the skin for more complex dosage forms where we can expect complex interactions between the drug product and the skin. So as I showed in my presentation, an example of these might be drug products that are multiphasic in nature, or drug products like petrolatum-based ointments that contain a large concentration of a complex and active ingredient. However, as I showed, there are situations where an IVPT study may not be necessary for these dosage forms due to, say, the indication of the drug product or for other reasons. So if you have a question about the need for an IVPT study for a specific drug product, we encourage you to submit a pre-ANDA product development meeting request for that product. Thank you.
Thank you for responding to those questions. Moving on to our next panelist. We've got a few questions that came in for Dr. Ahmed Zidan. And here's the first question for Dr. Zidan. Would you elaborate on the considerations of metamorphosis of topical products and the in vitro demonstration of BE for multiphasic topical products?
Yeah, sure. It's a nice question. So metamorphosis of topical drug product is a significant attribute, I would say, as it is applied to the skin surface. So I would say that the environmental condition affecting the changes to the drug product as it is rubbed to the skin. And of course, it can change the dynamics of drug release and percutaneous absorption that's happened after this metamorphosis. So demonstration of metamorphosis attributes, it depends mainly on the understanding of the formulation and the critical formulation attributes and the type of excipient that's used in in manufacturing this product. So, for example, water loss due to evaporation can increase API concentration in a continuous phase of multiphasic creams or ointment, and this may result in increasing flux for permeation, and of course it may change the overall in vivo performance of the drug product regarding the safety and efficacy profile. So characterizing metamorphosis using various analytical methods may be a tool to ensure that generic product have the same performance or Q3 characteristics to the RLD. And there is an advancing technique to study the metamorphosis where it is a dynamic process. For example, using vapor sorption or using spectroscopic and chromatographic tools and kinetics of solvent evaporation from the formulation may be like a nice tools to characterize metamorphosis.
Thank you for responding to that question. Moving on to another panelist. We've got a question just came in for Dr. Key Ren. And Dr. Ren, here is the question. For detector identification in topical corticosteroid BE evaluation, the criteria AUEC of D1 over D2 greater than 1.25 is only for simple Emax model, or does it also stand true for the sigmoidal Emax model?
Thank you for the question. That's an excellent question. So the detector criteria of 1.25 and both mean AUEC value at D1, D2 should be negative is also applied to sigmoid Emax model. But the only difference is the calculation of the AUEC for D1, D2 between the simple Emax and the sigmoid Emax model. So the calculation should be justified. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a couple of questions that came in for Dr. Kevin Fang. And here's the first question for Dr. Fang. The SAS algorithm fails to show changes in A1C values limitation, while it is not sensitive to parameter normality assumptions and strength. In this regard, which one of the SASR non-MEM is a preferred or recommended software for DD50 estimation for topical corticosteroid BE assessment, in your opinion?
Thank you, Jeff. As you can see from the presentation slide, the SAS did not produce the sensitive result for AIC when the parameter normalization assumption changed. So in that case, that FDA will recommend it is essential to choose a software platform that supports clearly defined model selection processes like Emax model selection, comparison of estimation methods, selection of model parameters like error model, initial estimation, and appropriate model diagnostic to overcome those challenges. So those of competitive software, non-MEM would be a preferred software. Thank you.
Thank you, Dr. Feng, for responding to that question. We do have another question for you, and here is the question. Which software referred by FDA for ED50 estimation when the agency raised a query, agency's ED50 estimation is different than the applicant's estimated ED50? Is it the same which applicant has used for estimation, or if different, will the agency disclose the software used at their end?
Yeah, thank you, Jeff. That's the question is really the crucial question that we are looking at. When we receive the submission package, we were using the same software as possible to evaluate whether we can get the same results. And also, we want to looking at some of the optimization whether the ED50 have been optimal. If the software is not capable, then we would looking at some of the software that we're using capable for the optimal of the ED50. That's the procedure that we normally we're looking at some of the software like as we have been mentioned, the Numen, Phoenix, and Actual. Those software is capable. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we have a question that's directed to Dr. Pahala Simamora. And here is the question for Dr. Simamora. When testing Q3, what are requirements regarding the validation of Q3 testing methods?
Thank you, Jeff. As we know, the Q3 property test method, they are called non-typical method as they are not addressed in ICH Q2R1. Some example are global size, particle size, viscosity, just some example. But generally, the requirements in the USP general chapters method are recommended. A couple of examples for USP 429 for particle size test, that one's light diffraction measurement, and USP 776 for the optical microscopy. And then in terms of the method validation parameters for Q3 procedure, we may want to include some examples: system stability, precision, accuracy, robustness, as applicable depending on the instrumentation. Thank you.
Thank you for responding to this question. We received another question for our next panelist, Dr. Rong Wang. And we're going to try to make some clarity out of this question. And here is the way the question is written. And it is referring to ED50. Can that be carried by the originator or the reference product to be part of the data? And is this done in order to have more generics available with a lower cost?
Thank you, Jeff, for the question. I will try to provide an answer here based on my understanding of this question. So the purpose of the pilot study is to determine the dose duration response relationship of the topical corticosteroid to be studied in the pivotal VCA study using Emax model in the pilot study to estimate the ED50 of the reference product is like using a standard curve to characterize the linearity range and the LLOQ limits of the quantification in a bioanalytical method. So ED50 represents the center of the sensitive portion of the dose duration response curve in Emax model curve fitting. Therefore, the pivotal study should be performed using the most sensitive dose duration, which is the dose duration approximately corresponding to the ED50 under the same conditions as the pilot study to detect the difference between test and the reference product. So that's why we use the ED50, the dose duration corresponding to the ED50 from the pilot study in the pivotal bioequivalence study. Thank you.
Thank you, Dr. Wong, for responding to that question. We've got a couple more questions that came in for you, and here is the next question. Is it necessary to perform a randomization among the study subjects in a VCA study?
Thank you, Jeff, for the question. The short answer is no. Randomization is conducted among skin sites on each arm within each subject. So for pilot study, various dose durations and untreated sites, usually 2 sites of untreated sites are randomized among skin sites on each arm within each subject. For pivotal study, it is recommended that 2 sites of treated control and 8 treatments, including 2 sites of the reference standard at the dose duration corresponding to ED50 and 2 sites of the test product at the same dose duration and 2 sites of the D1 dose duration of reference standard and 2 sites of D2 dose duration of reference standard should be randomized on each arm of each subject. And application patterns on each arm in the pivotal study should be complementary as well, meaning D2 is complementary to D1 and the reference standard is complementary to test product, and untreated control is complemented to untreated control on each arm. So please refer to the VCA study guidance for detailed instructions on randomization. Thank you.
Thank you, Dr. Wong. We have one more question. It's referring also to the VCA study, and here's the question. How much of the formulation should be used in a VCA study?
Thank you for the question. There is no requirement for the amount of a drug product applied to the site. The guidance includes an example. Investigators may use doses of 5 to 12 microliters of formulation per centimeter squares of skin surface area and 1.6 centimeter diameter sites. Based on the in-house ANDAs, and our observation is that the median of dose per site is 20 microliter, ranging from 10 to 40 microliter. Thank you.
Thank you for responding to that question. Moving up to the beginning of our panel, we have another question that came in for Dr. Priyanka Ghosh, and here's the question for Dr. Ghosh. If there has been a change to an RLD during the life cycle of a PSG, are the suitability of the recommendations in that PSG reassessed against that change?
Thank you, Ray, for the question. As Dr. Rennie mentioned in the first response, we do take into consideration the entire life cycle of that product when developing the PSG and during subsequent revisions. However, this question appears to be very specific. So if you have noted any changes during your characterization of a reference product and you don't think they are reflected in the PSG, we encourage you to discuss with the agency using appropriate communication pathways. Thank you.
Thank you for responding to that question. We've just got a couple of minutes left. I'm going to try to get in one more question that just came in for Dr. Megan Kelchin. And here's the question. Can you comment on the role of a systemic pharmacokinetic study in a characterization-based approach for topical products?
Thank you for the question. In general, a systemic pharmacokinetic study is included as part of a characterization-based approach when there's a question of whether the systemic drug concentrations, in addition to the local drug concentrations in the skin, contribute to the efficacy of the product. And so for those products, and there's just a few of them at this time, but for those products, a pharmacokinetic study is recommended to capture any differences in the systemic absorption between the test product and the RLD. Thank you.
Thank you for responding to that question. And that's all the time we have for questions in this panel. And a huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We'll now enter into our lunch break until 1 p.m. Eastern Time. Enjoy your lunch. Welcome back from the lunch break. We have just a few housekeeping notes and reminders that we'd like to share to get us started. This activity is eligible for an attendance certificate which supports continuing education by SOCR, RAPS, SQA, and ACRP. Please refer to our website at fda.gov/SBIA for more details. You'll be able to obtain the attendance certificate only upon completion of the survey, which will remain open for 2 weeks. Remember, that's September the 28th. Please remember to download the certificate immediately upon completing the survey. It cannot be retrieved after the fact. Let us know your comments and feedback via the survey. This will help guide the design of our future events. To summarize, for our physicians, pharmacists, and nurses claiming the 13.25 continuing education credits, attendees have 14 days from last day of the activity to log in, complete the required CE evaluations, and attest to attendance to claim credit. So be sure to complete the CE session evaluation on or before September the 28th. As a reminder, this email we sent to attendees later today will have information on both the CE claim code and the survey link. Moving into our next session on noteworthy guidances for nasal suspension and inhalation products. We'd like to introduce our presenters for this session. First, we'd like to welcome Dr. Susan Bach, pharmacokineticist in the Division of Therapeutic Performance 1, Office of Research and Standards and OGD. Next, we'll hear from Dr. Ross Wallinga, Senior Chemical Engineer in the Division of Quantitative Methods and Modeling, Office of Research and Standards and OGD. Our third presentation will be led by Dr. Nathan Reed, Chemist, Division of Complex Drug Analysis, Branch 2, Office of Testing and Research, NOPQ, and then Dr. Elizabeth Bielski, Senior Pharmacologist, Division of Therapeutic Performance 1, Office of Research and Standards and OGD. Please join me to welcome our first presenter in this session, Dr. Susan Bach.
Good afternoon. My name is Susan Bach, and I am a reviewer in the Division of Therapeutic Performance 1, Office of Research and Standards within OGD. Today I will be speaking about the utilization of a newly recommended in vitro-only bioequivalence option provided in complex nasal suspension product-specific guidances. Learning objectives are to explain the rationale for the revision of product-specific guidances, PSGs, on locally acting nasal spray suspension products, to describe the two options to establish bioequivalence, BE, for these products, and to list the newly recommended in vitro studies and the key considerations for each study. GDUFA-funded research supported the revision of 9 PSGs on locally acting nasal spray suspension products. Research findings included that Raman spectroscopy was capable of characterizing drug-specific particle size distribution, PSD, of nasal suspensions. Dissolution studies using various systems, USP apparatus 2, USP apparatus 5, Transwell, were sensitive in detecting differences in drug PSD. And pharmacokinetic PK studies were sensitive in detecting differences in drug PSD. A list of the 9 revised PSGs are shown here and available at FDA's PSG webpage. The revised BE recommendations now include 2 options based on qualitative Q1 and quantitative Q2 sameness of test and reference listed drug RLD product formulations. For the first option, option 1, in vitro BE studies are recommended for formulations that are Q1 and Q2 the same. This includes the traditional 6 in vitro BE studies: single actuation content, droplet size distribution by laser diffraction, drug in small particles droplets, spray pattern, plume geometry, and priming and repriming. And the addition of 2 new in vitro studies, drug particle size distribution and dissolution, combined to make up option 1. For option 2, in vitro and in vivo studies are recommended for establishing BE of formulations non-Q1 and Q2 the same. These include the 6 in vitro BE studies and 1 comparative PK study and one comparative clinical endpoint study. The remainder of this talk will focus on the two newly recommended in vitro studies, drug particle size distribution and dissolution. Let's first discuss the considerations for drug PSD characterization. Recommendations for drug PSD characterization include use of an optimized and validated analytical method such as morphologically directed Raman spectroscopy, MDRS. MDRS is an integrated platform that measures particle morphological characteristics using its microscopic component and performs chemical identification via Raman spectroscopy. The basic measurement procedures of MDRS, as shown in the figure, include sample preparation, particle imaging and morphology analysis, using morphology filters to select particles of interest, identification of particles using Raman spectra, and size measurement of the particle of interest. One consideration when conducting drug PSD with MDRS is sample preparation. Samples should be prepared to ensure that drug is in its suspended state post-actuation. This is because a dry dispersion may cause particle aggregation upon solvent evaporation and consequently yield inaccurate particle count, size, and morphology data. An example wet dispersion sample method preparation for MDRS is described here. First, shake and prime the nasal spray. Next, collect an optimized number of actuations into a glass vial. Then pipette a set volume of the collected sample onto a microscope slide and cover with a cover slip to form a thin layer. After, seal with petroleum jelly or similar substance along the edge of the cover slip to prevent solvent evaporation. Make sure to let the sample rest for a period of time to allow particles to settle before analysis. Ideally, the touching particles should be less than 5% of the total number of particles. For particle imaging and analysis, provide optimization and validation data for imaging settings. Aim for a threshold level that correctly identifies the particle edges. This should be when a thin border completely surrounds the perimeter of the particle as shown in the second illustration. The minimum number of particles to be measured is an important attribute regarding whether the drug PSD results are statistically meaningful and representative of the product. Drug PSD measurements with different particle counts should be compared to determine the minimum number. When enough particles are counted, the drug PSD results from repeated measurements should be stable. In this example, repeated measurements were stable when a number of particles analyzed reached about 400. The optimization and validation data should be provided for morphology filter selection. The use of analytical filters prior to Raman measurement should reduce total analysis time. Select filters and appropriate cutoff values to eliminate as many excipient particles as possible, while minimizing the number of drug particles excluded from the analysis. As an example, mometasone furoate nasal spray was used as a model drug product, which has drug particles as mometasone furoate and excipient particles as microcrystalline cellulose suspended in the formulation. A selection criterion of at least 85% of drug particles to be retained after applying the filters with appropriate cutoff values was set. A training set of 10,835 particles was used in this study. Of these, 1,335 particles, or 12.3%, were identified as drug particles by Raman analysis. The remaining were identified as excipient particles. When applying different morphology filters, aspect ratio and intensity mean were shown to be the two most efficient parameters to separate the two types of particles. In the plots, the drug particles are shown in blue and the excipient particles are shown in black. Drug particles were found to have a larger aspect ratio and a lower intensity mean. Cutoff values were initially set to a lower boundary of 0.6 for aspect ratio and an upper boundary of 150 for intensity mean, based on the points where the morphology distribution curves intersect. However, as shown in the table, this resulted in only 77.5% of the drug particles being retained in the measurement. Adjustments to cutoff values found two combinations for aspect ratio and intensity mean filters that resulted in more than 85% of the drug particles being retained in the measurement. The The aspect ratio filter with the lower bound of 0.6 and intensity mean filter upper bound cutoff of 160, as shown in green, and aspect ratio filter with a lower bound of 0.55 and intensity mean filter upper bound cutoff of 155, as shown in blue, were found to retain 87.6% of the drug particles for analysis. Validation of the filter selection should be checked by comparing drug PSC results before and after applying filters with, ideally, a less than 3% difference. The morphology distribution curves for the drug and excipient particles are again shown here for aspect ratio and intensity mean. The table below shows the size distribution of particles when no filters are applied and when the two combinations of aspect ratio and intensity mean filters are applied. The percent difference before and after applying the filters are shown in parentheses. As can be seen, the filter combination with the aspect ratio lower bound cutoff of 0.6 and intensity mean upper bound cutoff of 160, shown in green, resulted in similar particle size values to when no filters were applied, as indicated by the less than 3% difference. For the second combination, shown in blue, the percent difference for the D10 particle size was greater than 3%, specifically 7.6%, as compared to when no filters were applied. Thus, this filter combination at these cutoff values should not be selected based on the proposed criteria. The optimization and validation data for Raman spectroscopy settings should be provided. This may include settings such as exposure time, spectral correlation range, and Raman spectral correlation score. For the exposure time, the longer the exposure time, the better the Raman spectrum quality, so considerations should be made to balance the quality of the Raman measurement and the total experiment time. To improve the signal-to-noise ratio, compare the collected spectra to the reference library standard within the spectral correlation range specific for the drug of interest. Continuing with the example of metazone furoate nasal spray as the model drug, the image shown on the right is the Raman spectral reference of the drug in black and the excipient microcrystalline cellulose in blue. There are 4 signature peaks for metazone furoate with no overlapping excipient peaks in the spectral range of 1350 to 1750 inverse centimeters. Thus, this region should be selected as the correlation range for classification of drug particles. For drug particle classification, the cutoff for the spectral correlation score should be determined based on the limit of detection for evaluation of particle Raman spectra. Shown here are comparisons of particle spectra grouped based on Raman spectral correlation scores, which are highlighted in green. The selected correlation region of 1350 to 1750 inverse centimeters is depicted with a white background. Selection criteria of at least one of the 4 drug peaks had to be observed with acceptable signal-to-noise ratio proposed as greater than 3. The plot on the left shows drug particles with correlation scores between 0.56 to 0.59. The plot in the middle shows drug particles with correlation scores between 0.61 to 0.64. And the plot on the right shows drug particles with correlation scores between 0.66 to 0.7. It can be seen that with increasing correlation scores, moving from left to right, there's improved signal with at least one of the 4 drug peaks observed in the middle and right plots. Thus, a correlation score of greater than 0.6 was selected for identifying drug particles. As mentioned in the revised PSGs, an orthogonal method may be required if the selected methodology to measure drug PSC is not sensitive to measure particles beyond a certain size range. With MDRS, the lowest detectable size for the Raman component is 1 micron, which may require the use of another method to assess the submicron particles. For the first approved generic metazone furoate nasal spray, the applicant submitted drug PSC as measured in the drug product using MDRS. As one of the orthogonal methods to assess submicron particles, the applicant submitted particle size data up to 0.5 microns using the Morphology G3 instrument, which lacks the Raman component. In addition, the percent of particles below 0.5 microns, as determined by laser diffraction, was less than 1% for both generic and RLD products. Given the totality of evidence, the MDRS method was deemed acceptable to compare the drug PSC between test and RLD metazone furoate suspension spray products. Now let's look at considerations for dissolution studies. An appropriate apparatus may be used to determine dissolution measurements. Example apparatuses include the USP General Chapter 711 Apparatus 2, also known as the paddle apparatus, the USP General Chapter 724 Apparatus 5, also known as paddle over disc, or the transwell system. It should be noted that the suggested apparatuses allow for studies to be conducted under sink and non-sink conditions. Thus, potential applicants should take this into consideration when selecting an appropriate dissolution apparatus. Dissolution studies should use a sufficiently developed and validated method to support its sensitivity in detecting differences in performance between tests and RLD products. Dissolution media selection should be guided by solubility investigations and be optimized to be discriminatory. Stability of the drug substance should be carried out in the selected dissolution media alone and in the formulated drug product. Enough time points should be selected to adequately characterize the ascending and plateau phases of the dissolution curve as shown in the figure on the right. The example shows mean dissolution profiles plotted as % cumulative mass over time of 4 formulations compared to the RLD. The dissolution method should demonstrate discriminatory ability, such as the ability to detect meaningful differences in formulation or manufacturing processes, such as a difference in drug particle size. The goal is to understand release mechanisms and determine whether the dissolution procedure can show changes in critical quality attributes of the drug product. Thus, dissolution studies should ideally be conducted on formulations intentionally manufactured with meaningful variations for the most relevant critical manufacturing variable, or on stress samples. For example, the table shows the drug PSD in the final product of several nasal suspensions formulated to be similar to the RLD product, but prepared with different drug particle size. As seen in the plot, the dissolution method with the USP Apparatus 2 was able to differentiate the nasal suspension formulations with different drug particle size. For BE assessment, comparative analysis of dissolution profiles should be established using an appropriate statistical method, such as model-independent approach using a similarity factor, F2. F2 analysis allows for comparison of the dissolution profile rather than single-point dissolution comparisons. It should be mentioned that other statistical methodology may be used by providing appropriate statistical rationale with adequate justification. Continuing with the example of the nasal suspension formulations formulated with different drug PSD, F2 analysis was conducted on the average dissolution profiles of the different formulations for the first 60 minutes. Generally, F2 values should be close to 100, with values greater than 50 ensuring equivalence of the two curves. As shown in the table, nasal suspension formulations 3 and 4 were found to be similar to the RLD with F2 values of 59 for formulation 3 and 62 for formulation 4. Formulations 3 and 4 were also determined to be similar to each other, as indicated by the F2 value of 54. In summary, based on GDUFA-funded research, PSGs for locally acting nasal spray suspension products were revised to include 2 BE options. Option 1 is an in vitro-only pathway to demonstrate BE for test and RLD product formulations that are Q1 and Q2 the same. Option 2, which includes in vitro and in vivo studies, provides a pathway for test and RLD product formulations that are not Q1 and Q2 the same to demonstrate BE. Key considerations for drug PSD characterization using MDRS include sample preparation, particle imaging and analysis, morphology filter selection, and identification using Raman spectroscopy. Key considerations for dissolution studies include apparatus selection, method development and validation, and BE assessment. There are 2 challenge questions for this topic. For challenge question number 1, how many in vitro bioequivalence studies are recommended when the test product's formulation is qualitatively and quantitatively the same as the RLD formulation? Option 1. Is it A, 6, B, 7, C, 8, or D, 9? The correct answer is C. There are 8 in vitro bioequivalence studies recommended for option 1. For challenge question number 2, which of the following statements is not true? A. There are no in vivo studies recommended in the product-specific guidances on locally acting nasal spray suspension products. B. Sample preparation for drug PSD characterization should ideally be as a wet dispersion to maintain the drug in its suspended state. C. In vitro and in vivo BE studies are recommended for test product formulations that are not Q1 and Q2 the same as the RLT formulation. Or D. Recommendations for dissolution studies include the use of USP and non-USP dissolution apparatuses. The correct answer is A. There are no in vivo studies recommended in the product-specific guidances on locally acting nasal spray suspension products. I would like to thank my colleagues who have helped in the development of this research and who have contributed to this presentation, and thank you for your attention.
Hello, my name is Ross Valenga. Today I'm going to be speaking about complex nasal suspensions, utilization of in silico studies to support development and approval. I have 3 learning objectives for today's presentation. First, I'd like for you to be able to describe the utility of computational fluid dynamics, or CFD, and physiologically based pharmacokinetic, or PBPK, modeling for nasal drug product development. Next, I'd like for you to be able to understand how CFD and PBPK models for nasal suspension drug products are validated. Lastly, I'd like for you to be able to identify in vitro metrics that are predicted to influence posterior nasal cavity drug delivery. Considering the bioequivalence approach for nasal suspension drug products, recommendations from the U.S. Food and Drug Administration include 2 options for nasal suspension drug products. The first option includes only in vitro studies, as you can see in the list on the right, which includes single actuation content, droplet size distribution by laser diffraction, drug and small particles and droplets, spray pattern, plume geometry, priming and repriming, drug particle size distribution, and dissolution. There are also recommendations for qualitative and quantitative sameness, or Q1, Q2, and that the test device, or T device, is appropriate for an abbreviated new drug application. The second option includes in vitro and in vivo studies as listed below, but does not include Q1, Q2 sameness. So the first 6 in vitro studies of the previous list are included here, as well as 2 in vivo studies that include a comparative pharmacokinetics, or PK study with fasting, using a 2-way crossover design in healthy subjects, as well as a comparative clinical endpoint study. Considering those recommendations, why might a modeling approach be useful? Thinking about the value of in silico models, in this instance, an in silico model could be used to investigate the influence of device and formulation differences on regional deposition and absorption. If option 2 were to be selected for demonstrating bioequivalence, you could use the model to predict impact of formulation and device changes on PK. You also could take existing modeling techniques, modify those, and then use those to predict in vitro study outcomes such as spray pattern and plume geometry. Another application that I won't be speaking about today is that these models can be used to predict olfactory region absorption if you have a drug product that's intended for nose-to-brain drug delivery. This is expected to be an emerging area in the coming years, and modeling may play a significant role. Thinking about CFD modeling for nasal drug products, the way these models work is that they can predict the influence of device and formulation parameters on drug delivery to the site of action. The CFD model uses a physics-based approach based on the Navier-Stokes equations of fluid motion, such that fluid motion can be predicted, and then a particle trajectory can be calculated using that predicted fluid motion. So if you see the figure on the right, this is an example of a patient-specific realistic nasal cavity model where particle deposition was predicted. Such a model can predict the influence of particle size distribution, spray angle, and spray velocity, and consider intersubject variability by considering a variety of patient-specific models. Also, a PK profile can be predicted if combined with a PBPK model. Considering PBPK modeling for nasal drug products, this type of modeling approach uses a compartmental model, such as the one you see on the left, which is decomposed into several regions of the nasal cavity, including respiratory and olfactory regions, and then these regions are subdivided into mucus, epithelial, and basal cell layers. These models require regional deposition inputs that can come from a variety of sources, including in vivo, in vitro, or in silico, such as the CFD model that I spoke about previously. These models predict local and systemic PK by simultaneously considering dissolution, absorption through the nasal tissue, metabolism in nasal tissue, and integration with a systemic model. The results are typically validated using in vivo systemic or tissue PK data, although tissue PK data are often not available. Next, I'd like to walk you through a case study that includes CFD and PBPK modeling with adult and pediatric subjects considering nasal models. Adult and pediatric nasal models were created as part of a pair of contracts that were awarded to Virginia Commonwealth University, or VCU. The principal investigator was Lalik Elshahi. The first contract was awarded for development of adult models, and the second contract was awarded for pediatric models. That second contract is still active. The goal of these two contracts was to develop two sets of in vitro models for adult and pediatric subjects, where 3 models were developed for each to represent low, medium, and high deposition. The goal was then to consider intersubject variability for nasally inhaled corticosteroids. Once these models were developed, the final goal was then to examine relationships of in vitro metrics of spray properties with regional deposition. In the figure on the left, you can see an example of an experimental setup that was used to measure deposition in these in vitro nasal models. Alongside in vitro nasal model development, for these contracts, a CFD model was developed. This model was based on what was called an average adult nasal model. To develop the 3 nasal models for adult subjects, there was a pool of 20 patient-specific models that were originally created, and then each of these was subdivided into 2 sides, so there were 40 nasal cavities at the end. The average adult nasal model was considered to be somewhere in the middle of those models. So this was selected before the final low, medium, and high deposition models were selected. This model is decomposed into a computational mesh. This is a required step for a CFD model. If you look in the figure on the right, you can see the cross-section that shows smaller different elements within this nasal cavity model. And what that means for CFD is that CFD solves the fluid equations of motion for each individual element and then combines all of these to form a prediction for the entire domain. So this is a feature of CFD where you need to determine what is your best mesh for coming up with your computation. There were 2 methods that were used to couple fluid and particle motion, and the results were compared to in vitro data. For validating the CFD model, as I mentioned, there were 2 ways of considering the coupling between the airflow and the particle motion. So the first way is called one-way coupling, where you only consider the effect of airflow and particle motion. The second is called two-way coupling, which is a bit more computationally expensive, which also includes the effect of particle motion on airflow. So if you have a particularly large spray, these particles are going to have enough of an effect on the surrounding airflow to also change the airflow around them. Predictions were made for fluticasone furoate nasal spray and fluticasone propionate nasal spray as compared with in vitro data. The top 2 rows show the CFD predictions. So the top one is quasi two-way coupling. So as I mentioned, two-way coupling is rather computationally expensive, so a new method was developed by VCU that can still consider the effect of two-way coupling, but is a bit more computationally efficient. The bottom two rows consider the relative error for these two approaches as compared with in vitro data. And the results here are presented as percentages of the initial dose that was delivered to the system. As you can see, the results show that the quasi-two-way coupling provided better predictions when thinking about the relative error. VCU also has developed models for spray wall interaction in post-deposition liquid motion. What this is when a droplet deposits on a surface, there can be splashing or bouncing or any other kinds of interactions with the surface. And once the droplet is on the surface, it can then spread according to the surrounding airflow. These models were developed to improve the accuracy of predictions and have been used in some cases where it has been shown that some of the time it does improve the predictions. The CFD model was then used to conduct a parameter sensitivity analysis. The variables that were considered were spray cone angle, spray average velocity, plume ovality, and particle size distribution. You can see the ranges of these variables on the left, where spray cone angle was varied between 15 and 55 degrees. Spray average velocity was varied between 8.4 and 20.4 meters per second, and plume ovality was varied between 0.5 and 2. Particle size distribution was defined using a polynomial equation, where n and d bar were varied as you see below. Corticosteroid furoate nasal spray was the drug product of interest. And the medium and high deposition adult models were considered with 3 different insertion conditions. So this was after the low, medium, and high deposition models had been identified. The baseline condition included spray injection velocity of 14.4 meters per second and an average cone angle of 35 degrees and ovality of 1. So when one parameter was varied, the others were held constant. Looking at the results for spray cone angle, you can see, especially in the bar graph on the bottom left, that anterior deposition and posterior deposition were predicted, where anterior deposition was in the nasal vestibule, and then posterior deposition was all the deposition past the nasal vestibule. And as you can see, going from 15 to 55 degrees, posterior deposition was varied by quite a bit, between about 50% for 55 degrees and about 100% for 15 degrees. Overall, for the parameter sensitivity analysis, spray cone angle showed the largest influence on posterior nasal cavity drug delivery. Plume ovality also showed some influence that was dependent on insertion condition. Spray velocity showed very little effect, which provided some assurance that the recommendations that do not include spray velocity were correct. Particle size distribution shows some effect, depending on which of the two parameters were adjusted. And finally, there was the trends between the two different nasal models were similar, although there was some slight differences according to particle size distribution. There was a PBPK model that was also developed alongside the CFD model. This is called the Dissolution, Absorption, and Clearance, or DAC CFD model. It's a quasi-two-dimensional model with varying width based on nasal cavity perimeter, originally based on a model developed by Alex Rigg and coworkers. And it's coupled with a 2-compartment PK model and a simplified gastrointestinal or GI tract model. So as you can see on the right, the way this works is that you take a 3-dimensional prediction of droplet deposition, translate those particles to the quasi-two-dimensional model. These particles are then cleared via mucociliary clearance, where they can either be swallowed into the GI tract and then either excreted or absorbed into the central compartment, or they may dissolve, and then the dissolved drug can be absorbed through the epithelium and then into the central compartment, where it is either cleared or distributed to the peripheral compartment. A little bit more on the PBPK model development. These predictions were made with a 3D CFD and DAC models using the adult medium deposition nasal model. The previous model had used one model based on both nasal cavities, but here the new model was separated into a geometry with 1 or 2 single nasal cavity models. The absorption was based on nasal permeability, which was optimized to provide the best match with available vivo PK data for triamcinolone acetonide nasal spray. The previous model developed by Alex Rigg and coworkers was based on passive diffusion, so this was a change from that model. Distribution and clearance parameters were based on available intravenous PK data for triamcinolone acetonide, and deposition predictions were validated in a similar manner as described previously. Looking at the results, plasma concentration predictions were obtained for 110 micrograms, where there was 1 spray in each nostril, and 220 micrograms, where there were 2 sprays in each nostril, and the doses were compared with available in vivo PK data. For the 220 microgram dose, there were several approaches with 1 cavity that included 4 sprays, 2 sprays with double post-dissolution dose, and 2 sprays with double absorbed dose. And looking at the results, the 2-cavity model was generally superior to the 1-cavity model, and some of the 1-cavity model approaches were also acceptable. So you've seen me talk about CFD modeling, PBPK modeling. Perhaps you're interested in using these type of modeling approaches if you're developing a nasal drug product, a generic nasal drug product. So how might you use this type of modeling? Well, first you'd have to develop the capability. You can do this internally, preferably by hiring someone with previous experience, or you can look through a contract research organization or through academia, where you can find these types of organizations through either the literature, going through a literature search, or you can look through the internet to see if anybody's advertising these types of services. Strongly recommend that if you use a model that you build it early in development. This will give you the most benefit from using your model. And as mentioned before, you can use this model to explore the impact of formulation or device changes. So if you need to make a formulation or device change in your development, such a model can be useful for either understanding the impact of these changes on PK, or you could even alter these models to replicate certain in vitro studies, such as spray pattern, plume geometry. And you could even do predictions before doing in vitro testing. So coming to the end of my presentation, I have a couple of challenge questions for you. Which in vitro metric was predicted to have the greatest influence on posterior nasal cavity drug delivery? Spray average velocity, plume ovality, spray cone angle, or particle size distribution? Hopefully you chose spray cone angle. Which of the following statements is not true? If option 2 is selected for demonstrating a bioequivalence, modeling can predict impact of formulation and device changes on pharmacokinetics predictions. Computational fluid dynamics models may only be validated using in vivo deposition data. It is most useful to build a computational model to support product development early in the process, rather than the middle or the end of the process. I'm hoping you chose B. Computational fluid dynamics models may also be validated using in vitro deposition data. I have a few conclusions for you. CFD and PBPK modeling of nasal suspension drug products may be used to understand the impact of formulation and device changes on systemic PK or in vitro metrics. A combination of in vitro and/or in vivo data may be used to validate CFD and PBPK models of nasal suspension drug products. Model predictions suggested that spray cone angle has the largest influence on posterior nasal cavity drug delivery among the considered in vitro metrics. So I have a call to action for you. Consider developing the capability to use CFD and PBPK modeling to support development of your generic nasal suspension drug product. I have a number of acknowledgements from coworkers in my office and from other offices throughout FDA, and from VCU, especially calling out Andrew Babiskin, who has helped me with many of these research projects, Eleutheria Sokolosa, who is our current team leader, Stephen Chofsky, who has helped with many of the modeling for nasal drug products, our office leadership, especially including Liang Zhao, Lucy Feng, Lei Zhang, Robert Leinberger, and also from VCU, the researchers there were worth longest: Arun Kalangil, Rabijit Duda, Vale Shahi, and Michael Hindle. So finally, I'd like to close with a reflection on the value of generic drug products. This was from one of my OGD coworkers, where they said that the first time they were able to buy their son's inhaler as a generic and saw just how much the price had dropped, it was really impactful for them. So it puts into perspective why it is important to put more generic drug products on the U.S. market. I appreciate your attention, and I'll be ready for your questions in the Q&A.
Good afternoon, everyone. This is Elizabeth Bielski. In this presentation, my colleague Nathan Reed and I will be presenting on loxapine inhalation powder, the OTR research conducted to inform the PSG recommendations. The learning objectives for this presentation are as follows. Be familiar with the main features of the drug product Adasuve inhalation powder. Understand and describe the research efforts used to analyze the aerosol properties of Adasuve, and understand and describe the recommendations within the product-specific guidance, or PSG, on loxapine inhalation powder. The reference listed drug being described here is Adasuve, which contains 10 milligrams of loxapine as an inhalation powder. It is considered a single-use drug-device combination product. It is an atypical antipsychotic indicated for the acute treatment of agitation associated with schizophrenia or bipolar I disorder in adults. The drug product must be administered by a healthcare professional in a certified healthcare setting only, and is only available under a restricted program known as Adasuve REMS. 10 milligrams is taken by oral inhalation using an inhaler, which is pictured here on the right-hand side of the slide. Only a single dose should be administered within a 24-hour period. Prior to administration, patients should be screened for pulmonary disease and for any other respiratory abnormalities. Adasuve is administered by first opening the pouch that contains the inhaler. Next, the healthcare professional should pull the tab at the bottom of the inhaler to activate the device. Before pulling the tab, the indicator light should be off, indicating the inhaler is not ready for use. After pulling the tab, the green light indicator should turn on to indicate the inhaler is activated and ready for use. This may include flashing, a clicking noise, and the inhaler may become warm. These are all normal signs the inhaler is being activated. Once the inhaler is activated, the patient should be instructed to exhale fully to empty the lungs. After exhalation, the patient should put the mouthpiece of the inhaler between their lips and inhale with a steady, deep breath. Lastly, the patient should be instructed to hold their breath as long as possible, up to 10 seconds. At this stage, the green light should turn off and to indicate that the dose has been successfully delivered. Let's take a closer look at the delivery mechanism within the device. Before inhalation, a thin film of excipient-free loxapine coats the surface of the heated substrate. Upon activation of the inhaler and oral inhalation through the device, the controlled rapid heating of a thin film of excipient-free loxapine is initiated to form a thermally generated drug vapor. The vapor quickly condenses into aerosol powder particles that are dispersed into the airstream created by inhalation through the mouthpiece. Overall, Adasuve provides a rapid systemic delivery of an aerosol of pure loxapine that is quickly absorbed through the lungs and into the bloodstream.
To help inform the PSG recommendations for loxapine inhalation powder, the Office of Testing and Research conducted a study to better understand Adasuve and its critical quality attributes. Adasuve has a unique thermal aerosolization process that differs from other inhalation drug products, including dry powder inhalers. Dry powder inhalers often aerosolize a blended API and excipient powder, which then undergoes a deagglomeration process. With this in mind, we explored the impact of inspiratory flow on Adasuve performance and used laser diffraction as an alternative method for measuring particle size distribution. Since Adasuve is excipient-free, the laser diffraction data can be more easily compared to aerodynamic particle size distribution data. Aerodynamic particle size distribution is a critical quality attribute for meter dose inhalers and dry powder inhalers, and is often used to predict the amount of drug deposited within the lungs in different regions based on their aerodynamic size. One of the most common instruments for characterizing aerodynamic particle size distribution is the next generation impactor, shown on the left. This impactor is often referred to as the NGI. With this cascade impactor, coupled with chemical analysis, the aerodynamic particle size distribution, mass median aerodynamic diameter, geometric standard deviation, and fine particle mass can be measured. This data is readily available for ADASUV and shows an interesting trend. When the actuation flow rate is increased, we see that the particle size of loxapine decreases. In this study, we were interested in exploring laser diffraction as an alternative method to measuring ADASUV particle size distribution. ADASUV is a great candidate for laser diffraction analysis because it's excipient-free, so the volume-weighted size distribution data from the laser diffraction system should correlate with the mass-weighted size distribution data from a cascade impactor. Our methods tested flow rates of 15, 28.3, 60, and 90 liters per minute to determine the effects of inspiratory flow on particle size distribution. The instrument configuration used in this study provided size distribution data for particles between 500 nanometers and 175 micrometers in diameter. It's important to note that laser diffraction does not provide chemical or morphological data. However, supporting experiments were used to confirm the assumptions made to generate particle size distribution data using the laser diffraction system. To better understand the capabilities of the ADASUV inhaler, we measured the minimum flow rate needed to trigger loxapine aerosolization. Our attempts to actuate the inhalers at 15 to 18 liters per minute failed, and we found that the average minimum actuation flow rate was around 20 liters per minute. Since the ADASUV inhaler utilizes a mechanical switch rather than a solid-state sensor to determine when to actuate the device, it's likely that small deviations in device assembly and the mechanical properties of the flow switch cantilever may result in variability in the minimum flow rate required for actuation. It is worth noting that data has been previously reported for 15 liter per minute actuations. For 28.3, 60, and 90 liter per minute actuations, our laser diffraction data showed that particle size was flow rate dependent, with higher flow rates generating smaller particles. We can see in the table on the bottom left-hand corner of the slide that the DV50, or median particle diameter by volume, decreased from around 2.5 to 2 to 1.5 microns as flow rate increased from 28.3 to 60 to 90 liters per minute. We were also interested in the morphology of the particles that we characterized with laser diffraction. So using optical microscopy, we were able to show that the particles were spherical. When we combine our laser diffraction data and calculate the mass median aerodynamic diameter at different flow rates, we see a similar trend compared to the NGI data. As flow rate increases, the mass median aerodynamic diameter decreases. While this result is promising, our study has highlighted many of the limitations and assumptions necessary to compare laser diffraction data to cascade impactor data. Further experimentation, method validation, and supporting data may be needed to support the suitability of laser diffraction to other currently recommended studies characterizing aerosol performance.
When considering the establishment of bioequivalence for generic products referencing ADASUV in the PSG, the following were taken into consideration. ADASUV is a complex drug-device combination product. Loxapine has a systemic site of action. The novelty of the delivery device platform used in ADASUV. The substantial variability of loxapine plasma concentrations from ADASUV. And the outcomes of OTR's research, of which Nathan has just described. Based on these considerations, the in vitro and in vivo bioequivalence studies are recommended in the PSG. A measurement of in vitro product performance through sensitive and accurate measures may support the establishment of equivalence in PK performance and overall bioequivalence between generic loxapine inhalation powder drug products referencing ADASUV. First, I'll go over the in vitro BE studies recommended for this PSG. Aerodynamic particle size distribution, or APSD, is recommended to help predict the amount of drug deposited within the lungs in different regions based on aerodynamic size to help characterize aerosol performance. The recommended apparatus is the Anderson cascade impactor, the next generation impactor, or another appropriate method. The flow rates recommended are 20, 40, and 80 liters per minute, which were supported by RTR's investigation on the initial flow rate testing described by Nathan previously, along with seeing a significant decrease in the mass median aerodynamic diameters, or MMADs, with increasing flow rates within the range of flow rates thought possible for patients to achieve during inhalation. Such change in MMAD values is believed to be due to the increase in airflow causing a decrease in drug particle concentration and subsequent decrease in potential for drug particle collisions that can create larger nucleated drug particle sizes. As such, these three flow rates that cover this range are recommended. The bioequivalence assessment is based on population bioequivalence, or PBE, analysis of the impactor size mass. The ISM, or impactor size mass, is the sum of drug mass on all stages of the cascade impactor plus the terminal filter, but excluding that top cascade impaction stage because of its lack of specified upper cutoff size limit. Supportive evidence is also requested by providing the cascade profiles representing drug deposition on the individual cascade impaction stages, MMAD values, geometric standard deviation values, and fine particle mass values. The other in vitro BE test recommended is the single actuation content, or SAC, which characterizes the amount of drug exiting the device per actuation. The recommended apparatus is the US Pharmacopoeia, or USP apparatus B, or other appropriate apparatus. The flow rates recommended are the same as those recommended for APSD, 20, 40, and 80 liters per minute. Within this range, the amount of residual drug on the device was seen to decrease with increasing flow, allowing for more drug to be deposited within the airways. Equivalence is based on PBE analysis of SAC. The in vivo BE study recommended is the pharmacokinetic, or PK-BE study to assess systemic bioavailability. The design is a single-dose two-way crossover study. The 10 milligram strength is recommended using a single inhalation dose. Healthy males and non-pregnant females are the recommended subjects to be used. Since this product is under restricted use, the PK-BE study should be designed to follow the REMS with elements to assure safe use. The analyte to measure is loxapine in plasma. Equivalence is based on area under the curve from 0 to 30 minutes, or AUC from 0 to 30 minutes, and AUC from 0 to infinity. The 90% confidence intervals for the geometric mean test versus reference ratios, or T versus R ratios, of the AUC from 0 to 30 minutes and AUC from 0 to infinity should fall within the limits of the 80% to 125%. Due to the early onset of action and high variability, Cmax, Tmax, and partial AUCs from 10 minutes, 30 minutes, and 2 hours is recommended to be characterized as supportive data to assess the onset of loxapine. Additional device recommendations are also included in the PSG for loxapine inhalation powder to support the user interface assessment. The following characteristics of the RLD product should be considered when designing the generic product: the passive breath-actuated single-dose format of the RLD device, and device resistance of the RLD product, as they are important to the inhalation steps and the drug delivery process. The device activation system, or tab, that is pulled out of the device to activate the inhaler, and the indicator, or green indicator light, that the device uses to let the user know that the device is activated.
To summarize our talk today, ADASUV inhalation powder is a single-use drug-device combination product for the acute treatment of agitation associated with schizophrenia and bipolar I disorder in adults. Adasuve provides rapid systemic delivery of a thermally generated aerosol of pure loxapine from the staccato device, which is quickly absorbed through the lungs and into the bloodstream. The results from OTR's research study on Adasuve provides insights into the use of Adasuve, the impact of inspiratory flow rate on Adasuve performance, and the benefits and limitations of using laser diffraction characterization of particle size distribution. Ultimately, it was determined that further experimentation and validation are required to determine the suitability of laser diffraction as an alternative or orthogonal method to other currently recommended studies like aerodynamic particle size distribution using cascade impactors. Based on OGD's evaluation of the RLD, including OTR's characterization studies and supporting data, the PSG for loxapine inhalation powder was developed to include the following BE studies: aerodynamic particle size distribution, single actuation content, and a PK study with equivalence data for area under the curve from 0 to 30 minutes and area under the curve from 0 to infinity, as well as supporting data for Cmax, Tmax, and partial area under the curve at 10 minutes, 30 minutes, and 2 hours to assess the onset of loxapine. We would like to thank the following individuals for their help in this work. The references and resources used in this presentation are listed on this slide. Challenge question 1. What are the main bioequivalence BE studies included in the product-specific guidance for loxapine inhalation powder? A. Single actuation content, aerodynamic particle size distribution, and drug particle size distribution by laser diffraction. B. Single actuation content, drug particle size distribution by laser diffraction, and pharmacokinetic BE study. C. Single actuation content and aerodynamic particle size distribution, or a pharmacokinetic BE study. D. Single actuation content, aerodynamic particle size distribution, and a pharmacokinetic BE study. The answer is D. Single actuation content, aerodynamic particle size distribution, and a pharmacokinetic BE study. Challenge question 2. What is the effect of inhalation flow rate on Adasuve inhalation powder particle size? A. No flow rate dependence. B. Higher flow rates generate smaller particles. C. Higher flow rates generate larger particles. D. 60 liter per minute inhalations generate the largest particles. The answer is B. Higher flow rates generate smaller particles. Elizabeth and I would like to thank you for your attention and the opportunity to present our work today. We look forward to your questions, but feel free to reach out using our contact information below.
Thank you all for the great presentations. As a reminder, if you haven't had a chance to enter your questions into the Q&A chat pod, please do so now. Moving into our Q&A panel, we welcome, in addition to our presenters, Dr. Vipra Kundor, pharmacologist, Division of Bioequivalence 1, Office of Bioequivalence and OGD, Dr. Mai Tu, chemist, and the Liquid-Based Branch 4, Division of Liquid-Based Products 2, Office of Lifecycle Drug Products and OPQ, and welcome back Dr. Ahmed Zidan. Looks like we have some questions coming in right now. The first group of questions will be directed to Dr. Susan Bach. And here is the first question. What are meaningful differences that can be made in the formulation to show a dissolution method is discriminatory?
Hey, thank you for the question. For a dissolution method to be considered discriminatory, the method should show differences in the dissolution profiles of the formulations with meaningful differences. So for meaningful differences that can be made to the formulation, it should show it should be changes in the critical quality attributes, such as particle size of the active pharmaceutical ingredient or concentrations of the inactive ingredients. So for formulations intentionally manufactured with variations of these variables, for example, 10 to 20% change to these variables, it may be used to demonstrate discriminatory ability. If the dissolution method does not detect any difference when using a different particle size or changes in the inactive ingredients, then depending on the variables studied, then the dissolution method, development method may need to be revisited. Other attributes, I should mention, may also be used, but justifications for the method should be provided in the submitted ANDA. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Bach. And here is the next question. By providing the Transwell system as an example apparatus available for dissolution studies, does the agency expect that dissolution methods be biorelevant?
Thank you for the question. The Transwell system is provided as an example apparatus for dissolution studies since it was shown in the GDUFA-funded research to provide comparable results to the USP apparatuses also listed. As mentioned in my talk earlier, one goal of the dissolution test is to develop a method that can show changes in critical quality attributes of the drug product. So potential applicants should consider this aspect when considering which dissolution apparatus to use for development of the dissolution studies. Thank you.
Thank you for responding to that question. We do have one more question for Dr. Bach, and here's the question. While firms can provide justifications for dissolution media selection, does the agency plan to put out a guidance that lists dissolution media suitable for BE studies?
Thank you for this question. So at this time, the agency does not have any plans to publish a guidance that lists suitable dissolution media for nasal spray drug products. And as mentioned in the talk, dissolution media selection should be guided by solubility investigations and optimized to be discriminatory. And justification with method development and validation for selection of the dissolution media should be provided with the submitted ANDA. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. We've got a few questions that came in for Dr. Ross Walenga. And here is the first question. For nasal products, is it okay to provide in silico data along with in vitro data? And do we still need to perform in vivo BE studies if we find a favorable result through in vitro and in silico studies?
Thank you for the question, Ray. Yes, it can be acceptable to include an in silico study in an application. As for would it remove the need to conduct an in vivo BE study, so I'm assuming that the question would be about option 2, if an in vivo study was required. Using option 2, it could be possible to waive an in vivo study using in vitro and in silico studies, but it would be subject to the credibility of the in silico model and the acceptability of the in vitro studies for doing so. So if you were to do something like this, since this is not in our guidance, I would suggest that you meet with FDA through the pre-ANDA meeting process. Thank you.
Thank you for responding to that question, Dr. Merlingo. We've got a couple more questions, and here is the next question. How do we determine how much weight to give the different in vitro measurements, such as spray velocity, for nasal sprays, and Can you also please discuss the effects of inhalation coordination with the spray actuation of nasal sprays on nasal spray parameters?
Thank you for the question, Ray. As for the weight of the different in vitro measurements, so I had mentioned in my presentation that spray velocity did not show much impact. And as you have surely noticed from Susan's presentation, spray velocity is not one of the recommended studies. So in that case, you can see that that particular measurement does not have much weight. As for the weight of the other studies, it could be possible to use an in silico study to show what biorelevant limits might be for these different in vitro studies. And if well supported, this could be something that FDA would entertain. As for the second half of the question, regarding inhalation coordination with spray actuation, I think it's fairly well known that if the inhalation coordination is different from subject to subject, that this can cause a lot of variability. In fact, there was a publication that was just released from one of our contractors, Applied Research Associates, and they showed that just by moving the spray bottle a few millimeters one way or the other, that it would greatly change the deposition characteristics. So I think the variability is already understood going into these recommendations.
Thank you for responding to that question. We have one more question for Dr. Willinga, and here's the question. How might parameter sensitivity analysis that suggest relationships between in vitro metrics and nasal regional deposition be useful from a regulatory point of view?
To come up with a set of bioequivalence limits that might be different than what are recommended that are demonstrated to be biorelevant. So that in some cases, this could make the limits a bit wider and perhaps make it easier to come up with an approvable product. Thank you.
Sorry about that delay. I just have a microphone connection issues, but thank you for responding to that question, Dr. Willinga. Moving on to one of our next panelists, we do have a few questions that came in for Dr. Elizabeth Bielski, and here's the first question. Sorry about that, lost a little momentary connect. Here's the question for Dr. Bielski. Could laser diffraction be proposed in lieu of PSD as an in vitro test to demonstrate BE of generic loxapine product referencing adhesive?
Thank you very much for that question. Laser diffraction to measure particle size distribution could possibly be proposed in lieu of the APSD method currently proposed in the PSG. However, that is considered an alternative BE approach to what's currently proposed. Thus, the agency is open to reviewing alternative approaches to those that are proposed in this PSG, and we'd be happy to discuss those proposals through our product and pre-ANDA product development meeting pathway or through a controlled correspondence. So we just ask that if you do propose such a situation, just provide enough scientific rationale and justifications within your proposal, and then along with any of your specific questions, and we can have that discussion. Thank you.
Thank you for responding to that question. We've got a couple more questions for Dr. Bielski, and here's the next question. The proposed device used with the inhalation powder does not need to be previously cleared or approved by CDRH, is that right? Or is it just acceptable to include the device comparison in the ANDA and submit it to CDER?
That's a great question. Thank you for that question. As noted, the device in a proposed generic does not necessarily need to be previously cleared by CDRH, so it can be included as part of your ANDA submission. So what's recommended in the PSG is what we would consider from a bioequivalence perspective, or what device considerations that person may need to think of. But remember that when you are submitting your data, that you also have to consider the quality-related considerations. So your product performance with the device, as well as other device characterizations would be expected. So don't forget to look at those aspects of that device as well. Thank you.
Thank you, Dr. Bilski, for responding to that question. We do have one more question for you, and here is the question. In terms of a device resistance, is this referring to the tab to activate the device, or the inspiratory force needed to inhale the dry powder?
Thank you for that question. I would say that when we wrote device resistance as part of the PSG considerations, we're really talking about that inspiratory force needed to inhale the dry powder, because that plays a role in how the in vitro performance will play, as well as a user interface, how the patients will feel the force, or the force needed to activate the device. But that doesn't mean you shouldn't think about the tab as well as the force on the tab that would be needed. So when you're comparing your generic device to Adasuve itself, you should also consider the force it would take to activate that. But that type of information or data should be included in your user interface assessment, and that user interface assessment can be submitted from what we call a comparative analyses. So that also should be considered. Thank you very much.
Thank you for responding to the group of questions. Moving on to our next panelist, I've got a few questions that came in for Dr. Nathan Reed. Here's the first question for Dr. Reed. Have you explored biorelevant inhalation patterns to mimic the deposition process in vivo?
Thank you for the question. I think that this is a really interesting research idea at this point. It's not something that we've directly measured, but since this device has a flow rate dependence on the particle size distribution emitted from it, the biorelevance of the inspiratory flow could be critical to the deposition and dissolution of the drug product. So yeah, again, something that we haven't done currently, but it's something that we are capable of doing and are interested in doing. Thank you.
Thank you for responding to that question. We do have another question for Dr. Reed, and here's the question. What are the effects of particle size and deposition on dissolution kinetics?
Yeah, thanks again for the question. I think it's another interesting question, particularly for this device, since inspiratory flow is critical for determining the particle size. Deposition behavior, the aerodynamic behavior of these particles in the lungs will be affected. So the smaller the particle, the larger this surface area to volume ratio, we would expect that dissolution would change as well. So a higher inspiratory flow would result in smaller particles, a higher surface to volume ratio for those particles, we would have different dissolution kinetics. We would expect them to dissolve faster. There is some work going on in the Office of Testing and Research on these problems right now. Yeah, it's being explored. Thank you.
Thank you for responding to those questions. Moving on to our next panelist, we've got a few questions that came in for Dr. Vipra Kundor. And here's the first question for Dr. Kundor. Are there any considerations for selecting an appropriate orthogonal method for MDRS?
Thank you for the question. A major limitation of the MDRS method is its inability to measure the particle size below 1 micrometers. Therefore, an appropriate orthogonal method can be used in conjunction with the MDRS method. This selected method should attempt to distinguish between the API and from the excipient particles, and also the method should demonstrate that it is sufficient for evaluating bioequivalence in the API particle size distributions of both the test and the reference products. Thank you.
Thank you for responding to that question. We do have our next question is addressed to Dr. Kundor and also Dr. Mai too. And here's the question. What are the common deficiencies observed in morphological-based characterization of particle size and shape?
Thank you for the question. Some of the common deficiencies observed in ANDAs using the MDRS method were lack of adequate sample preparation and image size characterization studies, inadequate method validation data, inappropriate sample size, and the pivotal MDRS study was conducted without using the same parameters validated in the method validation, and also the orthogonal method was not supportive to the MDRS method. Thank you.
Yeah, thank you for the questions. I'd like to add to Dr. Kundor on this topic. The common deficiency that we have seen is sample size, as was said before, and the number of particles samples analyzed should be large enough to represent the entire samples. So example, especially for microscopy method, and this is in general, the different viewing area may be captured because the number of particles are determined based on the number of micrographs that are captured. So make sure that the sample size are adequate and good quality for the microscopy images, good resolution for that as well, for us to view with scale bar for accurate interpretation of the data. Another factor that I've seen is that particle size may need to be controlled in the finished product testing, and this depends on the product. So if you think this can, if there are potentially change in particle size, then this should be evaluated during product development to see if any trending of data, and if there is trending, then it would be really helpful to provide details interpretation of the data, and it could be due to many things. But those are the common things that we saw. Thank you.
Thank you, Dr. Mai Tu. And Dr. Tu, we do have another question directly just for you. And here's the question. From a product quality perspective, what are the key considerations for drug particle size distribution characteristics during product development?
Thank you, Ray, again for the questions. From product quality perspective, particle size distribution is very critical because it impacts the drug product dissolution behavior, resuspendability or redispersibility of the product, any sedimentation rate. So again, it would really depend on the nature of the product. The information that we would look for is first the method. The method should be demonstrated to be suitable for use in the test product, and it should have adequate sensitivity, especially if you consider that if you want to demonstrate that it's similar to the reference listed drug or the reference standards, then it should be sensitive enough to detect any difference compared to the reference listed drugs. And the product should be filled in the final container closure to be used for testing. And for, when possible, consider any specific instruction in labeling when you prepare the sample so that it will be a good representative of the products, especially for the combination products with the device. And in terms of data, provide the particle size data, 3-tier distribution of the particle size if applicable, and then also any relevant profile of the particle size distribution. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Ahmed Zidan. And here is the first question for Dr. Zidan. How may the device parameters be included in the model, and what are the limitations and challenges that need to be overcome?
Thank you for the question. So that here controls particle size distribution from one point, as well as the site of the particles may be deposited. So all the device parameter may be a primary factor that's affecting the performance. However, to include the effects of the device parameter in a model, biorelevant methods may be required. What's happening in the respiratory tract, and as Dr. Wolenka have shown in his presentation, some mechanistic model and computational models that mimic the administration site, for example, the nasal cavity, even to mimic the lung site, may give more impact to capture the effect of the device parameters. And is submitting a change to the device, or if a generic product have a different device than RLDs. In this case, the device parameter, for example, the valve, the actuator, the spray nozzle, that's affecting the spray pattern and affecting the aerodynamic characteristics of the product as it's leaving the device. All of this may affect the performance. So the short answer for this is determining what is the critical device parameter that's affecting the critical input for the method, for the model to predict the is the key here. So some DOE and screening study may be done during the development phase to determine this critical parameter of the device.
Thank you for responding to that question. We do have one more question for Dr. Zidan. And here's the question. What do you think about the biorelevance of the dissolution parameters and how it may affect the demonstration of BE?
Yeah, this is another nice question also. The biorelevance of the dissolution parameter, the ideal situation to have to predict the bioequivalence in vitro. How mimicking the dissolution, I say condition, it may be hard to have it in the lab, and this is reasons. For example, the dosing of the drug product during dissolution may affect. So I will just talk about one of the ceremony or one of the inhalation that Dr. Elizabeth and Nathan talked about. It's one of the thermally generated inhalers, and for this one, the drug is as it is heated inside the device, and the thermal behavior, the vaporization pattern, as well as the deposition of this all affect the deposition rate. So we may need to consider all of this aerodynamic interaction process between the particles when dosing the drug for dissolution testing. So this is the first part of the solution, how the drug is deposited and how we are loading it. Second part of biorelevance and dissolution is the drug clearance from the dosing compartment. So the ideal situation to have the same clearance at the site of application, and the best is to mimic the blood flow and the from the mucosal membrane. However, in dissolution, a lot of factors here affecting this. For example, the diffusion of the rate and the sink condition in the receiver compartment, and what is the biorelevant media to be used. Other than this, dynamic or agitation effect that we are employing in the receiver chamber to make sure that the drug is removed from the diffusion layer has to be considered in setting a biorelevant dissolution condition. And of course, at the end of the day, data that is developed or generated from this one, it is in vitro dissolution data. So making sure that the method is biopredictive, need to have some sort of physiologically based pharmacokinetic modeling, and this is the one to ensure that this can generate a biopredictive or biorelevant dissolution data, and this is from the biorelevance perspective, but the discriminating of this biorelevant or biopredictive method is also a key for consistency and unit-to-unit variability. So all of this has to be considered when we think about the biorelevance of the dissolution method. So yeah, thank you.
Thank you for responding to that question. Moving back up to the beginning of our panel, we've got a few questions that came in for Dr. Susan Bach. And here's the first question for Dr. Bach. Are in vitro BE studies the same for nasal spray suspensions as well as nasal spray solutions?
Thank you for the question. So the in vitro BE studies recommended for the nasal spray suspensions, as discussed in my talk earlier, are the same as those recommended for nasal spray solutions. But depending on the drug product, the nasal spray solution products may have fewer recommended studies. I would suggest to refer to the individual PSGs provided on the FDA webpage. Thank you.
Thank you for responding to that question. We do have another question for Dr. Bach. And here's the question. If a nasal product is non-Q1, Q2, what BE studies would be part of the option 2 BE studies?
Thanks again for the question. So for non-Q1, Q2 nasal spray suspension products, the PSG recommends in vitro and in vivo studies. So these include the 6 in vitro studies that I listed earlier, which includes single actuation content, droplet size distribution by laser diffraction, drug and small particles or droplets, spray pattern, plume geometry, and then priming and repriming. And it also includes the 2 in vivo studies, which are a comparative PK study and a comparative clinical endpoint study. And if you have any other questions, I would suggest you again to refer to the individual PSGs provided on the FDA webpage. Thank you.
Thank you for responding to those questions. Moving to our next panelist, we do have a couple of questions that came in for Dr. Ross Wallinga, and we'll try to get a couple of those in. And here's the first question. What is the value of validating nasal deposition predictions with in vivo data as opposed to in vitro data?
Thank you for the question, Ray. In vivo data can capture processes that the in vitro models might not be able to capture, such as moisture in the nasal cavity or mucociliary clearance or things like that. So even though in vivo data can be somewhat limited by usually being two-dimensional, in most cases they're preferable.
Thank you for responding to that question. And we have run out of time just about in this panel. Huge thanks to our speakers and panelists for answering numerous questions that came in from our audience. We'll now enter into our break time until 2:40 p.m. Eastern Time. Enjoy your break. Welcome back from the break. We're on the home stretch now. Moving into session 3 on noteworthy guidances for injectable products. Our presenters include Dr. Xingnan Zheng, Staff Fellow, Division of Therapeutic Performance 1, Office of Research and Standards at OGD, and Dr. Eric Pang, Senior Chemist, Division of Therapeutic Performance 1 in the Office of Research and Standards at OGD. Please join me to welcome our first presenter, Dr. Zhang.
Thank you for the introduction. Today, I will share the considerations for the in vitro BE approach of injectable drug substance suspension products using medroxyprogesterone acetate as a case study. By the end of my talk, you will be able to understand how the in vitro BE approach developed for the injectable drug substance suspension, describe the product-specific guidance on medroxyprogesterone acetate, recognize the common deficiencies observed for the in vitro BE study, as well as the data and the information be submitted for the in vitro BE study in ANDA submission. So, what is injectable drug substance suspension? In injectable drug substance suspension, the particles are generally made up only of micronized or nano-sized drug substance. The drug substance particles are the only insoluble component in the formulation or reconstituted formulation. For other excipients in the formulation, they are all soluble in the solution phase. The drug release for drug substance suspension does not rely on the release controlling excipient, for example, PLJ copolymer. The drug substance suspension has a simple drug release mechanism, then suspension products contain release controlling excipient. The dissolution rate is generally determined by particle size of drug substance and the solubility of the drug substance, as described by Noyes-Whitney equation shown here. When demonstrating bioequivalence, the utilized BE study should be sensitive, accurate, and reproducible. For systemically acting injectable drug substance suspension, FDA generally recommend the in vivo BE study with PK endpoints. In addition to in vivo BE studies, FDA has also recommended in vitro BE study for some injectable drug substance suspension products. Next, I will walk you through how the in vitro BE approach developed using a case study of PSG on medroxyprogesterone acetate. First, let us check the product-specific guidance PSG on medroxyprogesterone acetate. The PSG recommends 2 options to demonstrate BE. Option 1 is in vitro studies, and option 2 is in vivo BE study with PK endpoint. You may choose in vivo or in vitro approach to demonstrate bioequivalence between the test and the reference products. Note per Regulation 21 CFR 314.94A9, drug product for parenteral use must contain the same inactive ingredients in the same concentration as the ILD, with allowable difference with regard to preservative buffer or antioxidant, provided the difference do not affect the safety and efficacy of the proposed product. Following in vitro BE study, our general expectation is that the proposed test product is Q1, Q2 the same as the ILD to ensure the identical formulation components to begin with. We acknowledge the Q1, Q2 sameness alone may not be sufficient to ensure comparable formulation characteristics between test and ILD products, because difference may arise from manufacturing process and/or excipient source. Therefore, based on our understanding, we also recommend a list of comparative physicochemical characterization to be conducted to ensure comparable formulation characteristics. For example, polymorphic form, crystalline shape, and morphology of the API, appearance, pH, osmolarity, specific gravity, sedimentation rate and volume, and viscosity over a range of shear rate of the drug product. When a suspension product is formulated Q1, Q2 the same as an ILD, there could be difference in the arrangement of matter within the dosage form, which may impact drug product performance. For injectable drug substance suspension, it is recognized the particle size can impact drug release. This has been discussed in slide 3, described by Lois-Whitney equation. We also have GDUFA research conducted for understanding the relationship of the product critical quality attributes to the in vivo performance using medroxyprogesterone acetate as a model drug. Formulations with identical composition but different particle size were prepared. In vitro drug release methods with discriminatory power and reproducibility were developed. An in vivo PK study was conducted in rabbit to study the relationship between the particle size and in vivo performance. A level A IVIVC was established using data obtained from USP apparatus 4 with semi-solid adapter method. Therefore, with the improved understanding in drug substance injectable suspension, as well as our internal knowledge obtained from ANDA application data, the in vitro approach was recommended for medroxyprogesterone acetate injectable suspension. Then, let us see how an in vitro approach being executed in an ANDA application. This is a high-level summary of the information and the data to be included. First of all, a composition table that shows the test product is Q1, Q2 the same as the ILD should be provided. The following study should be conducted on the test product comparing to the reference standard, the IS products, comparative physicochemical characterization, BE Study 1, particle size and particle size distribution, BE Study 2, in vitro drug release. For particle size distribution, laser diffraction is generally used. We acknowledge the particle size measurement instrument setup is based on first principle. However, per regulatory perspective, it is expected ANDA application provide data for method development and method validation. The particle size study should also contain the statistical analysis that the particle size distribution should be compared using population bioequivalence based on D50 and span. The applicant should provide no fewer than 10 datasets from 3 different batches of both test and IS products for PBE analysis. If an applicant uses a different statistical method, it is recommended the ANDA applicant provide sufficient justification and enough data to support the proposed statistical method. For the in vitro drug release study, similarly, it should include the method development and validation study to support the utilized in vitro release study is properly developed and validated. The equivalence should be established using a proper statistical method from at least 3 batches of tests and reference standard products. One suggested statistical method is model-independent similarity factor. The common deficiencies in PSD study is that there is no method development and/or validation report. We are highlighting the method development for particle size measurement because the flocculation associated with injectable drug substance suspension. In formulation development for drug substance suspension, stabilizer or viscosity agent may be used to induce flocculation and prevent agglomeration or outward rippling. Flocculation is a process by which small primary particles in suspension collide to form loosely held larger secondary particles called flocculates. The polarized light microscopy here shows what it looks like. The smaller particles are 1 to 2 micron and are primary particles. The larger flocculated secondary particles are around 10 microns. The interconversion between these two types of particles is reversible and highly dependent on the shear condition. As shown in the figure over here, flocculation could introduce variations in particle size. Therefore, if the method is not well developed and validated, the variations in particle size measurement due to flocculation could impact the PBE conclusion. Here is a summary of things to consider for particle size distribution study. For method development, how the study analysis parameters have been chosen should be included. For example, how the dispersant, sample dilution range, stirring speed selected for measurement. It is also recommended to include precision and robustness study for method validation. For the particle size result report, it should contain full profiles of the particle size distribution, particle size result, as well as the statistical analysis. Next are the common deficiencies for the IVRT study. One common deficiency observed in the IVRT study is that there is no method development study. It was observed some end applicants use the FDA method from the dissolution methods database without additional information, and rationale to support the IVRT method is suitable for assessing the proposed generic product. Another common deficiency is that there is no validation report. It is expected and the applicant provide data to support the utilized IVRT method can discriminate the critical quality attributes of the drug product, for example, particle size. In addition to lack of method development and validation study, we also observe lack of statistical analysis of the release data in the ANDA submission. Therefore, for IVRT study, method development report should be provided to include how the experimental parameters are selected. For example, how the media composition, volume, stirring rate be selected. The IVRT study should also be properly validated by demonstrating the discriminatory ability and reproducibility. Formulations with intentional and meaningful variations with regard to particle size is a good testing sample to verify the IVRT method is discriminatory. For the IVRT result, because the product is for injection, The data should be provided as percent cumulative in vitro drug release, and the equivalence can be demonstrated using model-independent similarity F2 factor. To conclude, injectable drug substance suspensions are formulated without release modifiers, and the drug substance is the only insoluble component in the product. An in vitro B approach is based on the understanding of the critical quality attributes of the product that affect bioequivalence and relies on totality of evidence. The particle size distribution study and the in vitro drug release study should contain sufficient information regarding method development, method validation, and study result. Challenge question number 1. Is this statement correct? The particle size distribution method should be validated with regard to precision and robustness. Yes, this statement is correct. Challenge question number 2. Which of the following statements for the IVRT study in the PSG for injectable drug substance suspension are true? A. IVIVC is required to justify the used IVRT study. B. No method development and validation report need if the dissolution study information can be found in the FDA's dissolution methods database. C. The dissolution study information in the FDA's dissolution methods database serves as a starting point, and applicants need to submit sufficient method development and validation reports. D. The used IVRT method should be able to discriminate the critical quality attributes, for example, particle size. The correct answer is C and D. The dissolution information in the FDA's dissolution method database serves as a starting point, and applicants still need to submit sufficient method development and validation reports. The used IVRT method should be able to discriminate the critical quality attributes, for example, particle size. I would like to give thanks and acknowledgement to Dr. Bing Qing, Dr. Yan Wang, Dr. Darby Kozak, and Dr. Robert Leinberger. I also want to thank our internal collaborator, Dr. Xiaoming Xu and Dr. William Smith, as well as our external collaborator, Dr. Diane Burgess. Thank you for your attention. Please don't hesitate to reach out for any of the questions. Also, I will be back shortly to answer any of your questions in the panel discussion.
Thanks for the kind introduction. It is my pleasure today to discuss with you our recommendations in the product-specific guidance for peptide products, especially for the risk-based comparative immunogenicity assessment and impurity profile assessment. Our learning objective for today is, number 1, after the talk, you should be able to understand and describe some of the immunogenicity risk and considerations for peptide products. Then understand and summarize some of the comparative analysis that may be included in a PSG for peptide products. And finally, you should be able to correlate the types of product-specific guidance recommendations with the different immunogenicity risk of a peptide product. Next, I would like to start off by providing a brief overview of product-specific guidances, or PSGs. So the agency provides the current recommendations for demonstrating therapeutic equivalence through these PSGs. This is important for developing generic drugs because generic drugs are approved based on the findings of therapeutic equivalence to the reference listed drugs, or also known as RLDs. Because of the potential immunogenicity risk of the peptide therapeutics, the risk of each peptide can also differ. We will recommend appropriate studies in the PSGs based on our current understanding of a product's potential risks. Of course, when our scientific understanding changes, we will use the PSG to communicate and update the appropriate recommended studies. And later in this talk, I'll give an example of how we've done this with the vasopressin PSG. So peptides, they are typically shorter than biologics or proteins, and they typically have less immunogenicity risk. However, it is still just a shorter form of a protein if you think about it, and the same scientific concern and reasoning can also apply to peptides. And when we're looking at the immunogenicity risk, we're looking at the following considerations. First, from the peptide product considerations, which may include these following aspects. One of them is related to peptide size. So the longer peptides may have higher immunogenicity risk compared to the shorter one because it may contain more HLA binding epitopes. The route administration should also be considered because certain routes, such as sub-Q injections, will be more risky than, say, something that's oral route. Now, if the peptide sequence is highly homologous to endogenous human protein, then the concern is that if you raise an antibody against the peptide, it may cross-react with the endogenous protein and cause deficiency syndromes. And finally, if the peptide has a longer the usual half-life, you could have a concern where you may have greater chance of generating anti-drug antibodies, hence increasing its risk. We also should look at other considerations when we're assessing immunogenicity risk, such as patient population and the drug indication, whether it's indicated for patient with risky immunogenicity or immunity issues. Other clinical experience of the RLD can also be used, such as the level of ADA generated in the clinical studies or any severe adverse events that are found during the clinical study. So with all of these considerations combined, we can get some relative information about the potential immunogenicity risk of a peptide product. So in order to minimize the risk and ensure that the following peptide products are not higher clinical risk, including the risk of immunogenicity, then the reference we will want to see the comparative analysis showing no clinical meaningful differences. And these comparative analysis would include both analytical testing as well as functional and cell-based assays. API similarity should be demonstrated for all generic products, not just for peptides. But for shorter and simple peptides, this comparison can be as simple as just comparing the primary sequence. But for longer structured peptides, additional analytical studies to compare the HOS or high-order structures, such as secondary structures and aggregation states, should also be recommended. Impurity difference may impact the immunogenicity potential of a proposed generic. So for peptides with elevated immunogenicity risk, we'll recommend not only to conduct the impurity profile, which is typically done using LCMS, but if a peptide has a higher immunogenicity risk, such as the 5 peptides outlined in the synthetic peptide guidance, the adaptive immunogenicity assay will be recommended to assess the presence of MHC binding potential. And for certain peptides with high risk, the innate immune response assays, which could also be recommended on the whole peptide product. And please note that this comparison may be recommended independent of the findings from impurity profile differences, meaning that even if you don't see a difference in your peptide-related impurities, the innate immune response assay should also be conducted. So here's a slide that summarizes the different types of recommended studies in these peptide PSGs. And as you can see, not all peptides are treated the same. We will recommend studies based on the perceived risk of these peptide products. So if we start from the top of the list, we have few simple peptides such as octreotide simple injectable and brahmalanotide. Both are small peptides and they have a minimal risk. So we just ask for simple API sameness comparison as well as high order structure and oligomer aggregate study. Then we move down the list with peptides such as vasopressin and secretin have elevated risk. They are slightly higher, so we ask for some impurity profile comparison. But in general, we are not recommending the adaptive or innate immune response assay here yet. Of course, you could have also biological activity assays recommended for PSG such as secretin. And then there's the dasiglucagon, which is slightly higher above these other peptides when it comes to risk. So for those, we will ask for innate immune response on top of the recommended studies. And then if you have something that's semaglutide, pretty much on par with the other 5, when it comes to risk, on par with the other 5 peptides that are outlined in the synthetic peptide guidance, we'll recommend all of these studies that are described in the synthetic peptide guidance. So next, I'll take a deeper dive into the PSC for these 3 peptide products, vasopressin, dasiglucagon, and semaglutide. Vasopressin is a non-amino acid polypeptide hormone that causes contraction of vascular and other smooth muscles. So initially, we had concern with this product having the possibility of developing anti-vasopressin antibodies after chronic dosing. So we recommended during the review of these applications for new impurities, immunogenicity assessment of new impurities that are present in the proposed drug product, as well as impurities present at an elevated level compared to the ROD. So essentially, we're treating these at a higher risk consideration and ask for the immunogenicity assays. However, after the approval of the 505(b)(2) vasopressin, we noticed that there is a difference in how these are internally. There's difference in how we review the vasopressin and as well as the B2s. We internally initiate reconsideration of the review practice for the vasopressin products. And after the discussion and internal review, we find out after further studying of the post-marketing experience of the marketed vasopressin product, we arrive at the conclusion that this product is considered to have a relatively low immunogenicity risk. So in our PSG, we also reflected this, that in general, immunogenicity risk assessment for impurities are not recommended or not necessary. So in February 2022, we developed and published the vasopressin PSG to communicate this change in initial assessment and further clarify the comparative studies needed. So instead of recommending conducting adaptive and innate immunogenicity assays, the PSG will only recommend conducting API sameness, impurity profile, aggregation profile comparison. Now, unless there's anything new, unusual new impurity or aggregation state, or highly elevated impurity or aggregation state in the proposed generic product, the immunogenicity assessment will not be requested. And next, dasiglucagon. Dasiglucagon is a 29 amino acid human glucagon analog peptide. Dasiglucagon is similar to glucagon sequence and indication. The reference is a synthetic product that is indicated for emergency use. So immunogenicity risk of the peptide appear to be lower because it is based on the ROD's labeling, where less than 1% of the patients developed anti-drug antibodies in the clinical trials. Because the risk of the dasiglucagon is slightly lower than glucagon and the other peptides outlined in the peptide guidance, the synthetic peptide guidance, the PSG reflects this by recommending comparative impurity profile studies without mentioning the absolute need for adaptive immunogenicity study unless upon the agency assessment impurity of concern is discovered. However, other studies, such as the innate immune response assays on the whole product is also still recommended, along with all the other comparative studies, such as API sameness, including the biological activities. Finally, semaglutide. Semaglutide is a 31 amino acid GLP-1 agonist peptide. It is similar to liraglutide in sequence, also for the intended indication for the same patient population. So like liraglutide, it is a homologue of a non-redundant endogenous hormone and is chronically used through sub-Q injection. So because of these similarities to liraglutide, its immunogenicity risk, we can say it's pretty much the same as the other 5 peptides, along with the liraglutide, outlined in the synthetic peptide guidance. So therefore, in the PSG of semaglutide, we recommend the sponsor to follow the recommendations outlined in the synthetic peptide guidance. So although the peptide guidance is specific to the five peptide products listed in the guidance, scientific principles and elements of the synthetic peptide guidance may be applied to other high-risk products such as semaglutide. And for those unfamiliar with this guidance, it basically provides recommendations on how to address the potential immunogenicity risk for peptide-related impurities. It's specified for 5 high-risk products, including liraglutide, tetraglutide, nasiratide, and glucagon. So in summary, different risks of peptide products will have different sets of studies recommended through the PSGs. The higher the perceived risk or immunogenicity risk of a peptide, the more recommended comparative and immunogenicity studies will be outlined in the PSG for that peptide. PSG reflects the current agency's scientific thinking on the most appropriate methodology for developing drugs and generating evidence supporting therapeutic equivalence. However, it is not static. The agencies can update the PSG when current thinking evolves or additional scientific knowledge gained. And lastly, please reach out to us through control correspondence or pre-ENDA process to discuss any proposed scientific studies that may deviate from the PSG recommendations. I'd like to end the presentation by acknowledging the following people who have helped me develop and organize this presentation. So now let's move on to the challenge questions. Question 1, true or false, PSG only contains recommended studies for demonstrating bioequivalent studies for generic products. The answer is false. PSG not only contains bioequivalent studies, but other studies as well, to not demonstrating just bioequivalence, but also therapeutic equivalence of a generic product. Challenge question number 2. Which of the following statements is not true? A. PSG contains recommended studies to demonstrate product sameness for both pharmaceutical equivalence and bioequivalence. B. Immunogenicity risk assessment using non-clinical assays is recommended for all peptide products regardless of their risk. C. Adaptive and innate immune response assays are typically recommended for certain peptide products with immunogenicity concern. And D. Peptide products not covered by the synthetic peptide guidance may still reference parts of the recommendations outlined in that guidance. And the answer is B. Immunogenicity risk is not using non-clinical assays not only recommended for it's not recommended for all peptide products. It is depending on the risk of the peptide product. Thank you for your attention. If you have any questions, I'll try to answer them at the panel session.
Thank you for the great presentations. If you haven't had a chance to enter your questions in the Q&A chat pod, we remind you to do so at this time. We do have a few additional panelists we'd like to welcome to the panel. First, we'd like to welcome Dr. Da-Ping Cui, Lead Pharmacologist, Division of Bioequivalence 1, Office of Bioequivalence and OGD, and Dr. Cameron Smith, Branch Chief, Division of Liquid-Based Products 1, Office of Lifecycle Drug Products and OPQ. We do have some questions. The first few questions will be addressed to Dr. Xiangnan Zhang, and here is the first question for Dr. Zhang. For injectable suspension products, during in vitro drug release testing, what is the recommended temperature at which the dissolution media should be maintained?
Thank you for the question. Normally, we do not recommend a specific temperature for the dissolution media to be maintained. We recommend you provide the method development and validation data support your selected temperature. Thank you.
Thank you for responding to that question. We do have a few more questions for Dr. Zhang, and here is the next question. Regarding drug substance injectable suspension, to prove the discriminatory of the dissolution method, generic players should perform intensive experiments like the varying active and excipient concentration, PSD, non-sync condition. Please suggest leftover parameters to study the discriminatory.
Thank you for the question. I think you provide a great example regarding the discriminatory study for the selected dissolution method. The study of discriminatory ability can be achieved by intentionally manufacturing a few batches of the product with meaningful variations in formulation and manufacturing parameters. For example, particle size and drug loading. We recommend you identify the critical product attributes that affect drug release and demonstrate that your dissolution method can be used to discriminate this potential difference. Thank you.
Thank you for responding to that question. Our next question for Dr. Zhang is the following. You mentioned about flocculation of particles. Does flocculation need to be considered for particle size distribution, for a particle size distribution study? And also, if R&D data shows that flocks affect IVRT, will IVBE be done using flock size?
Thank you for the question. This is a great question. First, I would like to address the first part of the question. Does flocculation need to be considered for particle size distribution study? I want to answer this first question from two aspects: what to be submitted for particle size distribution study, and what data should be subjected to PBE analysis. In brief, when characterizing particle size of suspension formulation with flocculation feature, the general expectation is that applicants should characterize particle size under different conditions, capturing both flocculated and deflocculated particle. With regard to PBE analysis for establishing equivalence in particle size, only the condition that mainly capture the size of deflocculated particle, the primary particle, is needed. It is noted that the flocculation is a formulation characteristic, and it is mostly governed by formulation composition. Therefore, when a proposed formulation deemed Q1, Q2 to the reference formulation, the main source for potential difference in flocculation behavior is the particle size of the primary particle. As such, applicants should measure the PSD under continuous sonication, which allow better capturing the contribution from primary particle population for PBE analysis. To conclude, the flocculation behavior should be captured during particle size characterization, but only the primary particle is subjected to PBE analysis. For the second part of this question, I want to highlight the purpose of IVRT and particle size characterization are different. IVRT is a general performance testing. It should be noted that it is hard to have a condition that all particles are flocculated or deflocculated. So for the IVRT study, applicants can develop and validate conditions per reproducibility and discriminatory ability, and the condition will likely have both flocculated and deflocculated particle present. Thank you.
Thank you for responding to that question. We have one more question for Dr. Zhang, and here is the question. Is a separate method development report for particle size required if a method validation report is present and method development information is included in the product development report?
The short answer is no. So when we talk about the method validation and the method development, it can be one report in general. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that came in from Dr. Eric Pang. And here's the first question. The PSG did not recommend an impurity profile, adaptive, and innate immunogenicity studies for octreotide injectable. Under what circumstances does the agency request these immunogenicity studies for octreotide injectable?
Thank you for the question. So for octreotide injectable, simple injectable, is our understanding that it is a solution, it's a product with low immunogenicity risk. So typically these will not be required to have immunogenicity studies to be submitted upon review, unless there's some impurities. As always, if there's something in there that shows to raise the impurity immunogenicity risk, for example, or something that's highly unusual impurity, or if the formulation could, well, it depends. I think most of the time it's just that if there's a very, very different impurity or something that's very highly elevated impurity, those will be something to be a concern about. But still, it depends on what kind of impurity it is.
Thank you for responding to that question. We have another question for Dr. Pang. And here's the question. Regarding innate immunogenicity, is it adequate if the test drug product demonstrates lower innate immunogenicity potential compared to the reference listed drug, or should it demonstrate equivalence with the RLD?
Thank you for the question. So let me just clarify. So the purpose of demonstrating immunogenicity risk is not to demonstrate equivalence of your testing or data, but more of there is not a higher than the reference listed drug when it comes to risk. So it's preferable that your product demonstrate no higher risk than the reference listed drug. Thank you for the question.
Thank you for responding to that group of questions. Moving on to the next panelist, we do have a question that came in for Dr. Daping Kui, and here is the question for Dr. Kui. What is the recommended duration, dissolution runtime, for in vitro drug release testing for injectable suspension products?
Thank you for the question. Sorry, I was on mute. Yeah, so the short answer is that no fixed duration for the recommended IVRT testing for all the injection suspension product. It's case by case, drug product specific, and I think the general rule is that when you conduct the IVRT, you should ensure that the duration you use for your study is sufficient to have a complete release for your drug products in the testing in your IVRT study, and also you should meet the requirement for make your testing to be discriminative. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we've got a few questions that came in for Dr. Cameron Smith. Here's the first question for Dr. Smith. FDA has asked applicants to perform innate immunogenicity assessments using a sample of drug product batches at or near release and at expiry. What is the FDA's rationale for asking for this to be done? Wouldn't the at expiry provide the worst case for testing?
Hi, thanks for the question. Yes, I think I agree that the at expiry testing would likely be the worst case scenario. But the reason we ask for on our near release as well is because if there is an innate immune response modulating impurity that is stability indicating, that would help us get at that and elucidate whether or not that is the case. So, for example, if you had a leachable that was leaching from the container closure system, that would likely be a stability indicating impurity. And so you may, if that impurity induced an immune response, you may see a difference in the innate immune response between the release samples and the samples aged to the end of the shelf life.
Thank you for responding to that question. The next question for Dr. Smith is the following. Do you recommend using LC or LCMS data when comparing impurities in vasopressin and other synthetic peptides?
Yeah, so generally speaking, as the complexity of the peptide structure increases, we are more likely to recommend LCMS data or some sort of two-dimensional mass spectrometry and chromatography type data. It helps us get at two things, whether or not the analytical method is adequately separating all of the peaks that are present, all of the impurities that are present, because you can use the second dimension of mass spectrometry to elucidate peak purity. And so that's why we recommend that in most cases, so that you can adequately characterize the peptide profile that's present in your product.
Thank you for responding to that question. We do have one more question for Dr. Smith. Here's the question. Is there a possibility that for adaptive immunogenicity, the agency would require the testing of the whole product and not only individual peptide-related impurities?
No, generally speaking, the adaptive immune response testing is only conducted on purified impurities. We likely would not ask for that to be conducted on the formulated drug product. And so this is in contrast to what we'd expect for the innate immune response assays, which are conducted on the fully formulated product.
Thank you for responding to that question. We've got time just to try to get one more question in. We have a couple of questions left for Dr. Xingnan Zhang. And here's the question for Dr. Zhang. Is sync condition required for IVRT studies?
We acknowledge the drug substance solubility is generally very low in drug substance injectable suspension products, and we understand sometimes a non-sync condition is used for improvement of method discriminatory ability. Therefore, sync or non-sync condition either way may be acceptable as long as you provide sufficient justification. Thank you.
Thank you for responding to that question. And that's all the time we have for questions in this panel. A huge thank you to our speakers and panelists for answering numerous questions that came in from our audience. We'll move right into our final session, which is session 4 on noteworthy complex generic drug approvals, multi-phase systems. Our first presenter is Dr. Quickly Fan, Pharmaceutical Science, Division of Liquid-Based Products 2, Office of Life Cycle Drug Products in the OPQ. Dr. Yuriko Haragaya, Senior Staff Fellow, Division of Bioequivalence 2, Office of Bioequivalence and OGD. Then we welcome Dr. Ben Chen, Senior Chemist, Division of Therapeutic Performance 1, Office of Research and Standards and OGD. And our final presenter is Dr. William Smith, Research Scientist, Division of Product Quality Research, also Office of Testing and Research in OPQ. Now please join me to welcome our first presenter, Dr. Fan.
Thanks for the introduction. Hello everyone. Thank you for joining us today. I will focus on what's new in the CMC part of these two ophthalmic emulsion products. Dr. Haragaya will focus on the BE part. Learning objectives. Highlights of new draft PSG on cyclosporine and the new nuclear magnetic resonance NMR method for quantifying the drug distribution of difluprednate in different phases. The background of my talk. The PSG provides the in vitro BE option, which following criteria need to be met. First, Q1, Q2 thickness, which is the test and LD formulations to be qualitatively and quantitatively the same. Second, acceptable comparative physical-chemical characterization, Q3, including drug distribution in different phases. Third, acceptable comparative globule size distribution, which is now in vitro BE study 1 and BE study 2 RVRT. Again, I will focus on the first two criteria. First, what's new in current draft PSG for cyclosporine ophthalmic emulsion? This current draft PSG was published in August 2022, exactly 1 year ago. The major new part of this PSG is the footnote 3 on Q1, Q2 thickness. It's so important. Let me just read it out loud. For ophthalmic drug products, FDA has determined that as a scientific matter, any qualitative or quantitative deviations from the LD, even in inactive ingredients listed in 21 CFR, should be accompanied by an appropriate in vivo BE study or studies. Hopefully you have already noticed that and carried it out in your development work. For the part of parameters to measure, now the comparative globule size distribution moved to the in vitro BE study 1, again will be focused of Dr. Haragaya's talk. If you are interested in the background information of this PSG and other details of this PSG as well, I highly recommend this journal paper titled as Product Quality Research for Developing and Assessing Regulatory Submissions for Generic Cyclosporine Ophthalmic Emulsions. This is published in the AAPS journal in January 2023 this year. Also, there is a device information in this PSG as well. Second, the NMR method for quantifying the drug distribution of difluprednate in different phases. This research work has been done by our colleagues in OTR of FDA, and the work was published in Molecular Pharmaceutics last year. Actually, it was the cover feature of the July issue of that journal. From the cover photo here, you can see the vivid cartoon depicting API distributed in oil and surfactant phases, plus their well-separated peaks in the spectrum. Very briefly about the method. The method uses certain dilutions so that API in the surfactant phase is more resolved. And here you can see these well-separated distinct peaks of APIs in different phases. Black peak represents API in the water phase. There are two red peaks. The left one represents API in the surfactant phase, and the other red peak is API in the oil phase. Here is the brief description of the spectral and method. So what are the benefits of this new NMR method for drug distribution? First, it's non-invasive. Second, as I just showed you, distinct peak of API in each phase in the spectral is achieved. Third, it's easy to measure API distribution in different phases by integration. How about the mass balance? Here you are. In water phase, it's about 1.8%, very similar to the API's solubility in water. In surfactant phase, there is 35%. Of course, the majority API is in the oil phase, 59%. In total, it's about nearly 96%. It's very good mass balance results. If you want to use this method, it needs to be validated, and we recommend it as a one-time study. What about the other method for the drug distribution? Here you can see this one is the ultracentrifugation, and this is the ultrafiltration. Also, the third one will be dialysis. Here's my challenge question. Very easy. Which method for drug distribution study in different phases is non-invasive? Is it A, ultracentrifugation, or B, ultrafiltration, or C, NMR, or D, dialysis? Well, it's very straightforward. It's C, NMR. The summary of my talk. First, I presented highlights of new draft PSG for cyclosporine ophthalmic emulsions. And then I briefly described the NMR method for quantifying the drug distribution of difluroprednate in different phases. This is a non-invasive method. Finally, I want to thank my branch chief, Dr. Helen Zhang, Division Director, Dr. Pahala Simamora, Associate Director of OLDP, Dr. Andre Rao, for reviewing my slides, and also Dr. Kang Chen, who is PI of this method development work, answering a lot of my questions. Last but not least, SBIA organizers, thank you very much. With that, thank you for your attention, and I'm looking forward to your questions.
Thank you, Dr. Chu-Shin Fan, for very informative CMC portion of presentation. Hello, everyone. Thank you for joining us today. My name is Yoriko Harigaya from Division of Bioequivalence 2. In my presentation, we are going to take a look at two ophthalmic emulsions from the bioequivalence perspective. This presentation will tell you the differences between two cyclosporine ophthalmic emulsions in globule size distribution studies, how to select the membrane in in vitro release test for cyclosporine and difluprednate ophthalmic emulsions, how long in vitro release tests be performed, what is discriminative in vitro release test. As Dr. Fan mentioned, current draft product-specific guidance provides an option of in vitro studies for cyclosporine and difluprednate ophthalmic emulsions. My talk will focus on two in vitro studies, globule size distribution and in vitro release test. First, I will share the recommendations on cyclosporine ophthalmic emulsion for globule size distribution studies. Why globule size distribution study recommended in PSGs are different in two cyclosporine ophthalmic emulsions. For cyclosporine ophthalmic emulsion 0.1%, PSG recommends evaluation of D50 and span to demonstrate bioequivalence. However, cyclosporine ophthalmic emulsions 0.05%, PSG states D50 and span may not be sufficient and recommends evaluation of Earth mover's distance or other suitable metrics. It is because cyclosporine ophthalmic emulsion 0.1% displays monomodal distributions. D50 and span are reasonable parameters to be evaluated. While cyclosporine ophthalmic emulsion 0.05% displays multimodal distributions, thus D50 and span cannot describe the shape of distributions. Next, I would like to share the general recommendations for in vitro release tests for cyclosporine and difluprednate ophthalmic emulsions. These recommendations may not apply to your novel IVRT method. How to select membranes for in vitro release test. Since selection of the membrane will significantly affect drug release kinetics, it is important to test different types of membranes. Also, evaluating multiple replicates of each membrane will help identify the reproducibility. Pore size of the membrane should separate drug from oil globules. Thus, ideally, pore size is smaller than the globule size and larger than size of micelles. We also recommend to evaluate the inertness of membrane to minimize the membrane adsorption. Mass balance study identifies whether your in vitro release test measures drug release from oil globules. Most oil globules ideally stay in the donor compartment when drug and micelles are released to the receptor compartment. How long in vitro release tests should be performed? For ophthalmic emulsions, you may see a biphasic release profiles, such as aqueous and micellar phase will be released rapidly. Then slow drug release from the oil phase may be observed. In vitro release tests should capture the entire drug release profiles. So how long it should be performed? Ideally, in vitro release tests be continued till you observe a complete release, which is at least 85% of drug label claims. or continue the study till no significant increase observed over 3 consecutive time points. If your in vitro release test does not reach 85%, we recommend that you submit justifications for not reaching complete release. What is discriminative in vitro release test? In vitro release test should capture the gradual drug release from the oil phase. Thus, study duration can be long. From the bioequivalence perspective, in vitro release test does not need to mimic the physiological condition. Therefore, no need to predict the bioavailability. Your in vitro release test needs to discriminate different quality attributes or manufacturing process, such as different globule sizes or other physicochemical properties. Also, we generally recommend to use F2 similarity factor for comparison of 2 drug release profiles. F2 will compare the entire release profiles, not just one time point. And ideally, variability be reasonably low to have an adequate sensitivity. For instance, in this figure, if the red profile with the long error bars is a control from multiple batches, formulations altered 1 and altered 2 were considered not discriminative. This control may include test or reference batches evaluated in the method validation and pivotal studies to understand the batch-to-batch variability. In the bottom figure, in vitro release test displays low variability, and you can see the separation between control batches and altered formulations. Thus, this IVLT discriminatory ability is more desirable. Here is a poll question. Which one is not needed to be evaluated in the donor and receptor compartment in the mass balance study for ophthalmic emulsions? A. Active pharmaceutical ingredient. B. Oil. C. Diluent. D. Surfactant. The answer is C. Diluent. In summary, different cyclosporine ophthalmic emulsions may have different shapes of globule size distributions. For in vitro release tests for ophthalmic emulsions, your membrane should separate drug from oil globules. Mass balance study will help identify the reasonable in vitro release test. For discriminative in vitro release test, the entire drug release profile should be compared, not just a single time point. I would like to acknowledge countless support from Honlin Zhang, Xiaozhen Zhang, Xinlan Li, Heesun Chen, Den Zhang, Xiaomin Xu, and SBIA Organizing Committee. Thank you for your attention. This concludes my presentation. Now I would like to hand it over to Dr. Bin Qin for his presentation of amphotericin B liposome.
Hello everyone, my name is Bin Qin. I'm a Senior Staff Fellow in Office of Research and Standards at the Office of Generic Drugs. My talk today is about amphotericin B liposome and its product-specific guidance revision. In this presentation, I will first describe the approved amphotericin B drug products and discuss potential challenges to develop generic amphotericin B liposomes. Next, I will explain FDA's latest revision to the product-specific guidance on amphotericin B liposomes. And lastly, I will illustrate GDUFA research on this product. Amphotericin B is a protein antibiotic first discovered in 1950s. It's an amphiphilic molecule. The monocyclic protein lactone ring of amphotericin B contains a hydrophilic polyol subunit and a hydrophobic protein subunit. Amphotericin B can bind to ergosteroids of the cell membrane of susceptible fungi. It forms transmembrane pores, leading to compromise of membrane barrier function. Ions can leak out, resulting in cell death. Amphotericin B can also bind to cholesterol components of the mammalian cells, with a binding avidity more than 10 times lower than that of ergosteroid. The binding to cholesterol is the main cause of toxicity. Amphotericin B is poorly soluble in water. It's presented as monomer or aggregates. Heat treatment can induce superaggregation of amphotericin B, reducing in vitro toxicity. As we know, amphotericin B has very low water solubility and membrane permeability. So many efforts has been made to develop drug products for parenteral route of administration. There were 4 approved drug products. Fungizone was the first amphotericin B drug product approved by FDA. It was approved back in 1966. Fungizone uses biosol detergent deoxycholates as excipients, and it can form micellar suspension when reconstituted in 5% dextrose solution. In 1990s, 3 lipid-based formulations were approved by FDA. They were Abelset, Enfotec, and Emulsion. Abelset contains ribbon-like particles, and Enfotec contains disc-like particles. Emulsion is 2 single-layer liposome formulations among the 4 approved drug products. The drug substance in Emulsion is intercalated within the membranes rather than loading into the aqueous core. This is very different from liposome products such as Topical. Emulsion has been widely used for the treatment of fungal infections and other indications. The drug product has been included in WHO lists of essential of medicines, even though it may be difficult to assess the products in many countries. The global sales of Emulsion is about $500 million, and the US patent expired in 2016. There have been a lot of interest in generic production of this product. However, like other liposome injection drug products, development of generic version of amphotericin B liposome is not straightforward, and there are many potential challenges. For example, how to demonstrate bioequivalence and technical difficulties in manufacturing. Like other liposomal products, to demonstrate bioequivalence, conventional PK may not be sufficient. Additional supportive studies are needed. For manufacturing, a special consideration is the toxicity of the finished products. Manufacturing process can impact the product toxicity by change the aggregation status of amphotericin B in the lipid bilayer. So in this case, the toxicity assessment should also be considered when you establish bioequivalence of amphotericin B liposome. This slide shows you an example how manufacturing process can play a role in the in vitro toxicity of amphotericin B liposome. There were 2 follow-on amphotericin B liposomes products approved outside the United States. These 2 products were reported to have identical chemical compositions as Emulsion, but were manufactured differently. The result of a comparative red blood cell assay shows that the drug concentration inducing half maximum potassium release also known as TC50, for both follow-on products are substantially lower than that of Emulsion. This suggests that they have lower in vitro toxicity, or they have higher in vitro toxicity than Emulsion. Of note, one of the products, Emulsion, has been withdrawn from market due to toxicity concerns. The agency has posted a guidance for industry to adjust topics related to liposome products, including CMC, human PK and bioavailability, and labeling documentation. Additionally, the agency has issued a product-specific guidance on amphotericin B liposome to facilitate generic drug development. The PSG was initially posted in 2014 and revised for 2 times. The latest revision was made in 2020. I will talk about the latest revisions in the following slide. The current product-specific guidance on amphotericin B liposome recommends in vivo studies and in vitro studies to establish bioequivalence. There are 2 changes in the 2020 revision. The first change was made on the in vivo study. Previously, a multi-dose steady-state PK study in patients was recommended in the PSG. This revision changed the study to a single-dose PK study in healthy subjects. This change was made based on new healthy subject information provided by generic drug industry through control correspondences and pre-ANDA meetings. Another change was adding in vitro red blood cell assay and measurement of state of associated amphotericin B and the lipid bilayer in the in vitro studies. This change was made based on finding from GDUFA research project. In the next few slides, I would like to provide you an overview of GDUFA research for amphotericin B liposome. This is a list of GDUFA research projects that have been conducted for amphotericin B liposomes. I would like to share with you some of the key findings from the last 2 contracts, as this data help us to better understanding what are the critical process parameters of amphotericin B liposome manufacturing, and how these parameters influence the in vitro toxicity, as well as what type of characterizations can be used to inform the in vitro toxicity. In the research projects, a 4-step process described in the US patent was used. These 4 steps include Step 1, mixing lipids and drug and organic solvents. Step 2, removal of organic solvents by spray drying. Step 3, hydration of the spray dry powder and microfluidization to form liposomes. And Step 4, lyophilization to a dry powder as the final product. The patent protocol has no comments on the extended heating step and only reference a 40 to 60 minutes incubation at 65 degrees Celsius during lipid hydration in Step 3. Through accidental observation and literature survey, the research team identified that an additional heat treatment, so-called curing, is needed to produce formulations that have similar in vitro toxicity profile to Emulsion. So the effect of the curing step was extensively investigated throughout the projects. The manufacturing process was evaluated using quality by design approach, 9 critical process parameters, and 5 critical quality attributes were used. Figure on the right shows the variable importance plot. This plot presents the impact of all process variables on all quality attributes simultaneously. The most important variable is the curing temperature, followed by quantitatively formulation difference, and lastly by liposome processing parameters. To study the potential impact of curing on different steps of the process, liposome samples that were generated with curing after Step 1, solvent mixing, Step 2, spray drying, and Step 3, microfluidization, respectively. UV-Vis methods is a commonly used method to study the aggregation status of free amphotericin B, and it's indicative of aggregation status change of amphotericin B. So this method was adapted to categorize the liposome formulations. The red dash on the right figure presents the main peak position of Emulsion, 321 nanometer. As you can see here, only when the formulation was heated in step 3 did the main observation peak shift to 321 nanometer. This is consistent with that of Emulsion. Red blood cell assay was also conducted to evaluate the in vitro toxicity of these samples. Only the samples heated after microfluidization, step 3, show a significant reduced toxicity. This tells us the curing incubation should be introduced after step 3. Curing temperature and curing time can also have an impact to the product. Liposome formulations cured at different temperatures and time were also prepared. The spectrum on the left shows as curing temperature increase, main peak underwent a blue shift from 325 to 350 nanometers to 321, 322 nanometers. This suggests the aggregation status of Emulsion undergoes change from the loose aggregates such as monomer or dimer to a tight aggregate such as oligomers. The figure on the right shows the change of main peak position as a function of time under different temperatures. You will see here only a curing temperature above 50 degrees Celsius can achieve similar main peak position as the Emulsions. And here, the study also indicated that the timing of curing is also critical. The in vitro drug release of liposome formulations of those samples were also studied using a method developed by University of Michigan in a previous grant. The cumulative drug release profiles over first 80 minutes and over 25 hours are presented on the right. The release profile of Emulsion is in the blue color, and the uncured control sample is in the gray color. You can see here a general trend is when incubation time is fixed to 6 hours, higher curing temperature samples have slower drug release rate, and the profile is closer to the release curve of Emulsion. To better understanding the drug release behavior, 2 first-order release kinetics models were used to fit the release curves. When the first 1 pool release model was used, we can see there were good linear correlation coefficients for all the curves of initial 75 minutes data. And we also observe high curing temperature resulted in a slower release. However, this model poorly fit the full 24 hours release curve. We then use first-order 2-pool release model to fit the curves. We can get a good fit for all the complete hour data set. This suggested the 2-pool model can better categorize the release kinetics. This model indicated that amphotericin B may exist in 2 different aggregation forms, loose and tight. This is also deduced from UV-Vis analysis. Figure on the right shows the changes in time of the 2 aggregate pools. This is expressed as concentration. It shows that the tight aggregation pool, pool B, increases at the expense of loose aggregate pool, pool A, while the total drug concentration remained fixed. Potential correlation between the in vitro toxicity and in vitro release kinetic parameters were explored. The results below show that they have a good linear correlation between normalized in vitro toxicity and the initial rate constant from the 1-pool model and the CA-CB ratio from the 2-pool model. There were also good correlations between normalized in vitro toxicity and the peak position and main peak ratio. In this slide, I would like to provide a quick summary about the GDUFA research I just presented. Curing was found to be very important for reducing the toxicity of amphotericin B liposome formulations. As the curing progressed, amphotericin B can change from the loose aggregate to a tight aggregate. You can find this shift from the UV-Vis methods and in vitro release testing. The two physicochemical and analytical methods, UV-Vis and IVRT, correlated very well to in vitro toxicity. As a side note of this presentation, the first two generic amphotericin B liposome products were approved in 2021 and 2022 respectively after the 2020 product specific guidance revision. Here are some take-home messages of my presentation. GDUFA research and information submitted by the generic drug industry has been very helpful to the guidance revision on amphotericin B liposomes. Manufacturing process play a very important roles in product toxicity. The product toxicity can be informed via physicochemical characterizations, in vitro drug release, and in vitro potassium release assay. We have two challenging questions. This is challenge question number 1. Is this statement correct? The in vivo bioequivalent study should not be conducted in healthy volunteers, as amphotericin B liposome is toxic. A. This statement is true. And B. This statement is false. Please take a few seconds to think about this question. The correct answer is false. Based on the new healthy subject information provided by industry, it's deemed safe to conduct bioequivalent studies in healthy volunteers. Challenging question number 2. Which of the following supportive comparative physicochemical categorization is not relevant to bioequivalence of amphotericin B liposome? A. Liposome size distribution. B. In vitro red blood cell potassium release assay. C. State of associated drug and lipid bilayer. And D. Internal environment. The correct answer is D. Internal environment. For amphotericin B liposome, the drug substance is intercalated in the lipid bilayer of the liposome. So the internal environment is not relevant to the bioequivalence of this drug product. With that, I would like to conclude my presentation. I would like to take this opportunity to thank my team leader, Yan Wang, my supervisor, Dabi Kozak, and my office director, Rob Leinberger. Thank you for their great help in the preparation of this presentation. Thank you for your attention. If you have any question, I will be happy to answer your question in the panel discussion session. Thank you.
Hey everybody, this is William C. Smith. I'm a research scientist over at the Office of Testing Research. Today I'm going to talk to you a little bit about some of the lab work we did on phytonadione injection relating to how the thermodynamics and self-assembly of the system work. So hopefully during this talk, you'll learn something about how to recognize the difference between macro, nano, and micro emulsion properties, how to identify some of the key aspects of the manufacturing of these different formulations, and hopefully at the end you'll learn something about how to describe the difference between thermodynamically and kinetically stable systems. Okay, so let's look at our system of interest here, which is phytonadione injectable formulation. So phytonadione injectables have three principal components: phytonadione, which is an oil-like immiscible API, vitamin K, a polyethoxylated fatty acid derivative, which is the surfactant, acts like the emulsifying agent or a soap or a detergent, and it's all dispersed in an aqueous phase. Now, the reference listed drug and the reference standard have subtly different ways that they denote the dosage form. The first being an aqueous colloidal solution, and the latter being an injectable emulsion. Now, you might think because we have an immiscible oil phase in an aqueous phase and some kind of surfactant that we could, choose either of these two options. But the fact that there is this subtle difference in the dosage form and the question of the dosage form really has some interesting implications on the regulatory pathway. So this subtle difference in the dosage form actually has an implication on the bioequivalence pathway, where something that's low risk, like a solution, if deemed to be qualitatively and quantitatively similar to the other product, is assumed to be therapeutically and bioequivalent. This is in contrast to something like an emulsion, where even if qualitatively and quantitatively similar, things like the manufacturing process can impart different physical and chemical properties to the formulation, which may necessitate additional in vitro testing or a different suite of in vitro testing in order to establish or demonstrate bioequivalence. So the question isn't really, is this a solution or is this an emulsion? The question is really, is this a thermodynamically stable system or a kinetically stable system? Here you can see we have emulsions up in the high particle size range, nano emulsions in the center here in the 500 to 100 nanometers. And then at the end, our 100 to 10 nanometer particle size is the micro emulsions. And so you can think about these different forms of the dispersion as kind of in a continuum, both in the components, so they can be made of all the three same components, maybe in different ratios or maybe at the same ratio. But then the processing equipment or the conditions that are used to manufacture the particles or the dispersions can actually change the particle size. And so the interesting thing is that the particle size is what is imparting all of these differences you see here in the appearance, so things like turbidity, whether or not it's opaque, whether it is an optically transparent or isotropic system, like you see here with the thermodynamically stable microemulsion. So I'm going to take just a slight sidetrack here to kind of make a point. And that is, some of the keen eye amongst you might have seen that I'm referring to the smallest particle size you saw there as a microemulsion. So according to the Encyclopedia of Colloid and Interface Science, a microemulsion is basically a micellar system which is transparent or translucent, so optically isotropic, and can be described as a swollen micelle. And so this is really just kind of a holdover of kind of when this was introduced in colloid science. So back in the 40s and 50s, when all of this kind of started, we had swollen micelle and microemulsion, and really the nano revolution started in the last 30 years. And so nobody was really using that terminology at that point in time. And you can see here the very first publications on microemulsions where they started to go away from the swollen micelle system, which was kind of typical back into the 1800s. So if you remember back to before our detour, I was saying that the composition and the processing can impact the particle size, which can kind of inform what state our dispersion is. So if we look on the left side here, this is a ternary phase diagram where we have 100% of the composition of our three different phases, so surfactant, water, and oil, representing these vertices. And you read this basically as this vertice represents 100%, and then directly orthogonal to that line is 0% for that one phase. And so right in the center represents a 33%, 33% mixture of these things. And so how you can kind of think about microemulsion system is kind of existing in this where you just have, your soap or your surfactant dissolved in water, and you are adding a little bit of oil into it. And so our microemulsion systems tend to live in this kind of edge region. Whereas our emulsion systems tend to be in kind of a lower surfactant regime with kind of a higher oil content. Now you can think about how to get your particles to smaller and smaller sizes in kind of two ways. First, you can make a bunch of micelles and then add some oil into them. And secondly, you can also think about taking the oil and breaking it up into smaller and smaller pieces and having the surfactant stabilize that surface. And so you can think that there's kind of this immense amount of energy at the oil-water interface. In order to reduce that energy, you can do something like add a surfactant or a surface actant agent, which actually decreases the interfacial tension and leads you to be able to create smaller and smaller particles. The other way that you can impart enough energy into the system to reduce the oil globule size is through just introducing a high energy process like mechanical shearing or increasing the temperature. So for your typical emulsion or nanoemulsion, you're taking your oil droplets and you're breaking them into smaller and smaller pieces using some kind of high energy process where you've reduced the size to where there's a sufficient amount of surfactant that can kind of stabilize that surface and kinetically trap it at that particle size. And so two main techniques that are used here are things like high speed homogenization or something like a microfluidizer, which uses high shear through high pressure changes. This is in kind of stark contrast to a thermodynamically stable system like a microemulsion where you only need a low energy input. So here on the right hand side we can see we're just basically dissolving a surfactant and oil stock solution into an aqueous phase, and it will slowly just disperse over time at ambient temperature and at low shear, so just with stirring. And it's important to remember that with the high energy techniques, you tend to only be able to get into that sub-micron range to around above 100 nanometers, whereas with the microemulsion system, you get into that kind of, 10, 20, 30 nanometer range. So another kind of critical aspect of the thermodynamically stable system is that it's really kind of process independent. So if you mix it slowly over the course of an hour, just dissolving it, versus you homogenize it over the course of about a minute and a half, you still end up producing the exact same dispersion. So in this investigation, we can take the path independence idea one step further, where instead of adding our organic phase into our aqueous phase, like in the previous examples, we can add the aqueous phase directly to the organic phase and go through this kind of phase inversion process. So at an equivalent composition, what we end up seeing is that we kind of get this contradiction where we get two phase diagrams, one where we're pretty much always producing microemulsions, and the other where we're producing these opaque nanoemulsions. The important thing to note here is that when we're using an aqueous to organic addition, we do see a dependence in the particle size and the optical properties or turbidity based on the processing condition or the stirring. So the rate of stirring, the amount of shear, whether or not we use a homogenizer. Whereas when we use a stirring of any variety or homogenizer in the organic to aqueous direction, we pretty much always get the same thing. So based on changing the formulation directionality and the processing conditions, we were able to form both nanoemulsions as well as microemulsions. Now the question becomes, at the composition of phytonadione injections, what is the most thermodynamically favored state? For any kind of kinetically stable system like our nanoemulsion or like salad dressing, it's kind of trapped in this high energy state, and it wants to spontaneously minimize its energy, albeit maybe very slowly. And so for an emulsion system, if you are to disturb it either through giving it enough time and energy, it will tend to phase separate, say if you heat it or leave it for a long time in the refrigerator. This is in contrast to a microemulsion, where actually the microemulsion is the thermodynamically stable or favored state, where if you take two separate phases and leave them for a long enough time or impart enough energy, the microemulsion will spontaneously form. So the question is, if we give our systems enough time and or energy, are they going to split, demonstrating that they are not thermodynamically stable, or are they going to change from a nanoemulsion spontaneously into a microemulsion? So what does happen when we try to split our nanoemulsions that we formed by adding the aqueous into the organic? Well, if we incubate them for about 30 minutes at 70 degrees C, you can see here that we have two different particle size populations, around 15 nanometers here, and then something in the, say, 100 to 200 nanometer size range, which is imparting this opaqueness to our system. And then after that, we get one single particle size distribution in that 15 nanometer range that looks nearly identical to the microemulsions that we formed when we did the original directionality of adding the organic into the aqueous phase. So when we heat these nanoemulsions, they don't split, they end up turning into a microemulsion. So we talked about two of the kind of critical additions here. So adding our aqueous into our organic phase, adding our organic phase into our aqueous phase, and lastly, and maybe the most non-intuitively, we could just add our oil directly into a surfactant dissolved into the aqueous phase and then homogenize them. And so what this looks like is this, where we have an initial macro or nanoemulsion after homogenization. After 24 hours, we get this biphasic macroemulsion split, so it forms two phases. But given enough time, about 30 days at room temperature, our macro or nanoemulsion becomes biphasic and spontaneously forms a microemulsion, given enough time and energy. So what this tells us is that those nanoemulsions that we made are actually kind of a temporary intermediate where given an increase in the temperature or just enough time at the formulation composition of phytonadione injection, the thermodynamically favored state of the system is the microemulsion. Just a challenge question here too, to see if you're paying attention. True or false, microemulsions can only be produced using high shear mechanical processes. Is that A, true, B, false, or C, I have no clue? This, of course, is false. Okay, last question. Rank these dispersions in order of increasing particle size. Is it A, macro, micro, then nano emulsion? Is it B, nano, micro, macro? Is it C, micro, nano, then macro? Or D, none of the above? This happens to be C. Microemulsions are actually the smallest in particle size. And I just want to thank the whole Novel Complex Generics team who worked on this with me, and thank you all for listening.
Thank you all for the great presentations. We're going to be moving into our final Q&A panel of today, and we'd like to welcome some additional panelists. First, we would like to welcome Dr. John Zhang, chemist, Division of Liquid-Based Products 2, Office of Life Cycle Drug Products and OPQ. Dr. Hee-Sung Chung, lead pharmacologist in Division of Bioequivalence 1, Office of Bioequivalence and OGD. Dr. Kandakar Alam, senior pharmacologist, Division of Quantitative Methods and Modeling, Office of Research and Standards, and Dr. Salming Su, Division Director, Division of Product Quality Research, Office of Testing and Research and OPQ. Do you have some questions? Or 1 second. My question sheet is repopulating. Just 1 second. All right, there we go. And the first group of questions will be addressed to Dr. Quixie Fan, and here is the first question. According to the current draft PSG on cyclosporin, are deviations of pH adjuster allowed?
Thanks for the question. That's a good question. It is always allowed, but if the deviation within 5%, according to the footnotes, you don't have to submit the in vivo B study. If it's higher than 5%, well, you have to submit extra in vivo B study. Hopefully this answers the question. Thank you.
Thank you for responding to that question. We've got a few more questions for Dr. Fan, and here's the next question. For a one-time study on drug distribution by NMR, is method validation required?
Yes, it is required. That's a short answer, because drug distribution is such a critical property and directly related to the bioavailability. It's just a one-time study. However, to make sure the quality of your study, the results of your study, validation is required. Thank you.
Thank you for responding to that question. We've got a few more for Dr. Fan, and here's the next question, also on NMRs. What are the advantages of NMR methods comparing to other methods of drug distribution?
Again, thanks for the question. As I mentioned, the AMR method first and foremost is non-invasive compared to other methods. Second, it has these distinct peaks, if you still remember the spectra I presented, and easy to measure just by integrate. The mass balance also excellent. So those are the advantage of this NMR method compared to other methods. Thank you.
Thank you for responding to that question. Another NMR question for Dr. Fan. Can the NMR method also measure the globule size?
Thanks for the question. The short answer is yes. If you get a chance to read the paper of NMR, which I refer to, there's a certain section about how to use NMR method to measure globule size. If you have a further question, I would encourage you to contact the PI and corresponding author, Dr. Kang Chen. He will be glad to answer any question you have. At least he is very helpful to me and to our group. Thank you.
Thank you for responding to that question. One last question for Dr. Fan in this round, and here is the question. How can we make sure our formulation is good to go for ANDA submission?
Thank you for the question. That's a very good question. You can certainly use the controlled correspondence process to make sure your formulation is good to go. Just let FDA to check to assess your formulation before all your development work. That will certainly save a lot of your time and labor later on. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist. Got a few questions that came in for Dr. Yoriko Harigaya. And here is the first question for Dr. Harigaya. For any ophthalmic products that are non-Q2 to the RLD, can in vitro studies still be used to demonstrate BE?
Thank you for your question. The short answer is no. As current draft PSG for ophthalmic emulsion recommends, the test product and reference rescue drug has to be Q1, Q2 the same to choose in vitro option to demonstrate bioequivalence. And also, as Dr. Chu-Shin Fan mentioned, the current refused to receive standard guidance as well as cyclosporine ophthalmic emulsion guidance states exception excipient is not applicable to ophthalmic emulsion. Thank you.
Thank you for responding to that question. We do have a few more for Dr. Harigaya. And here is the next question. In the pivotal IVRT study for ophthalmic emulsions, do 3 batches need to be tested for both test and reference products?
Thank you for the question. The short answer is yes. We recommend you to evaluate 3 batches for each test and reference product to evaluate batch-to-batch variability. And also this information help us understand the discriminatory ability of your IVRT method. Thank you.
Thank you for responding to that question. The next question for Dr. Harigaya is the following. For mass balance studies for ophthalmic emulsions, do both donor and receptor compartments need to be evaluated?
Thank you again for the question. So for mass balance study, we recommend you to evaluate both donor and receptor compartment to ensure that total concentration is close to 100% throughout the IVRT study duration. Thank you.
Thank you for responding to that question. The next question for Dr. Harigaya. Is it acceptable to use the Higuchi model to compare 2 drug release profiles for ophthalmic emulsions?
Thank you for your question. The short answer is no, because Higuchi model is for semi-solid skin topical product, and this model will evaluate only the early portion of drug release profile. However, ophthalmic emulsion, we would like to evaluate entire drug release profile. So we generally recommend F2 similarity factor. Thank you.
Thank you for responding to that question. Just a couple more questions for Dr. Harigaya. Here's the next question. In the IVRT discriminatory study, does altered formulation need to pass F2 value of less than 50 for ophthalmic emulsions?
Thank you for the question. That is a good question. So desirable, ideally, F2 value is less than 50 for discriminatory ability testing. However, it is not required. if your method is providing greater than 50 F2 value, but control batches has low batch-to-batch variability or inter-batch variability, and there is a clear differences between control and altered formulation, and it's reproducible, we may consider it is discriminative. Thank you.
Responding to that question, just one final question for Dr. Haragaya. And here is the question. Is providing GSD data and serial dilution recommended for ophthalmic emulsions?
Thank you for the question. So it depends on the method you use. If you are using dynamic light scattering method, we recommend you to provide serial dilution to make sure your selected dilution provides stable globule sizes and polydispersity. And also make sure to have no interference from polymers. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions for Dr. Ben Chen, and here is the first question regarding potassium release assay. Is it adequate if the test drug product demonstrates a lower toxicity potential compared to the reference listed drug, or should it demonstrate equivalence with the RLD?
Thank you for the question. I think from a toxicity standpoint, a lower toxicity potential for the test product should be acceptable. However, additional consideration is that if this value deviates too much from the RLD, there may be additional concern whether other physical chemical property has changed. As we know, like the thermal treatment can also, if it's too long, can also make the impact the API stabilization and other property as well. Thank you.
Thank you for responding to that question. And the next question for Dr. Chen is the following. How does the heat treatment affect the toxicity of amphotericin B liposome?
Thank you for the question. Yeah, for the heat treatment, what we find during GDUFA research and also in the literature is that the heat treatment actually can have an impact on the aggregation status of amphotericin B inside the liposome bilayer, lipid bilayer. If you have extensive heat treatment, you can shift the aggregation status from a more loosely packed status to a more tightly packed status, which can result in less toxicity. Thank you.
Thank you for responding to that question, Dr. Chen. We do have one more question for you in this round, and here is the question. What characterization studies can be used to measure the toxicity of amphotericin B liposome?
Yeah, thank you for this question. To measure the toxicity of amphotericin B liposome, the most common way is to use the red blood cells to measure the potassium release. And there are some other categorization methods you can also use to interact to show, for example, use the UV-Vis methods as well as IVIT methods. From the GDUFA research, we found those categorization can also have a can indicate the toxicity. Thank you.
Thank you for responding to that group of questions. Moving on to our next panelist, we've got a few questions that just came in for Dr. William Smith. And here is the first question. Is globule size distribution considered a CQA for aqueous non-lipid injectable emulsions?
Hi, yeah. So in general, For emulsions, you can consider globule size distribution critical, as the manufacturing processes tend to impact the globule size distribution. And you can think about that as being important because of the impact that the globule size distribution can have on the drug distribution as far as the aqueous proportion to the globule or the oil phase concentration of the drug, as well as anything dissolved in, say, a micelle. But it is really kind of depends. So the difference between like an ophthalmic and an injection, say for IV administration, would be the impact of the globule size distribution on the bioavailability based on that drug distribution. So in general, we kind of say globule size distribution for those types of products tends to be critical, but it really depends.
Thank you for responding to that question, Dr. Smith. We do have a couple more, and here's the next question. With the different modes of preparation for a phytonadione injection, did you see any differences in product stability?
Thanks for that question also. There are two different types of stability, right? There's physical stability and there is chemical stability. So when you produce an isocompositional product that is in one type of dispersion, so the nanoemulsion form and the microemulsion form, the chemical stability, so whether or not the phytonadione is degrading in the presence of UV light, is something that can be controlled for. The physical stability of a microemulsion is extremely high because it's the thermodynamically favored form of the dispersion. So things like heating and cooling or freeze thaw tend to not have a very large impact product. And for the nanoemulsion, what happens is it basically, because it's not the thermodynamically favored state of the system, when you do those kind of stability testings, they just become microemulsions. So in general, the stability is basically that the thermodynamically favored state of the system, depending on the composition, is what comes out. So if you made a different composition, you could get it to be destabilized because you have too much oil in there. But at the composition, the formulation composition of that product, when you undergo stability testing, it tends to just form into the microemulsion state. Thank you for that.
Thank you for responding to that question. We're moving on to the next panelist. We do have a few questions that came in for Dr. Hee-Sun Chung. And here's the first question for Dr. Chung. Why is FDA not including the in vitro option as part of the phytonadione product-specific guidance? And are there any challenges in the in vitro option to achieve bioequivalence?
Thank you for that question. I'm going to provide more general response here. And it is that if you want to propose an alternative B approach than what is currently recommended in the PSG, you always have the option to approach the agency with your proposal. If you're at the pre-ANDA stage, then you can consider pre-ANDA product development meeting, for example. And if you're qualified, there's also control approach in which you can also approach the agency in that way. And so I leave it there. Thank you.
Thank you, Dr. Chung. We do have a few more questions here, and here's the next question for these dosage forms. What are some suggestions to ensure a complete ANDA package? Thank you for that question. So this is an area, especially with these complex dosage forms, oftentimes we run into incomplete information within the submission, and we may end up asking for additional information, sometimes through information request, if it's a very straightforward information. If not,
we may end up issuing a discipline review letter, and this may potentially increase the review cycle. So some suggestions in this regard are, for example, make sure that your method validation report is complete. And in general, especially when you're dealing with the study to demonstrate bioequivalence, then you want to use the reference product to validate your method. Of course, there's a certain experiments, for example, selectivity, where you may need to use a modified form of the test product, and inherently, then you cannot use a reference product for that validation. But in general, do use the reference product to demonstrate that method is validated for this drug product. Another is be sure to submit the method development reports, because oftentimes we don't have a set method to say this method works for this product or not. So it helps us to understand how you ended up with that specific method for the drug product that you're testing. Also, those two documents. Another thing that we run into is that sometimes we see from the difference or discrepancy between the formulation of the test product versus the manufacturing details from the chemistry-related documents. So if we see some discrepancies in that regard, then we may need to reach out to you to clarify that, and we cannot make, for example, Q1, Q2 determination without this clarification. So those are the things that I would watch out for. Another is time to time, some of these drug products may be drug device combination. And if that's the case, then you should provide the comparative analysis results, and you can refer to the guidance, comparative analysis and related comparative use human factors studies for drug device combination products submitted in ANDA. So that's also very important, and you should consider with respect to the presentation of your product, etc. I do want to add to one of the earlier questions regarding the formulation of Q1, Q2 sameness. I think someone asked about the cyclosporine and the pH adjuster difference. In that regard, you may very well be aware that the agency posted a guidance last year related to pH adjuster for the ophthalmic, otic, and parenteral drug products. So if your product has pH adjuster difference compared to the reference listed drug, then you definitely want to look at the guidance closely and what other options are there for your drug product, even if it's not a simple solution, for example. Thank you.
Thank you for responding to that question. We do have another question that came in for Dr. Ben Chen, and here is the question for Dr. Chen. Can I have a meeting or a controlled correspondence if I have an ophthalmic product that is non-Q1, Q2, for example, a pH adjuster difference?
Thank you for the question. Yeah, actually, I'm glad you asked this question. Yes. So the quick answer is yes. The applicant, however, they should discuss with FDA through communication pathways such as control correspondences or pre-ANDA meetings. So in general, an applicant, they may be recommend to do an in vivo B study to adjust this, but we do recommend the applicant should first discuss with FDA. Also, we are aware there is a pH adjuster guidance, right? I mean, the main message we want to mention here is that we recommend the applicant discuss with FDA first, and then to to see what type of study needed and what type of justification needed to submit in the NDAP. Thank you.
Thank you for responding to that question. We have a question that just came in for Dr. Khandakar Alam, and here's the question for Dr. Alam. Can the agency comment on modeling and simulation-based approach for generic drug approval?
Yeah, very important question. I think that this question is relevant for other session as well. So we have seen increased interest in the model-based alternative bioequivalence approach in recent years from prospective and applicants. In many cases, the prospective and applicants proposed model-based virtual bioequivalence in lieu of PSG recommended in vivo BA study. In fact, FDA has approved generic diclofenac sodium topical gel where the end applicant used virtual bioequivalence instead of PSG recommended clinical endpoint study. So model-based generic drug approval is not a fairy tale right now, it is a reality. And other than virtual bioequivalence, model-based approach can also be used to justify the potential difference between test and reference drug product, and to justify that the difference is not going to impact the in vivo performance. For example, when you have the mechanistic model, you can use the model to justify your case that the potential difference in Q3 data of complex systems such as difference in the globule size or viscosity or IVPT profile may not impact the in vivo performance of your test formulation. So agencies open and supportive of model-based alternative approach, but I also want to clarify that model-based alternative bioequivalence approach should not be taken as an opportunity to get around from the regulatory requirements. Model-based approach rather provides an opportunity to overcome the challenges that we have seen or the applicant have seen with the in vivo BA study. So model-based approach still needs to satisfy the requirement of the applicable statutes and regulation that FDA has. We have from the agency, we have presented in different public workshops to share our regulatory thinking on the requirement of model development and validation. So I would suggest the generic applicants to use these resources if they are considering model-based approach. I would also suggest that prospective applicant can interact with the agency early through proper communication channel like pre-ANDA meeting request or CC to get feedback on their model-based approach before they submit the ANDA applications. So hopefully my response is helpful to the audience to some extent. Thank you.
Thank you for responding to that question. Moving on to our next panelist, we do have a question that came in for Dr. Xiaoming Su. And here is the question for Dr. Su. What are the alternate approaches to determine the drug distribution of the active drug in the nano emulsion?
Yeah, thank you for the question. It's a good one, and there's always alternative approach. So as Dr. Fan mentioned in his talk, that NMR is one methodology that can be used to measure the drug distribution in emulsions. But of course, there are other approaches that can also be used, for example, dialysis and centrifugation, or even ultrafiltration. So as long as the globule portion of the product can be separate from the non-globule portion of the product, and that you can actually analyze the content in each portion, and then by definition, you are measuring the distribution of the drug in different portions. So, but of course, we have to recognize that the methodology, each methodology has its own limitations. Not every technique is necessarily suitable for or has its own limitations. We need to recognize as part of the development and validation, need to be really careful in considering how to demonstrate that method is suitable to demonstrate the intended purpose. Focus on the reproducibility, focus on the precision. It's going to be very critical. But other than what's mentioned in the talk as an MR certainly the other few can also be utilized to support the demonstration of the bioequivalence. Thank you.
Thank you, Dr. Zhu. We're going to try to squeeze in one more last question that came in just right now for you. In a few of the presentations, it was mentioned some work done by FDA or sponsored by the FDA. Can we use a different method to support our application?
Thank you for the question again. Different methods can be used. I think one important perhaps takeaway, if you've seen a lot of FDA's work or the contract or grants sponsored by the FDA, is we're really trying to providing alternative pathways or conducting research to fill the gaps. And some of the published work really is intended to share what we have accumulated understanding to the public, to the industry, so that you may adopt some of the method that has been developed. But by no means those are the method that has to be used or the only method that can be used. So certainly if there are alternative method that can also serve its purpose, certainly we are welcome to submit. I think as Hisam mentioned earlier, that there are different pathways that you can communicate with the agency through pre-ENDA, through controlled correspondence pathways. So all of that can be very helpful. So, but of course, our research work, whether it's done at FDA or sponsored by the FDA through grant, it's through GDUFA research. So it's paid by the industry. So it would be beneficial for you to consider looking into that and to see how maybe beneficial to adopt in your development program. But thank you very much.
Well, that's all the time we have for questions in this final panel of the day. A huge thank you to our presenters and panelists for answering numerous questions that came in from our audience. We'd like to welcome Dr. Lei Zhang, Deputy Director of Office of Research and Centers and OGD, to provide some closing remarks from day one. Welcome, Dr. Zhang.
Thank you, Ray. Good afternoon, good evening, and good morning to all the participants across the world. I hope that you, like me, enjoyed every session of today's workshop. Time just goes by so fast. This is the 6th year that we hold this workshop. We clearly see a continued interest from our stakeholders in this workshop. We have more than 3,000 people registered from 86 countries. Today, we covered several types of complex products, including topical and transdermal products, nasal suspension and inhalation products, complex injectables, products with complex APIs, and more. We learned how research support PSG and general guidance development, and eventually ENDA approvals. The scientific research founded by Generic Drug User Fee Amendments, or GDUFA program, builds an essential bridge from the scientific knowledge of the modern era. Science and research are critical to modernizing the generic drug program and the generic drug industry to continue to supply safe and effective generic drugs to the American public. As reflected by the title of this workshop, we are elucidating how we translate science to approvals, better translate some of the complicated science behind generic drug development into more generic drug approvals. Now we are close to the end of the first year of the GDUFA III. We already know there are a number of enhancements under GDUFA III that have been implemented to further enhance efficiency, transparency, and gain more first cycle approvals. These include PSG enhancement on new goals for newly approved complex products, options to request PSG-related teleconferences and meetings, and additional scientific meetings with industry both prior to and post-submission, so that applicants can communicate with the FDA in a timely manner throughout the product development cycle for input on the development program to improve efficiency and enable better ENDA submission or resubmission. Enhancements also include the suitability petition process, including the establishment of new suitability petition goals, which will begin next month in fiscal year 2024. Tomorrow, we will have another array of exciting topics, including additional complex products, such as orally inhaled products and locally acting GI drug products. In addition, the last two sessions of tomorrow will cover enhanced processes, GDUFA research and assessment tools to support generic drug development, and also global collaborations to support efficient generic development and regulatory assessment. A new pilot industry meeting program will be presented. You do not want to miss it. Before I end, I would like to thank the session leads, speakers, panelists, and the subject matter experts who worked diligently together to design and deliver the content for this workshop, both today and tomorrow. I also would like to thank FDA Commissioner Dr. Califf for delivering the keynote speech this morning, and always our SBIA colleagues, Brenda, Ray, and others in moderating the sessions and coordinating many behind-the-scenes efforts. The breadth and the thoughtfulness of information provided by our speakers and additional discussion and clarity provided by speakers and panelists and the Q&A sessions are enormous. Thank you for your participation of day one of this workshop. I look forward to your attendance of day two of the workshop tomorrow. Day one of this workshop is now adjourned. Thank you. And now I would like to turn back to Ray. Thank you all.
Thank you, Dr. Zhang, for those exciting closing remarks for day one. We do have a few reminders for our attendees. Please remember the 14 days deadline for the certificate of attendance and the CEs for healthcare professionals. Remember, the deadline is September the 28th. Recordings will be posted in approximately 7 business days. On behalf of Cedar Small Business and Industry Assistance, we hope that you enjoyed today's presentations and found them helpful. We look forward to your presence at day 2 of AGDD starting at 9 a.m. Eastern Time. Thank you for attending, and this concludes today's event.